Thank you, ma'am, for introducing me. Respected all staying here. Very good morning to all of you. Basically, yesterday I attended all the sessions, and this was very informative regarding the lectures that are delivered by the dignitaries, especially the welcome note from our respected DG ICAR and Secretary there, Dr. Mahapatra sir, regarding the sustainability of agriculture. Basically, this topic, I am working on bees from last 20 years in Kashmir. As you know, you all are aware about the conditions of Kashmir. We are from temperate region. When I came here, we have a different culture here. Our culture is totally different from rest part of the India. But we are working under the same umbrella. What we are working here in Kashmir, this is very important to interact with the scientists from the other parts of the country, what they have done. Basically, working from last 20 years on bees as my home level. I am an apiculture born scientist. I am an agriculturist. I am a horticulturist. I have my own orchard, apple orchards, apricot, dry fruits, each and everything. But it is mandatory to share my views to the audience, to the people coming from different parts of the country. Basically, this is a burning issue in Kashmir, so far the apple is concerned. Because our 70 to 80% of the economy comes from apple. And apple without pollination by the indigenous honeybee, apple Sinana is incomplete. Due to lost, as you know, apple is our main cash crop, and 70 to 80% of our economy stands on apple. As you know, apple is incompatible crop. Incompatible means the male pollen of this flower cannot fertilize the same female part. It's a self-incompatible. That is why we need the pollinators to transfer the pollen from the male part to the female part. And you will see that under the cultivation, we have 1.5 lakh hectares under apple cultivation. Out of 3.3 lakh hectares of total temperate fruits. And recently in 2021, we get the 20.3 lakh metric tons of apple, which becomes the 60 to 70% from the state, out of 100% from the country, 70% of the state. As you know, I have told this apple is incomplete without the pollination of honeybees. Basically, it relies 100% pollination. It is a false fruit. It is a cross-pollinated plant. It is not just the stone fruits. We have we have these true fruits. There is now no need of a pollination. But when we talk about the apple, it is totally reliant on the honeybees. And our indigenous honeybee, Apis cerana, from last 10 to 20 years, it becomes a threatened species due to some due to some constraints. Because climatic changes at its peak, we have a disturbance, we have an urbanization, just like the other parts of the country. We have an urbanization in the state. We have a destruction of flora. We have a conversion of land from agriculture to horticulture and some other word. We have an intensification of agriculture. That is why this species, it totally migrates towards the higher altitudes. And the plain areas where we are growing apple, we have a dire need of these species. because we have also the exotic species Apis mellifera, but most of the beekeepers, they are migrating their colonies towards the other pods because of the low temperature during winter. And at the time of flowering, just after 15th of March, our apple flowering starts. But none of the beekeeper is present in this time in valley. That is why we are talking the Apis cerana. This is a feral bee. We are not migrating this Apis cerana towards the other pods. That is why I have some innovative ideas how to conserve these species at Kashmir conditions. You will see the taxonomic biodiversity of this honeybee Apis cerana in Kashmir, as I have already talked, that there are so many constraints which affect the diversity of Apis cerana. This basically we have a destruction of natural habitat. As you know, there is a lot of flowers which provide nectar as well as pollen to these species. There is a lot of disturbances. I have some photographs also in the next slide. Second is inclement weather conditions. As you know, we have a unexpected rainfall, unexpected snowfall in different seasons. As you can't imagine that we get the snowfall at 30th of, I was talking with the ma'am, that 30th of May, 30th of April, we get the snowfall in 2003. You can't imagine. In ending April, you get the snowfall. And what will be the weather conditions that time? It was the development stage 2nd of April when we get this snowfall. And you can't imagine how much loss our orchardists, these orchardists have occurred in their apple orchards due to the heavy snowfall. Next is insect pests and disease. As you know, this indigenous species heavily affected by some insects and some diseases. As you know, wax moth is an important insect. So far this Apis cerana is concerned, because we are rearing this Apis cerana in the indigenous hives, that is mud hives, indigenous species in the mud hives. Basically, this is local species. We are not rearing in the Langstroth hive. Basically, we have some disadvantage of this species. It is only surviving at the higher altitudes, and because of their absconding behavior and some other swarming conditions, it is very difficult to rear in the plain areas. But for the pollination purpose, we are in a position now. We are in a position to utilize the services of this Apis cerana for pollination of apple and other temperate crops. Another thing is indiscriminate use of pesticides. As you know, we are giving 10 to 15 sprays in apple because the pest load is very high. We have a lot of pests, including Sanjo scale, woolly aphid, green leaf aphid, and some types of mites. We are affecting this apple, but we are spraying a lot of insecticides, a lot of fungicides on apple. Due to this indiscriminate use, this species has been totally affected in the plain areas where we are in a use. Next is agriculture intensification. Agriculture intensification, as you know, we are going towards the monocropping system. We have not the diversified agriculture system in Kashmir. Our agriculture land is totally converted into horticulture land. Now, if we talk about our village, I am residing in a village. Our village has just 150 household families, but all the household families, we have a horticulture land. We have not the agriculture land. That means our village is totally goes towards the monoculture system. This becomes one of the threat for this bee species for their survival. Next is important point that I came here because of this point, because as you know, this apple industry goes and comes bring a collapse because of this threat for this apis cerana. As you know, you see there is a destruction of habitat in the forest areas. The apple, this livestock grazing is there, destruction of forests are there. You will see the fires in the forests. As you need the plant invasion, there are so many plants which are not giving the pollen and nectar to the bees. They are invaded to the plants which provide nectar and bees to the nectar and pollen to the bees. And also encroachment of forest land by the household family. There is a population is rising day by day. There is encroachment of land in the natural habitat. Next, you will see that this, I have already talked, the indiscriminate use of pesticides. This is one of the important threat for this bee species for their diversity. This is next, as you see, insect pest and disease. The foul brood disease for this apis cerana. This is very important. Foul brood disease is one of the important threat for this bee species, and also wax moth, because when we rear this bee species in the mud hives, in the local hives, there is very difficult to transfer this comb to the Langstroth hive. Basically, this is shifted. I have some photographs of the local beehives also, but this comb is totally shifted on the top floor of this hive. But it is very difficult to shift. The sanitation for this hive is very difficult. So this species under these mud hives, this is very prone to this wax moth. Now you will see the inclement weather conditions. We have a snowfall, we have an unpredicted rainfall. That is why this species is totally disturbed. If you see, we have worker bees from last 25 years, and this presentation, as you saw, we have investigated, carried out this research from last 2 years in the valley regarding the colony losses. We have collected the data from the Indian Meteorological Department body regarding the climatic from last 20 years. We have collected the data from the Horticulture Department regarding the conversion of land, from also from the Agriculture Department regarding the conversion of land from agriculture to horticulture and some other crops. We have also collected data from the Forestry Department, how much forest, how much these encroachment is occurs towards this forest area. And out of these, you will see 1 to 2% of the temperature has been rises from 20 years. 20 years in Kashmir. So far, the different climatic zones are concerned. We have a different climatic zones in Kashmir, but 1 to 2% of this temperature has been rise. This becomes also the important threat for this species to survive in the plain areas. Next thing is untimely snowfall, and I have already discussed this. Data regarding the pesticidal, I have already collected the data. What are the different pesticides since 2000? Different pesticides we are spraying by the orchardists in their orchards, organochlorines, organophosphates, carbamates, neonicotinoids, all these are data, but this is a technology. I have mentioned technology, I have not mentioned all the data. This is unpublished data, and I have submitted it as a patent for the conservation of this local honeybee species. And also revenue record from the Revenue Department regarding the conversion of land. Also the insect pest diseases. And last thing, this is important, the preparation of bee model. The preparation of bee model hive was carried at the different material. We have collected the material from some of the material, imported material. We have prepared one of the important bee hive. This is also a mud hive, but I have modified this hive in such a way that we can transfer this colony to the another Langstroth hive. This is totally this is shifted. We can shift this colony towards the another hive. And one thing important, we can sanitize, we can clean this hive easily. There is not any chance of the wax moth. There is not any chance. We can feed the hive in this modified hive easily. We can give the sugar feeding. We can clean the hive. we can shift this colony in the another hive also. You will see these are the mud hives. Basically, this is feral bee. These are the mud hives locally for the pollination purpose in the apple. But you will see, as in this photograph, this is you will see the colony inside this hive. It is very difficult to shift in the another hive. Basically, this is totally fixed in the hive, and this hive is totally open on the back side. When I have make the model. In the new model, we closed the hive in the front as well as in the back, but it is opened on the top. We have a window type. We can remove the frames and we can close. And this model is also made up of mud, but we prepare with some imported materials. We can preserve the heat in the model hive for 3 to 4 degrees as compared to the old hive. That is why we are going to conserve this species. Investigation have been carried out in the 5 districts of the Kashmir. Anantnag, district, I am belonging from the Anantnag, Shupiyan, Pulwama, Badgam and Srinagar. As you see in this study, we have worked on the 2 aspects. First is bee mortality during the sub-zero temperature during winter. We have worked on these. What is the actual mortality that is happening in the local mud hive during the winter? And what is the mortality happening in the new model that I have prepared? During the study, we have studied that 30.5% of the mortality occurred in the old hives, 30.5%, which includes 8.3% by the destruction of forest, different natural habitat, 6.2% by inclement weather conditions, and 6.8% indiscriminate use of pesticides, and 3.4% by the agriculture intensification. But by this model, by the preparation of the model, we can reduce the mortality rate of this Apocyrana from 17.5% to 2%. Because when we have observed from the lot of honeybees, we have this colonies has been monitored from the different districts, and we have observed in the higher altitudes 17.5% of the colonies have been totally died due to the winter conditions of the Kashmir. But when I have laid the trial in the modified hive, there is only 2% of the mortality, but there is not the complete death of the colony inside this modified hive. Only few bees are died, because there is the age factor also. The age factor of the bees is also concerned. That means we are not losing the whole colony during the winter in the modified hive. You will see these are different. I have mentioned only one slide because of the unpublished data and submission of this technology and patent in the university. You will see only the major portion is destruction of natural habitat, loss of flora, and followed by the agriculture intensification. And you will see the mortality in the mud hive compared to this modified mud hive compared to the normal mud hive. Novelty. What is the novelty of why I have prepared this model? As I have already talked, this is only for the 2 parameters. First is to reduce the mortality during winter. Basically, we are not migrating this Apocyrana to the other parts of the country. Second thing is to enhance the breeding of this species, because the breeding will stop at the end of November. But when we rear this species in the modified mud hive, this will extend towards the end of the December. When we get the brood, when we get the fresh bees till the end of the December, we will get the strong colony of the upcoming season for the pollination of apple. The conclusion, basically it is only the 28 seconds. The conclusion will get the conclusion in the way that this climatic change in the Kashmir, climatic change is everywhere, but our state is much more affected by this climatic change, as this affect this apple industry as well as this apple serrana from last few decades. Second point is utilization, because I already said that this apple industry is incomplete without the pollination services. Utilization of this apple serrana for the pollination services of apple. And third thing is to reduce the loss of this honeybee by applying this application of this modified hive. And last point is very important, that conservation efforts also need to be reinforced, and ensuring that endangered population will be protected from uncontrolled introduction of imported strains to keep them available for the future generations. That is all about the presentation. Thank you very much. Thank you.
Thank you so much, sir. That was very detailed and well explained. Now, I would insist Dr. Sutar Suhas Bharat, Assistant Professor from Centurion University of Technology and Management, to come on this podium and talk about the topic Oxford Nanopore Sequencing Technology and its application. Let's have a wonderful round of applause for the gentleman. Yes, thank you so much.
Hello. Yeah. Good morning, all. First of all, I would like to thank organizer for giving me the opportunity to present my work here. So I'm Dr. Suhas from CUTM, and today I'm going to talk on Oxford Nanopore sequencing and its application. So before moving to our core topic, some basics. Yeah. So the question here, because the science start with the questions. In fact, the life start with questions, and we are working to get the answers. So the question here is why we are the way we are, why we have the skin color, the fair color, dark color, and why there is a height difference, means phenotypic differences are there, why there are human diseases, and how we get evolved. So these are the questions. This is our big picture, but we are taking small steps to, yes sir, yeah, and we are taking tiny steps to reach to the answer. So in this all these pictures, the central dogma works very well. The central dogma is like, we know that central dogma is the DNA transcribed into RNA, and RNA translate into proteins, and that proteins actually works in the body, and that is the proteins are responsible for the phenotypes. So this is central dogma of our life. And the again question is here, my whole genome is everything about my phenotypes? That is no. Then what is phenotype? Phenotype is actually genotype interact with environment, and not only environment but interact with management also. It means if you have the fair skin or dark skin, if you are high in height or lower height, it totally depends on three factors, that is genotype plus environment, genotype into environment into management. So how to study the DNA? Because here we are focusing on the genotypes. So if you want to study the genotype, then we have to study the DNA. And how to study the DNA? DNA is initially we have to extract the DNA, sequence the DNA, then do the sequence analysis, and after sequencing analysis we have to retrieve the biological important information from that DNA, because when we sequence the DNA we just have the ATGC sequences. We don't have any straightforward information in that, so we have to decode the DNA. We have to get the biological important information, and that information we can use for our further research. So here is the history about genome sequencing technologies. We start with Sanger method, and now we have the cutting edge nanopore technology. So I am totally focusing on the nanopore now. So nanopore actually, its overarching aim is to make sequencing easy. It means actually we don't need the expertise in molecular biology or bioinformatics. So if you have the basic knowledge about molecular biology and bioinformatics, we can go for the nanopore. So here is the statement that anything by anyone, anywhere. So this statement tells a lot of information. First part of the statement, anything, means here I would like to say, or this statement says that if you have the big genome to study, it means plant genome or human genome. For that, there are special machines or there are special devices in the nanopore sequencing. So that is Promethean. It is a larger end machine, and in one go, in one run, we can sequence up to the 7 TB of the data. This is for big genomes. And if you have the small genomes like bacteria, viruses, there are small machines also available. That is the fungal or mini ion. We can sequence up to 2 to 3 GB data at one go. So nanopore has range of products or range of machines or devices we can use for our projects. So whatever projects you are designing, nanopore is fit for you. Then anyone, actually here it is a commitment that nanopore sequences has made very easy protocols and very how to say, a very simple way to do the sequencing. So here and anywhere means, nowadays if you want to sequence means before 5 years or 10 years, if you want to sequence means we have to extract the genomic material and we have to send it to the place where they can be sequenced. But nowadays we can bring the sequencing machines to the on the spot, means in silico, in vivo we can do the sequencing. Suppose, and this is the first machine they have done sequencing in the space also. So this is means there is a huge advantage of this technology. So here is the range of products. The first one product is this is the fungal. Then we have the mini ion, then grid ion, and then Promethean. That Promethean has capacity of give the 7 TB of the data at one run. And this actually currently we have this mini ion in our laboratory and we are working on it. And it is actually half of your mobile size, so it can be fit into a pocket. So what is Oxford nanopore sequencing? As in picture you see, they have array of microscaffold proteins. Actually, nanopores are the proteins, and we fix the proteins on the glass slides. And there is array of around 2048 pores, and we pass the current through the slides, and that machine works and gives the sequence one by one. Yeah. So here we have the different nanopore means nanopore material, that is polymer. We have two types of nanopore materials, that is biological nanopores and solid state nanopores. In biological nanopores we have the protein hemolysin, and in solid state nanopore we use the material that is silicon dioxide, and we make a pore into silicon dioxide, and through this DNA passes and we get the sequences, a sequence of the DNA. Yeah. So here is the principle. I think in the 9th standard or 12th standard we have studied that if we pass the current through the glass chamber which contains the NaCl, then we see the ion movement. But we place the plasma membrane across this glass membrane, then we don't get any current or any ion movement in that. But if we insert in that assembly, if we insert the nanopore, then we get the current flow. And in this first picture, we can say here in this picture, Actually, here we have the plasma membrane and we inserted the nanopores here. And here current is flow continuously. But when we insert some object into the nanopore, we get the disturbance in the current. And the current disturbance depends on the size of the size of the material, size of the molecule. And if it is a size of the molecule is small in size, means comparatively to compared to other picture, then we will get small disturbance. And if the size is the bigger, then we can get the big disturbance. So this all these disturbances in the current get recorded. And if we this is very means if we insert one by one molecule, then we will get pictures separately. But in case of DNA, it's a long stretch of ATGC, and DNA continuously flows through the nanopores. So we will get the continuous peak like this. And this cannot be read by human beings, or this is not readable to us. So we have to use some different how to say softwares for that. So that is all about the electronics of this nanopore. And here we have the chemistry, because DNA, when we sequence the DNA, we have to adapt, we have to attach the adapters to the DNA and motor proteins. That motor proteins and DNA and adapter, this assembly we have to load onto the nanopore sequencer machine, and that will go and find the nanopores. And after that, actually this is the picture of the nanopore, and here is the tether, and it will attach with the motor protein and bring to the nanopore, and then DNA gets start passing through the nanopore, and simultaneously it will give the sequence to you. Here, when we sequence, that time we get all the electric peaks, means electric current peaks, and that cannot be readable. And so we have to change that FASTA file, FASTQ file into the FAST5, because that is the electric current file, and we have to change into the ATGC. So we use Guppy software for that, and that software does all these things. And then we have to then we go for the data analysis, and for data analysis, nanopore sequencing company has provided the epitome, and there is a dealer in our India, that is Genotypic company. They have produced the one commander software, and that they can do all this analysis. Means how to say, means actually we don't need any expertise in bioinformatics. If you have the basic knowledge about mole bio and some computer knowledge, then you can go for this data analysis also. And after data analysis, there is a primary analysis, and there is a deep analysis also. If you want to do deep analysis, then you have to have some aims about your projects, and that for that we have different pipelines. So here is the general methodology. If you start the DNA study, then we have to start with DNA extraction and purification. Then we go for the library preparation. The third step here is the nanopore sequencing by using mini-ion, grid-ion, ProMethod, depends on your projects. And lastly, we have to do the DNA sequencing analysis by epitome, commander, or there are different ways, or there are different pipelines to do this. So here are some applications. The nanopore has played key role or very important role in outbreaks, because in corona outbreaks in 2020, the 400 machines are exported to the China from the UK. So nanopore has a huge role in outbreaks. Also, nanopore worked in very well worked in human genetics. Then we can use it for transcriptome analysis, environmental analysis, and plant genome sequencing also. So here is another one, the threat we are inviting. So that is AMR, that is antimicrobial resistance. In coming days, the more people die because of this, because this is an emerging threat, and it is gonna very worse. It is going to be very worse than cancer. So we should be ready for it. And nanopore will work very well, because it has a quick identification facility in nanopore. So the AMR is also a good application. Then food safety. Yeah, we have the plenty of processed food in our diet daily. So for example, rasgulla, and there is a QC control, or there is a quality control department in every company or in every industry, and they directly controls their products after formation or after manufacturing. But if you process, or if you check all this, or if we monitor all this products at their initial stage, I mean the raw material, then we can save the millions of dollar, million or billions of dollar there, because we do no need to check the whole range of the products. If you check initial products, that's all, we can go for the processing. So in this way, we can save a lot of money. And the next and very important application of this nanopower is to detect the variants in the cancer, not only in cancer, but in different molecular biological study. If you have the gene deletion or gene insertion, or there are some mutations in there or translocation, but with the small reads, means nowadays we have actually, there are many other sequencing machines are available in the market, that is Illumina and all, but they gives very short reads, and with the short reads we cannot identify the deletion and all. So nanopore is the big solution for this. So already I discussed about this outbreak story of this nanopore. Why it is so important? Because it is easy to distribute. We can carry it in pocket, and if you have the one laptop and small mini machine, we can start the sequencing anywhere, and we can take it to the outbreak spot. Means no need to take the samples out of the area and have the risk of contamination at other place. So we can take this machine to the spot and we can start the sequencing. So, and it is have the rapid results. Means actually we have done viral sequencing and bacterial sequencing. It takes only 1 day for us, because in daytime we prepare the library and night time we run the sequencing, and next morning we got the data. That's all. So actually, last time we completed bacterial genome sequencing and we got about 1.2 GB data within 12 hours. So that is the, I would say, advantage of these technologies. So nanopore sequencing at our lab, means my lab. My lab means obviously Dr. Rukmini Mishra is leading us, and till now we have completed standard lambda DNA sequencing. Then we have completed bacterial whole genome sequencing, and in future we have the plans to go for the fungal genome sequencing, metagenomics, RNA sequencing in tomatoes, and antimicrobial resistance testing. Here are some clicks from my laboratory. In this picture, my team is there, and there my students are working on the nanopore. So it's quite good technology. Yeah, and these are my lab members. Yeah, first one is Dr. Rukmini Mishra. She is our group leader, and then we have the 3 PhD students. Debasmita Das, PhD student, she is working on market development. Then Sunanya Das, she is working on medicinal plants. Then Sonu Priya Sahu, she is working on genome editing in tomato. And last one is Rashmi Ranjan Sutar. He is JRF in our laboratory, and he is working on genome editing in Chile. So these. Next, our collaborators, we have collaboration with IRI. Dr. Amol Solunke from the IRI, he is connected with us for the tomato work. Then NRRI, then Onion Garlic Research Center, Pune. Dr. Yogesh Khade is working with us. Then next, Ramadevi, from Ramadevi, Dr. Rajkumar Joshi is working with us. From TNU, actually is my master college. So I am connected with my supervisor for means guidance and all. And CAS, here, the Chinese Academy of Agricultural Science, where I complete my PhD, and my professor is working on genome editing, and so we are collaborating with them for our projects. And last one is CDFD for data analysis or some other genomic sequence facilities. We are working with them. So this is all about our collaborators. Thank you. Thank you so much. Okay, na? Yeah, thank you.
Thank you so much, sir, for finishing on time. Proceeding further, let's have Dr. Rukmini Mishra, HOD Department of Botany, Centurion University of Technology and Management. She'll be talking on CRISPR, Cas9 genome editing towards enhanced disease resistance in chili pepper. Let's have her on stage with a wonderful round of applause. Thank you.
Hello.
Hello. Yeah. A very good morning to all of you, respected delegates from different part of India and outside, and all the students present over here. So I must thank the organizers for giving me an opportunity to present my research today. So it really feels great after a long time we are physically meeting, and that's really a good thing. So before starting my research topic, I just want to say that yesterday also we had few good talks, and regarding CRISPR-Cas9, already Dr. Kutubuddin Molla, he has explained, he has given a very basic this understanding about what is CRISPR and what is the technology and what are the applications we can do. So I won't be going in very detail, but rather I will be explaining what is the application of this technology, how we can use CRISPR-Cas9 technology towards disease resistance. Particularly, I'll be focusing on disease resistance today. So in our lab, we are working on chili and tomato, but today I'll be explaining how CRISPR-Cas9 technology is used in chili pepper towards fungal disease resistance. Okay, so here I'll be starting my topic. So before moving, I'll just give a very brief idea about what is exactly anthracnose. So anthracnose is a fungal disease in Chile. So what it does is, when under a favorable condition, when the pathogen is coming and colonizing, it enters into the plant through the host epidermis, and it forms this concentric rings of acebuli. So, and these symptoms, what they do is like they reduce the market value of the crop. So it almost leads to more than 30% yield loss in India, and almost 40 to 50% yield loss all over in particularly in Odisha also we have lost lots of yield loss. So the causal organism, it's not a single species. It is called caused by multiple species. Coleotrichum truncatum and gliosporids are mainly prominent in India. Also, it is caused by acutatum. So these are a complex of species that cause this disease. So next thing is what is important is what are the strategies that we apply or we take up to fight the disease or against the disease, what we do. Basically, the first method that we prefer is using the biocontrol agents, or we go for some fungicides and all, but we don't see that much noticeable change, or it's for a short time. We cannot have a very it doesn't bring that much change. But anyways, that is one method what we can do. The second thing, which is really very much appreciable, or we feel that it brings a change, is the host resistance breeding. Now, when I say host resistant breeding, it means that we search for some resistant source in the plant itself. Like if it is having some resistant source, then why not get those or screen the germplasm and then put it in the breeding method for enhancing the productivity. But again, that the problem with that also, even though it is a widely acceptable technique, but the problem is that we don't receive, till now we haven't received any tangible success in that. But still research is going on in that area also. But with all those new technologies and new methods, we as researchers, we keep on seeing that how we can get some tangible success in a short time, and we keep on exploring new things. So that's the because of that curiosity, we thought of doing a comparative analysis of the resistant and susceptible varieties. So we took and we did a comparative analysis. So there we found that, see, a lot of transcription factors and the phytohormones and also the secondary metabolites there. So they play a great role. A lot of genes are responsible. But to our surprise, we found that this ERF transcription factor, or the ethylene responsive factor, and this NAC72, it was playing as a negative regulator. Okay, it was whenever there was a resistance or susceptibility, these factors were playing as a negative regulator. So we were not confirmed. It was just an initial study. So for confirmation, what we did is we went for some overexpression and VIX silencing studies so that we can confirm that whether this ERF28 is actually acting as a negative regulator. So that's the reason we went for some overexpression studies by using agrobacter method and also the VIX by using the TRV, the tobacco rattle virus vector system. And similarly, after what we saw that when we did the overexpression studies and the VIX silencing, we found that this calcium ERF28, it was not, it was highly expressed in the overexpression system, but in VIX, we saw that there was not much effect. So in the same thing, the fruits which were taken from the overexpressed line, there was, you can see the symptoms, the anthracnose symptoms, it was highly expressed. But whereas in the VIX system, it was not present, like, it was not expressed. So that's how we conclude that this ERF28 is acting as a negative regulator. So now what we thought of doing is, if this ERF factor is acting as a negative regulator, then why not go for editing, as we know that it's a very like recent advanced technology where we can go for very specific modification or targeted sites. So that's how I will not go into the detail. We know that this CRISPR system, among all the sequence specific nucleases, CRISPR system is now widely used, and it's a very specific and Cas9 will cut down. So once the cutting has been done and basically the cell, it has its own repair mechanism. So it will repair that, and it can happen either through non-homologous end joining, which is a very natural method, but it's an error-prone method because the insertion and deletions happen, that is the mutagenesis. And then we have the more precise one, precise method, that is a homologous recombination. Okay, there you can go for gene replacement, you can go for a specific piece of DNA, or you can go for single base change also, but that is more precise method. So these are the types of repairing methods that we go for. So that's the reason we have used CRISPR technology here. So I'll just give a few brief ideas like what exactly this CRISPR is. So for the students who are present over here, basically this is a defense system in bacteria. So bacteria, what it does, it's very cunning all the time. So what it does, whenever it is affected with any foreign entities like any virus or bacteriophages, and it just comes and bind here. So when they infect the bacteria, every time they leave a DNA, like a segment of DNA is present, which the bacteria keeps it as a memory chip kind of thing. You can see that the CRISPR array, I cannot move here, I think. So I'll just say there is an array, or there are repeats are there. So these DNA, they are stored in that spacer. You can see that it stores that whatever virus or bacteriophages are infecting, it is storing that as a memory chip. So when transcription happens, Cas locus, it will form Cas protein, and the CRISPR array, it will form pre-CRISPR RNA and the stressor, and then that will form a complex. This stressor, CRISPR RNA, and the Cas cascade, you can see that. So what happens whenever that particular virus or bacteria phase, again it will infect the bacteria, it will store it as a memory, it will recognize that, and it will cut it. Okay. So that's how the bacteria defends itself from the foreign entities. That's the mechanism. It's in a bacterial defense system. But what we have done is, we have taken the idea, and people like Jennifer Doudna and Emmanuel Carpentier, who got the Nobel Prize in 2020 for this discovery, what they did, they have used that defense system as an editing tool. Okay, so that's the same thing we are also doing. If we have any target region and we want to go for any modification, so we designed that guide RNA, which is a synthetic RNA, whatever reason we want to target, we go for that 20 base pair synthetic guide RNA. Along with that, anyways, we have the Cas9. So it forms as a complex we design, we put it in a single vector, and we transfer to the plant. And once it goes inside the plant, it will integrate into the chromosomal DNA, and then it will go for that specific editing. Okay. But usually the CRISPR system, it acts as a dual promoter system. One Pol 2 promoter is used for Cas9, and the guide RNA is basically followed by a Pol 3 promoter. But here in our research, we have used a single transcript CRISPR Cas9 system, where a single promoter, that Pol 2 promoter, is only activating like Cas protein and the guide RNA. So as a single unit, it is transcribing. So, but if it is like a single unit, we cannot, because it's a protein and that will stay as a RNA. That's the reason they have also put that ribozyme cleavage site. So once it is transcribed as a single unit, then after cleaving, Cas9 protein will separate it, and the guide RNA will be separate as a separate entity, it will stay. So this was done by Professor Daniel Voitas in their lab. So it's a very good system. Instead of having both the promoters, it's not required. In some non-model organism, you will find that you don't have this U3 or U6 promoter. So in that case, you can use this single transcript system. So we also thought of using that same thing, and also we found good results for that. So before starting, we designed two vectors, two guide RNAs. You can see that is the ERF28 sequence. So one guide RNA would have been enough, but it's because we were starting, so we thought it may not work. So we designed two different vectors. You can see the Cas9 protein and the guide RNA, and the whole vector construction, like vectors we got from Agri, and we were supposed to add the guide RNA region into that. So these were the two different vectors that we designed. And this is transformation and regeneration. So these results you can see the transgenic plants that we got in out of 35 we got 26 edited lines, and all these categories like different mutant genotypes, chimera, biallelic, homozygotes and heterozygotes. It's because it's a natural NHEism method, so you don't expect any single type of mutant. You will obviously get homozygotes, heterozygotes and biallelic and chimeric, but our concern is that we must go for homozygotes so that we can proceed with the further results. So these are the different allelic types of mutations that we got. Now, what is very important is that we say that it's not transgenic or it's not GMO, because we have to go for the tDNA free plants. Otherwise, there is no point. So for that reason, we designed two primers, forward and reverse primer in the region of Cas9. And then we selected the plants that we got, the edited plants. We extracted the DNA and we run with those primers, and we found, if you can see, somewhere wherever there is no band, those are edited lines, but that is the free from tDNA region. Okay, those are Cas9 free plants. So we have to select those plants for further analysis. Okay. So this is the assay where we have done just to see that editing has been done for confirmation. You can see that wild type is not having any multiple bands, but the mutated lines are having multiple bands. This is just to confirm that editing has been done. Okay. And this is a phenotypic expression. Just we have taken the fruits from the edited lines, the mutant lines, and we have inoculated them with the pathogen. And you can see there is, in the wild type, there are symptoms like a disease is happening, but there you don't see any symptoms. Just for a phenotypic confirmation that editing the mutant lines, they don't have the fungus. Okay. So these are the conclusions that definitely if you we have done the targeted mutation for ERF and it has enhanced the resistance ability. Actually, the work is still going on. Recently, we have also received a project from DST to carry out this research, and also Dr. Pushpalata madam, she is here. She is one of the co-PI in that. So we will be continuing that research, and hopefully we will go for a broad spectrum resistance in chili. So besides that, I would like to thank my team. They are all here. Today they will be presenting their posters also. And Professor Zhao, Keijun Zhao, I was doing my postdoc there in Chinese Academy of Agricultural Sciences, Beijing. And Dr. Joshi, he is from Ramadevi University. Long time I have been associated with him. And Dr. Pushpalata madam, she is from our university. So we are all working together. And I would like to take the privilege of this platform at least to say the students that we have initiated this Synomics in our lab. It's an initiative from our lab. We are giving hands-on training for different molecular biology experiments, also gene editing and gene sequencing. So soon from April first week, we will be starting these small workshops. So you people can contact and you can just enroll yourself. So thank you. Thank you so much.
Yes, that's okay.
So although the day 2 for AgriVision 2K22 has begun with some promising sessions, some promising talks, let's now officially begin the day with seeking the blessings of Lord above. To do the honors, I would request Shri D.P. Das, AGM NABARD, to please join me on the stage. Shri B.K. Sahu, Dean IAS SOA. Shri Jitendra Sundarai, Principal Scientist ICAR CIFA. Dr. Sangamitra Samantrai, Principal Scientist NRRI. Shri Soren Das, ISI Kolkata. Requesting Shri Divyansu Prasad Das, Program Director, AgriVision 2K22, to do the honors with all the dignitaries here, and let's lamp the light together. All of us, let's please stand up for a while. This is the official beginning. This is the official beginning of the day 2 of AgriVision 2K22. So much, everybody. So it is said a day started with seeking the blessings of the Lord above. Definitely is something special in itself. So let's start with the opening remarks now. I would request Shri Divyanshu Prasad Das, Program Director, Agri Vision 2K22, to join me on stage. Can we please have a wonderful round of applause for the gentleman? Thank you.
Namaskar, Jai Jagannath. I welcome all our esteemed guests, renowned scientists, research scholars, student delegates from different institutes of ICAR and state universities of Odisha. I welcome you all again to the second day of Agri Vision 2022. So today we'll be having some special sessions along with poster presentations and young investigator forum presentations. So I wish all the best to all the poster presenters and the young investigator presenters. So because we are having different categories of awards, so once the poster sessions get evaluated, so the best 5 in the poster and the best 5 in the young investigator award will be awarded tomorrow in the valedictory ceremony. So all the best to all the participants. It's an honor to have Mr. D.P. Das, who is the Assistant General Manager from NABARD, who is here because NABARD is the sponsor for this Agri Vision 2022. So we have him with us. So he'll be delivering a special, he'll be taking a special session. So before we start, we are really blessed and thankful to all our sponsors and supporters who have extended their kind support towards organizing this Agri Vision 2022 and making it a great success. So not in order, Larsen & Toubro, Central Bank of India, Odisha Tourism, Bank of India, APEDA, NABARD, Tea Board of India, National Medicinal Plant Board, Department of Biotechnology, NALCO, TriFed, CTCRI, Coir Board, Botanical Survey of India, SIVA, Central Institute of Women in Agriculture, Central Institute of Brackish Aquaculture, Central Institute of Fisheries, Freshwater Aquaculture, Central Horticulture Experiment Station, Mahanadi Coalfield Limited, Canada Bank, Coffee Board, ICARDA, CMFRI, Coconut Development Board, NRRI, Odisha State Warehousing Corporation, Bharat Petroleum, Odisha Agro Industry Corporation, NBPGR, National Seeds Corporation, CRIPCO, PCRA, Ruchi Food Line, Omfed, Paradip Phosphate Limited, Ormas, last but not the least, Department of Culture of Odia Language, Literature and Culture Department, Government of Odisha. So I believe you all will enjoy your day today. Thank you. Now I request, so before proceeding for the next session, which will be taken by Mr. D.P. Das, AGM NABARD. So I would like to give a brief introduction about him. So it's a great news that we have a Ravenshaw alumni with us. Sir, I would request you please come on the dais. Please give a big round of applause for DP Das, sir. He is a postgraduate in economics from Ravenshaw College, MPhil from JNU, 3 years research work as a PhD student in GNU with UGC Senior Research Fellowship. So he joined NABARD in 1996 as an economist and worked in UP, Jharkhand, Odisha and Andhra Pradesh regional offices of NABARD at various capacities. He worked as a nodal officer for NABARD consultancy services for 5 years in Odisha and Jharkhand office. Presently, he looks after the corporate planning department and the Department of Economic and Analytical Research of NABARD Odisha. He has wide experience in banking, credit planning, financing of infrastructure development projects, designing and evaluation of development projects and climate change adaptation projects. He has conducted a large number of studies and in-house research projects of NABARD. So today, I would request Mr. Das to present your topic. I welcome you, sir.
Thank you, Divanshuji. I don't require, but I require a small presentation in case it has already been provided. I have my laptop there, but I have already shared it to Divanshu sir. In case it can be just in case it is possible, that's quite nice. Sorry for a small disruption. Anyway, it is quite nostalgic that I am back to this respectable, this institution, respected institution across the world. I know Ravenshaw is known across the world as one of the premier college earlier, and now it is a premier university across the globe. So it is quite nostalgic. I am coming back to this center after 35 years. So in 97, I might have left this college those days after completing my post-graduation in economics. So anyway, I have nothing much to spend with, to share with you in context of research, because this is a special thing you are doing. It is a very specialized activity. Every single student, every single researcher must be doing a very in-depth research on her own, and it could be interdisciplinary, it could be very specific to your inquisitiveness. All what those days when we were studying, our professors were interested that be sure that you are adding to the pool of knowledge. Unless you add a single drop to the world of wisdom or the pool of wisdom, then a lot of efforts are wasted. So this is the thing. The thing is that from day one, we should be very convinced that our efforts, all these investments, and all our madness to complete and do something must add to the pool of knowledge, whatever small it could be, but it will be a supplement. Anyway, I'm here just to explain how a national level organization like NABARD has been doing. It is a brief 10-slide presentation that gives you a brief idea that what exactly NABARD does and how we are associated and what specifically we can contribute also in terms of research and development. Can we move ahead with this slide? So, you know, since it is a botany specific research,
and research-based conference, I specifically bring this to your knowledge that whatever we do as an NABARD or as a researcher or a scientist on all those things, we owe this, our existence to this small layer of topsoil, and how we are contributing, that is practically depends how we maintain and sustain these development efforts and keep our natural resources and endowment intact. So this is the basis on which even NABARD works. Next, this is the journey for NABARD, started in 82, then it has been almost 40 years complete. Within this 40 years is not a very big time for an organization like to be a national organization, but we have contributed substantially for different things. I just expect you to just go through this, and I am not elaborating on this. Basically, we have grown from 2000 crore in 92 to 6.5 lakh crore entity now. We have our presence in all the districts. Any district you tell, we have a senior officer who takes care of the development efforts. He is the person who converges the efforts, and he is associated with banks, development institutions, government, all angles, all verticals of the government, and pursue the development agenda of NABARD. Then again, through three big things I just explained, we have started SSG, Swayam Sahayata groups, long back, 92. And this has been the movement across the world, you can say, in even the African countries, all the underdeveloped countries have this concept in their mind, and they see it, the mass movement that carries the development into the grassroots level. So now the extent of this presence, you can feel the model created by NABARD is available worldwide, and more so in all states, and they are pursuing the development agenda. Next is our Rural Infrastructure Development Fund. Practically, with an honest beginning in 96, we started, and now all states, they are asking for NABARD fund for all the irrigation projects, roads, bridges, and everything. So all type of rural infrastructure is being supported. And next, okay. In context of this climate change also, NABARD has been the implementing agency, national level implementing agency, and we coordinate some climate adaptation programs, climate change programs for the nodal central government and state government departments. Then so many funds have been created in NABARD to monitor, mobilize the institutions who can support in different forms. This is all about food processing, irrigation. This is and a large number of government subsidies that is being monitored by NABARD. So these are the activities. I just give you some idea that how NABARD has been pursuing the agenda. And next, please. You may be knowing watershed projects. I think these are the fields where the researchers need to see how our efforts can be upscaled, because the farming practices in deprived area, low endowed area, low irrigated area are the areas where we require equitable development. So we struggle there with bigger challenges, how to keep development at par with the national level, international level. So bringing development into those deprived area is our struggle, and we see how these researches specific to technology innovations that can propel the development in those areas. So many climate proofing projects, so many spring side small developments in hilly areas, Anyway, these are the developmental initiatives. We create pilots that can be replicated by the government organizations, and it can be implemented in mass level to bring development. So even corporate social responsibility is coming forward to support NABARD in those things. Next. This is specifically, I keep interest in Odisha. How much we have done in specific areas. Every year we propel a big ground level credit for farm sector, allied sectors. We extend infrastructure development specifically in a bigger scale in Odisha because Odisha is low endowed in terms of infrastructure. Large number of watersheds projects are there. Large number of tribal development projects were there, where the tribal people are supported with the small orchards in their backyard. And when they produce in small quantity, in terms of large number of households, this gives a good pool of production. And now you see there has been good availability of horticulture products like mango, cashew in Odisha. This has practically created a lot of interest in them to expand these horticulture production in their areas. And we require a lot of technical support to upkeep their technical standards and capabilities. Next. And you know, farmer producers organizations are being supported because they form the implementing module for development. Unless they get together, the small farmers, small holdings will not give them the required strength to compete with the big markets, big capitalistic procedures. Here are the concentrated activity that we do for research. We have a department of economic analytical research. That is, when we created NABARD, this was there in the article that with the start of NABARD, we have this mandate that we should support the research and development that will propel the rural development and banking, specifically for rural clientele. So all types of action research, all types of technology transfer type of activity which are specifically linked to the rural development, we extend support in a big way. And this could be a collaborative research, it could be an independent research. We have a type of specialized services that what type of things and how we do this collaborative research. So practically it should be inclined to rural development and it should be inclined to banking. That must, the research should be helpful for the fraternity, banking fraternity and the rural development and the promotional activities. You know, we take students under our summer internship schemes. Every year we take some students with a good support system. Then we invite students from premier institutions. We are also in association with OUAT and some other big research institutions where PhDs are also supported. And you know, this is a one component, as Priyansh have already explained. We are here as a sponsor and we know all types of research, whatever small or big, that brings development and it has some impact, some repercussion, some benefit to the rural segment in long run. Next. So, in my own experience, I see we have grounded so many climate change projects, climate adaptation program, soil sustainability programs. Specifically, we have done wonders in many centers, but this requires replication and multiplication. So, I think this is the concept we are working in some segment. And this is the basic requirement. When I started with this slide, this was the biggest requirement, that how soil quality can be maintained and this can give us sustainable development to all the economic activities, soil-based activities. This is the challenge that always we struggle for collaboration And we expect that any institution that will be supporting us in this context will be extending our support. Next, these are some activities we have done across the country. Large number of watersheds we tried that the soil improvement can be taken care of. Next, these are the basic photos from the fields. I just bring this that solar, then percolation tanks, the soil improvement measures. These are we are doing in pilots to show that remarkable change can be possible by these small interventions. It is not very expensive, but anyway, sustained effort and concentrated plan of action should be there to propel this activity in long run, and this will give us sustainable development all the way. Next. Thank you. This is a small brief presentation I wanted to share with you. Thank you very much giving me this opportunity, and I am privileged that I am again speaking from a institution where I have studied. Thank you. Thank you.
Thank you so much, sir. That was nostalgic. And now I will request Dr. Shoren Das to join us on stage. He is the Associate Scientist, Indian Statistical Institute, Kolkata, and he'll be talking on the topic, the most vulnerable tropical forest and its sustainability.
Hello. Good morning, everyone. Actually, the post that previous session, we listened some hardcore biotechnology, how to handle the genetic material of a organism like DNA, RNA, etc., etc. If you think that all hardcore thing or modern technologies have some previous history. That means that is the hardcore genetic material have some phenotypic appearance that attracts several people, not more people. Because hardcore material, work with hardcore material, some handful people work with them, but we, the common people, always go for a broader way. Now, I'm talking about the vegetation pattern, which is mostly tropical vegetation. It is mangrove vegetation. It is obviously in some tropical country throughout the world, it suffers very much as extending the civilization or modern technologies, industrialization, etc., etc. We common people encroaching the nature area, and unfortunately, unknowingly, we are putting our hand in nature, which may destroy ourselves. Pardon, how much time I have allowed? 30, 15 minutes. Okay. Now this is the estimation of mangrove forest, mangrove vegetation. Mangrove are not a single plant. It is a combined of community of several, almost 30 to 32 plants that have some several families, species, but have some common criteria to withstand the salinity. Not salinity as much as sea, but it has some estuarine. It has to withstand estuarine salinity. 120 through 23 countries we have the mangrove vegetation across the world and having almost 150,000 kilometers square, and the largest mangrove in the world covered in Indonesia. It is about 22%. And why the mangrove is so important? Mangrove provides several ecosystem service such as coastal protection, one of them. It has some protective wall or frontline barrier of several natural sea surges like that, storms, some heavy devastating tsunami like these. All these things that mangroves have to cross the mangrove barrier and then devastation caused to mankind. The distribution of different drivers of change in mangroves temporally extend across the tropic. If the first one, it is advance and regrowth of mangrove extent through 1996 to 2010, sorry. The red mark is the advancement mangrove losses. You can easily see the loss of mangrove is happening across the world during this time. And most of the loss has become from the South Asian countries, I think. And degradation and from anthropogenic drivers change, including evidence of prior disturbances. These pictures, satellite pictures showing the temporal conversion of mangrove vegetation to aquaculture. It is the most profitable business to nowadays to encroach the mangrove area, as because mangrove area is some estuarine area and aquaculture is mostly preferably this estuarine situation. That's why the mangrove area encroaching by the people who are using the aquaculture. And this is the picture. 1996 red mark is denoted the vegetation in false color composite, the satellite images from the satellite images, how it becomes red to dull and green. These all Indonesian mangrove picture during the 1996 to 2020. From this picture, you can easily understand the how can we destroy the mangrove vegetation time to time. This is about the 40% of mangrove covers found in, this is an Indian scenario totally. In Indian scenario, West Bengal, we are proud of that we have the largest mangrove forest in a single block, that is in our Sundarban. But there is a big question. Do you know why that mangrove forest is named as Sundarban? And there are dual answer. One is Sundar, beautiful land. That is Sundarban. And another is, that is our concern, a plant called Sundari. It is vernacular name. Scientific name is Heritiera fomis. Once upon a time, at least about 50 years, Sundarban was densely have some Sundari trees, that Heritiera fomis, that Garth, at least I cannot encroach this one like this. But nowadays, it is hard truth. Nowadays, if you go to Sundarban and search for Sundari plant, hardly you can find a Sundari plant which is lean and thin like this. In biodiversity, we always talk about the loss of one ant is a serious issue. But nowadays, Sundari plant is now estimated as a, this is the cause of loss of total Sundarban. I am coming to the pinpoint, the plant Sundarban. This is the satellite images of different delta. The temporal changes over the year. The red portion is having the greenery. How they abolish the red portion in the area, Krishna River Delta, Vitarkonika. I personally visited several times in Vitarkonika Delta. That in 1985 it was such red, dark red, and several times, and 2004 it is somehow abolished. That means this forest area, this tropical, economically rich and protective, basically protective forest, have been are abolishing time to time because of us, mankind, not other. There are two reasons. One is man-made reasons, and another is environmental factor. Environmental factor is there, but not so much prominent. Environmental factor not even what sudden within a day. It not create much factor, much devastating factor within a week, within a month like this. Environmental factor works day long, and it take much time to abolish the total forest area. But the man, we can finish. You know, the USA and Ukraine, how they fight and finish the environment. That is a separate issue. This is just numerical data about the mangrove coverage. Now, that means sustainability, if we talk about the sustainability of the mangrove plant, any plant, have some depend on some factors. I just managed to gather three things. One is anthropogenic threat, environmental threat, and efficient forest management policies. This is on us, on the human being, and environmental threat we cannot avoid. And we only manage this thing and this thing, anthropogenic threat. Now come to the study area in Sundarban. Based on the salinity level, Sundarban forest divided into three parts. Less than 5 ppt of salinity, less than 10 ppt of salinity, less than 15 ppt of salinity. Unfortunately, we, the Indian Sundarban, have fallen into this portion. That means vulnerable area we have, and we have to protect them. Why? Why Indian Sundarban is getting the salinity level rise? Salinity level always managed by the influx of the nearby sweet water to the sea. Mangrove plants cannot grow across the sea level, never. It always prefer where the intermix of seawater and sweet water. If the influx of sweet water is less, then salinity automatically will be rise and the plant suffers much there. But if we see the Ganga river, it is coming from the Gangotri and etc. Crossing Uttar Pradesh, Uttarakhand, Bihar, Jharkhand and West Bengal. It enters West Bengal. Before it comes to the sea, it diverges by the name of Padda in Bangladesh area. And the main stream of the Ganges is running through the Bangladesh area. And we made a mistake. I'm sorry to take it mistake. In the lower Gangetic plain, the river flow always slow, as it is in normal situation. In Ganga, in West Bengal, it is lower Gangetic plain, always water flow, the current flow, current of water flow is always slow. And we put a barrier there where it is Farakka. Then Farakka barrage was constructed during 70s like this. Dr. Triguna Sen is a famous scientist, and the Triguna Sen committee recommended at least 13 dams should be there. 13. This dam, the encroachment of this dam should be there, and the dam reserves the water during the rainy season, and during the lean season, the water should go through this channel to wash out the silt of the entire area. Unfortunately, how much dam is prepared in Farakka? Do you know? 5. Out of 13, it creates 5. I think that is a blunder. We just, Indian government don't have so much of money. I think that's why it comes only 5. But in long run, totally the silt formation of the Gangetic bed cannot be wash out through the sweet water. That's why in Indian part of the Sundarban, sweet water influx is restricted somehow. But the main Gangetic flow runs through the Bangladesh and join Brahmaputra from Assam by name Meghna. That's why the salinity level of the Bangladesh part is 5 ppt to 10 ppt only, whereas it is in 15 ppt. But nowadays it is old data. Nowadays my present experience is 35 ppt. How the plant survive there? That means the plant who are surviving there are always fighting with the natural threat itself. It is not a matter of joke. And the plant itself coming from the several families, several species. That's why the adaptability of the individual plant are not same. But we, our forest department, we always are going for the plantation effort throughout the banks of the Sundarban region, everywhere, without knowing which plant have the ability to which extent, without knowing. We don't have the data. Now, time is now that we have to judge each and every species, the adaptability level and the salinity withstand level have to judge. I took some example. One is Sundari, that is Heritiera fomis, which is in red listed plant by ICNU. And there are some other plant which is Bruguera, Cereops, Abyssinia, like this. This is Abyssinia fruit. Yeah. These are the root system of the Sundarban plants. One thing, leaf thickness must be a good criteria to withstand the salinity level. The leaf thickness means have some water storage tissue within the leaf. They can balance the osmotic factor of the seawater and the cell itself within the plant. That means leaf succulence is a criteria. See, the leaf succulence in Sundari Heritiera fomis is much less than the other three. That means morphologically or anatomically it is weak. The seedling germination pattern like this, it is advanced in some mangroves, but in Heritiera it is a normal seed germination pattern. That means it is also weak. Now, some micromorphological adaptability. See, there are some more water storage tissue in the leaf, but the Heritiera don't have water storage tissue like this. That means anatomically, micromorphologically, Heritiera is also some lagging behind. This is also wood anatomical feature. In wood anatomical feature, the tracheary elements feature in Heritiera, it is somehow long and narrower. Long and narrower is somehow primitive feature, we know. This is osmotic potential. That means I want to say that all physiological, anatomical, not genetical, I don't say genetical, not now. This is not the time to cover this genetic. And biochemical estimation, it reveals that Heritiera fomis do not withstand the salinity level nowadays. But this plant is flourished much more in Bangladesh region. That means their salinity level is much lesser than this side. There are some ROS factor, reactive oxygen factor. There are some enzyme factor. In all the cases, these abiotic stresses cannot withstand for the Heritiera fomis nowadays. That's why it should be placed in such a area where the salinity level is somehow less. That means the inundation, water flow cannot reach up to this ridge. That's why Heritiera should be put in a ridge area. More than just trees, the mangrove forest ecosystem, we should protect them. That is why. Thanks for your concern, and anything to ask, I'm happy to answer. If there is no question.
have a special question.
Oh, okay, thank you.
Thank you so much, sir, for this wonderful talk. Well, we have this very special person here with us today, Shri D.P. Das ji, who could take his time out and be here to express his feelings, gratitude, and talk to the wonderful gathering over here. So we have this small token of gratitude, but definitely not small by the size. Only it's very special from everybody's side. So let's welcome Shri Divyansu Prasad Das, Program Director, Agri Vision 2K22, on the stage and present this token of gratitude to Shri D.P. Das ji, Assistant General Manager, NABARD. Sir, can we please have you on stage? Please accept this token of gratitude from all of our side on behalf of the entire family of Agri Vision 2K22. This is for you. And I'll request everybody to please have a wonderful round of applause for the gentleman. Thank you so much, sir, for taking us down the memory lane and also briefing us about NABARD.
I feel I am always a student because researchers are always a student. There is go on learning. It is a privilege that you are pursuing research, and the students also I request that you get inclined to get an additional bit compared to the others in context of a specialization. So specialization is the thing you prosper with, and general
Hello.
is I'm somewhere not so happy with. So be general for your study, but be a special researcher in your special area and your specific competence in those specific areas. Thank you. Thank you.
Thank you so much. Thank you so much, sir. Please. So this one is from the entire family of Agri Vision 2022, which is presented by Shri Divyanshu Prasad Das, Program Director of the same. Thank you so much, sir. Thank you so much, gentlemen, for joining us on stage. Now we have Professor Pradipta Kumar Mahapatra, HOD, Department of Botany, Ravenshaw University, on this podium to talk about the topic photosynthetic pigment fluorescence under pesticide stress.
A good day to all of you. Initially, I have been allotted 20 minutes to have this keynote address. And in between, because of the shortage of time, this has been rescheduled to 15 minutes. But I will try my best to complete in 15 minutes, or if it is more than 15 minutes, then I beg the pardon from the organizers. I will be speaking on chlorophyll fluorescence, primarily photosynthetic pigment fluorescence under pesticide stress. Before I go to the topic, I just want to clarify why fluorescence and in what way fluorescence is a tool to be used in determining the health of the plant. As you know, when you go to a doctor, and say to the doctor of some of your ailments, what the immediately doctor does? He immediately measures your blood pressure, your pulse, maybe some other morphological diagnosis. Fluorescence is like that: quick, non-invasive, and rapid diagnosis tool to determine the health of the plant. And fluorescence can predict whether the plant is healthy or not healthy, whether the plant is under stress or not under stress, and whether the plant is receiving any type of environmental biotic, abiotic, insecticide, pesticide, whatever, drought, stress, flood, all type of stress. If I go to fluorescence, as you know, because in case of plants, whenever there is any stress, the immediate response is change in photosynthesis. And this change in photosynthesis can range in different scales. I will go to that later. And if you go to the photosynthesis, you can basically divide this into two parts, the light part and the dark part. And these two parts operate in 4 different stages, like say light absorption. This light absorption is a very fast process and is a physical process. It doesn't matter whether the plant is alive or dead. It doesn't matter the chlorophyll is present in the membrane or outside the membrane. It also doesn't matter whether the thylakoid is intact or not intact. Whenever it receives light, if the chlorophylls are intact, they will absorb. It is a general properties of the pigments. But after they absorb light, if the entire system is integrated and is working, then only that light energy will be converted to chemical energy. Otherwise, that will be released in different forms. So this first part, absorption of light, which is very fast and is completed within femtoseconds, 10 to the minus 15 seconds, is the very beginning process, starting process in photosynthesis. And once the light is absorbed, then that light energy will be converted to exciton, which moves from one pigment to the other to reach at the reaction center. And in the reaction center, the second event will be charge separation. It will be excited, it will release one electron, and that electron will go to a higher energy state, and then there will be electron flow, so electron transport. So if you start from the absorption which occurs in femtoseconds and then charge separation in picoseconds, and electron transport occurs between nanoseconds to microseconds or even milliseconds, but not more than 500 milliseconds. And thereafter, that process drives an electron transport, and this electron transport is converted in converts the NADP and ADP to ATP and NADPH, and that is used in the carbon fixation, which ranges between minutes to hours. So you can summarize this in a way that the first one is the photon, which is absorbed and is converted to exciton. Then the exciton is converted to electron, and electron passes through the electron transport chain, and in the process it reaches finally the terminal electron acceptor, that is NADP plus, to produce NADPH, and in the process it builds a chemical gradient which is called delta pH, and that chemical gradient is responsive for driving ATP synthesis. And pigments are arranged in such a way that the absorption of light and transfer of absorbed light to the reaction center is maximized. Therefore, they are present in the form of a hexagon, and in the hexagon, those pigments who absorb lower wavelengths, they are present in the periphery, and those who are present very close to the center or in the reaction center, they are absorbing the higher wavelengths. So there is gradual an increase in the absorption maxima of pigments as you move from the periphery to the core, and you can see these absorption peaks also, they differ with the pigments. So as I told you, they are arranged in the form of a hexagon, and these are the pigments absorbing lower wavelengths, and those which are absorbing higher wavelengths are in the center. And in the process of photosynthesis, when all the pigments are in functional and plant is not under stress, plant will try to maximize the conversion of absorbed energy into exciton and exciton to electron. And under such conditions, all photosystems are functional. But in the process also, some part of absorbed energy is released in the form of fluorescence. That fluorescence which is released when all photosystems are in active state is called the fluorescence maximum, or F0. But when plant receives stress and theoretically say all photosystems are closed, or if you provide a stress and close all the photosystems, whatever light is absorbed, that will never go to the reaction center. And instead, that will be released in the form of fluorescence, and that is called fluorescence maximum. So you can say that had there been no stress, the plant would have converted all the absorbed energy into chemical energy. So the difference between the maximum fluorescence and minimum fluorescence is the capacity of the plant to convert the absorbed energy into chemical energy. And from this dynamic of changes of this F0 and Fm, one can diagnose whether the plant is healthy or not, and whether the plant is functional or not. Fluorescence is very easy to see. Sir David Brewster is the first person who saw the fluorescence, and he saw it in a very simple way. He extracted pigments and put the pigments in a extracted pigment in a flask and just exposed it to a strong beam of light, and he could see the fluorescence. And you can see fluorescence in your laboratory. Just take either chlorophyll extracted solution or a green algal culture in a cuvette and give a strong beam of light from any projector. You will see it fluoresces red. It fluoresces red because chlorophyll fluoresces red. And here you can see, irrespective of what is the pigment and irrespective of what is the light that plant is getting, whether it's blue light, red light, or actinic light, fluorescence always comes in the red end of the spectrum. You can see the absorption spectrum. You can see the absorption spectrum, light being absorbed at different peaks by different pigments, but fluorescence at room temperature primarily come from photosystem 2 at an wavelength of around 685 to 690 nanometer. So 95% of the fluorescence that we are measuring by using equipment today at room temperature are primarily from photosystem 2 and a very small contribution from photosystem 1. But if you expose put the cells in low temperature in liquid nitrogen, say for example, at minus 196 degrees centigrade, you will find contribution of photosystem 2 will be very high, photosystem 1 will be very high. So this is a common electron transport chain, electron moving from chlorophyll to NADP plus, and in this chain you will see there are so many stations. Electron starts its journey from water and finally reaches at ND plus, NADP plus, and there are so many stations. And for each station, there are different time kinetics. It takes different times. Therefore, because in some cases the times are very short, it is very difficult to visualize the change of fluorescence. Nevertheless, there are some stations like say this one, about 800 microseconds, about 2 milliseconds, about 1 to 30 milliseconds, about 200 to 500 milliseconds. These 4 places, it is easy to see a fluorescence peak. And on the basis of this idea, the equipment plant efficiency analyzer was prepared, manufactured, which gives a polyphasic fluorescence rise. And from this polyphasic fluorescence rise, we actually take the signals, and those signals are just converted into the informations. So fluorescence is a small fraction of the light absorbed by a plant. This is what Reto Strasser, the person who initially designed this equipment, has told. It is like looking at the Earth from the Saturn. If you look from the Saturn, if you look at the Earth, you will see Earth will look like this much of size, nothing. But if you do this from Moon, you will see Earth is so big, having so much of colors, having forests, having water. If you see it from the satellite, fluorescence is like this. Fluorescence, when all the photosystems are closed, you get only 3% of the absorbed energy released as fluorescence. And when all the photosystems are open, you get 0.6% of energy absorbed from the fluorescence. The difference is only 2.4%. But that 2.4% says a lot of untold stories. And the entire production, photosynthetic production, can be measured in the form of light absorption to the form of yield. Say when we are measuring rice performances, we are measuring in different forms. We are measuring root length, shoot length, dry mass, wet mass, number of tillers, length of tillers, etc., till the grain size, grain weight, 1000 grain weight. All are the parameters for measuring the plant performance. So that can also be done very rapidly within seconds by using fluorescence. And for measuring fluorescence, the time interval is between microseconds to maximum a minute. Most of the work of fluorescence measurement is completed in seconds, microseconds to milliseconds, maximum up to 10 seconds, 20 seconds. If somebody is interested in delayed fluorescence, he can measure up to few minutes, 2 minutes, for example. Here there is a comparison. The liquid culture, when there is no stress, light is absorbed, you find efficient photosynthesis, very little amount of energy is released. But when the plant is exposed to stress, you will find the amount of energy that is lost will be measured in the form of heat or in the form of fluorescence. And this fluorescence gives you a polyphasic rise, which we call 0 to 2 milliseconds. This is called the OJ rise. It is a reflection of photosystem 2 function from photosystem 2 quinone A movement or electron movement. And the next phase is from electron quinone A to photosystem 1. And then the third phase, IP phase, is from photosystem 1 to NADP plus. These 3 phases, these 3 peaks are visible. If you take the equipment and measure the leaf fluorescence for a second, the entire measurement has been made for 1 second. If you do it, you will get only 2 and 3 peaks. But if you mathematically derive from this native spectrum, you will get many different peaks. And these peaks says about the electron movement from one component to the other in the electron transport chain. Like 20 microseconds to 150 seconds in the light harvesting complex, like water splitting complex. From 150 to 3 milliseconds is up to tyrosine. Then in the J up to plastoquinone, from J to I is up to cytochrome B6F complex, from I to H is to photosystem 1, and from H to G is pyridoxine reducing system, and G to FM by FT or P is to NADP plus. And these are mathematically derived by this formula. There is no time, I cannot tell you anything about this. So, under pesticide stress, what are the responses of plants with regard to fluorescence? There may be reduction in fluorescence. And if there is reduction in fluorescence, your conclusion is dephosphorylation. So, photosystem 2 antenna is dephosphorylated and is separated from the reaction center, or quenching. Fluorescence inductions, then you will have a polyphagic inductions, and these polyphagic induction will indicate blocks. And from the peaks, you can say where actually the electron is blocked. And for shifting of peak, you have delinking and dissociation. If the peak is shifted from, say, not in 685, rather than the lower wavelengths or higher wavelengths, then you can say which part of the antenna is separated. This is a presentation. I will take 20 minutes. I will take 5 more minutes. This is a presentation where multiple flashes have been given. Plant is under normal condition, no stress. And in each flash, you can see the zero fluorescence and the maximum fluorescence are constant. That means within 2 flashes, the plant is relaxed, again returning back to the normal metabolic state. But if you see the next one, you can see here, with each flash under stress, at an insecticide stress of 50 micromole, you can see that with flash, gradually there is reduction in the maximum fluorescence and you get a strong increase in the ground level fluorescence. So F0 goes up and FM comes down. F0 goes up indicates that when you are applying the pesticide, the plant photosystems are closed. They do not respond to light. It doesn't matter whether the plant is receiving light or doesn't receive light, as if the plant has slept. And therefore, you get a very high level of F0. And because many photosystems are not functioning, automatically you can easily say that these photosystems are not participating in the photosynthesis. Automatically it is reflected in terms of plant growth or in terms of the yield or plant performance. Here you can see how rapid this process occurs. Pesticides was given at a concentration of around, you can see here, these are different concentrations, highest concentration, and this is the control. Reverse is for the FM, and this is for non-photosynthetic quenching. Non-photosynthetic quenching and photosynthetic quenching are 2 opposite processes. When plant is not able to convert the absorbed light into photochemical energy, which is called photosynthetic quenching, it will try to protect itself. And in the process, it will release that energy in the form of heat, so that the photosystems are not damaged, pigments are not damaged. And if the plant fails to do so, say for example, concentration is very high, plant is not able to do so, then the plant will bleach. It will degrade its pigments as a protective mechanism. It's called down regulation of the photosynthetic machinery. It will shrink its light absorption capacity. So you can see how fast this process occurs. Within 20 minutes, almost all absorbed energy are converted to non-photosynthetic quenching. Within 20 minutes of application, all absorbed energy are, one is the 100%. So all absorbed energy are converted into heat. Plant is not utilizing any of the absorbed energy for photosynthesis. So a photosynthetic process is completely stopped. The same reflection of this, you can see a comparison here at different concentrations, and you can see opposite processes. Non-photosynthetic quenching goes up, and consequently also you will get photosynthetic quenching coming down. And different pesticides have different impact on fluorescence. Here there are 4 pesticides. 2 are phosphorothiols and 2 are aromatic pesticides. You can see quinalphos and chlorphenifos. In these 2 cases, there is a decrease in fluorescence. Whereas dimethoate and phorate, in these 2 cases, there is an increase in peak. This is control, blue one is control, and this one is the highest concentration. Everywhere you can see these lower 2, they are causing increase in fluorescence. When you get increase in fluorescence, you can easily say the block is between photosystem 2 and photosystem 1. When you get decrease in fluorescence, you can easily say the block is before photosystem 2. That means between water and photosystem 2. In between this, it may be the proteins of photosystem 2 or the oxygen evolving complex of photosystem 2 or even the extrinsic polypeptides which are present in the luminar side of the thylakoid membrane. Here you see the polyphagic fluorescence, the highest concentration and lowest concentration. The same way in quinalphos and chlorpheniphos, you get a decrease in the polyphagic fluorescence, whereas in case of dimethod and borate, you get an increase here at 2 milliseconds. There is a block in the plastoquinone. Plastoquinone cycle is blocked. Therefore, the electron cannot move beyond plastoquinone. So it doesn't reach the photosystem 1. There are a number of derived parameters that are derived from the polyphagic fluorescence, and these parameters are basically divided into two types: the efficiency indicating parameters and stress indicating parameters. So if there is an increase in a set of parameters which are called efficiency indicating parameters, it indicates the plant is efficient, and the stress indicating parameters indicate the plant is under stress. For example, M0 is the proportion of reaction center entering into the closed state. This is a stress indicating parameters. But the primary photochemistry, exciton movement and electron movement, these three are efficiency indicating parameters. Dissipation is a stress indicating parameters. That means if the plant is under stress and is not in a position to convert the absorbed energy into photochemical energy, it will try to dissipate. So more dissipation means plant is less photosynthetically active. So dissipation is a stress indicating parameters, electron transport, exciton transport, these are two performance indicating parameters, and these are three performance indices. The performance indices of absorption, performance indices of electron movement, and performance moment of absorption, electron movement, and primary photochemistry. These three are performance indicating parameters. So here there is a comparison up to 2 milliseconds. 2 milliseconds means fluorescence has been measured up to plastoquinone, not beyond this. And 2 millisecond fluorescence is a very, very important point, measurement of 2 millisecond fluorescence, because at 2 milliseconds, the electron transport rate is very slow as compared to the electron transport that occurs from water to quinone A and quinone A to plastoquinone or beyond plastoquinone. So any peak at 2 millisecond fluorescence is an indicator of the trafficking of protons across the thylakoid lumen, because the plastoquinone cycle is the cycle which in one way gives electron to cytochrome B6F and in the other way pumps proton into the thylakoid lumen. So if this cycle is blocked, you will get a fluorescence at 2 milliseconds. The plastoquinone movement across the membrane, plastoquinone moves in the membrane from outside to inside and vice versa. So if this movement is stopped, then you do not get the movement of electron beyond plastoquinone. And here you will get a rise in the fluorescence. So you can see here with increase in the concentration, there is a rise in the fluorescence. This is by with dimethod treatment, I think. Yeah, it is by treatment of dimethod, cyanocystis with treatment of dimethoid at different concentrations. I will be skipping. We have used fluorescence for measuring the tolerance development by enrichment and by repeated exposure of the plant to a concentration of the insecticide. We made the plant tolerate the insecticide concentration, and then we compared this tolerance with the normal normal strain in case of algae. But this is in brinjal, in which we measure application of insecticide in the first time, second time, third time at 45 days intervals, and then in each time after application we measure photosynthesis continuously up to 96 hours to see if the insecticide is applied at the field dose, whether it causes any effect on the plant. So we applied the insecticide in the field dose, and for first 6 hours there is a reduction in photosynthesis. Thereafter, however, the plant recovered and again entered into the normal photosynthesis 96 hours, okay, 50 hours, okay. And it occurred in the first treatment, in the second treatment, in the third treatment also. But in the process what happened, if you compare the metabolism of insecticide degradation, if you see the metabolism of insecticide degradation, the plant gained ability to degrade the insecticide. So in the first application, the degradation was slow, but in the subsequent applications, gradually the plant gained capacity to degrade. This is not a good signal. Because if the plant becomes very efficient in degrading the insecticide, then the insecticide concentration in the plant will go down and the insect will be resistant. And therefore, repeated use of the same insecticide in the same crop is not beneficial. That is what was our conclusion. And we also could able to publish this. And here we just compared, this is a tolerant strain developed with enrichment culture, and this is a normal strain developed with the enrichment culture, and this is a tolerant strain. And you can see at different treatment concentrations, the tolerant strain could able to tolerate and maintain the normal photosynthetic activity. Of course, there is slight increase in the J fluorescence here, but this is not significant. But you can see here the normal strain or the wild strain has a strong increase in the fluorescence. So this one is affected, this remains protected because of the tolerance. And this is the separation spectra. I am not going into the details how the spectra is separated, but I can only tell you this is 2 millisecond fluorescence, and this side is between water and plastoquinone, and this side beyond plastoquinone. And you can see in case of a normal strain, about 30% rise in the J fluorescence, but here in the tolerant strain, this rise is only 6%. So it indicates that the strain has tolerated the insecticide. This is in case of application of the agent to develop tolerance against biotic stress, and in the biotic stress, the tolerance development has been studied by measuring the difference in the fluorescence between those who have been bioprimed and those who have not been bioprimed. You can see here chemical use has been used, salicylic acid has been used as a chemical to develop the tolerance against the pest. Salicylic acid. What else? You can see here salicylic acid in different concentrations, and disease plants, control plants, and those disease plants having salicylic acid at different concentrations, 0.5, 1 and 2 millimole concentrations. And salicylic acid could able to protect, and accordingly we could able to record this in the form of change in the J fluorescence. So and also to predict the insecticide toxicity, you can use fluorescence by applying normal equation of additivity. We took two insecticides, one insecticide, one herbicide, dimethoid and atrazine. In case of in field, these two are combinedly applied, because atrazine application with dimethoid increases the susceptibility of the insect to the insecticide. But we wanted to know whether the same happens to the soil microorganisms like algae. So we just, because this one is a photosystem 2 block, this is a photosystem 2 inhibitor, this is also has been found to be a photosystem 2 inhibitor. So we applied the theory of additivity, then we measured the fluorescence, and equation from equation we calculated the fluorescence, observed predicted toxicity and calculated toxicity, and we found that this is not as in case of insects. In case of insects, it is synergistic, but in case of the soil cyanobacterium, it is just additive. It is not synergistic. So thank you very much. I had to rush, and all these works I have presented here is not of mine. I have not done any of these works. My students have done. So I acknowledge the contribution of my students Prashant, Mudhasini, Rinakshi, Sidevi, Aryawala, Chandrakala, Varsha, Sasmita, and Devasis. Thank you very much. And if you have any questions, I will be happy to answer. Thank you.
Hello. Hello.
So anyways, we have a 10 minutes tea break. Parallelly, we have poster session. You can enjoy that in the coffee. And after 10 minutes, we'll start the young investigator forum in the second hall, that is in Annex 2. And come back here in 10 minutes because we'll start the session very soon. Thank you. Sure.
Come home to nature. Come home to nature. Come home to divinity. Come home to calm. Come home to the world of art and craft. Come home to beauty. Come home to nature's nest. Come home to adventure. Come home to self-discovery. Come home to serenity. To history. Come home to eco in Orissa. Orissa, India's best kept secret. Come home to nature. Come home to divinity. Come home to calm. Come home to nature's nest. Come home to adventure. Come home to self-discovery. Come home to serenity. To history. Come home to eco in Orissa. Orissa, India's best kept secret. Come home to nature. Come home to divinity. Come home to calm. Come home to the world of art and craft. Come home to beauty. Come home to nature's nest. Come home to adventure. Come home to self-discovery. Come home to serenity. To history. Come home to eco in Orissa. Orissa, India's best kept secret. home to nature. Come home to divinity. Come home to calm. Come home to the world of art and craft. Come home to beauty. Come home to nature's nest. Come home to adventure. Come home to self-discovery. Come home to serenity. To history. Come home to eco in Orissa. Orissa, India's best kept secret. Come home to nature. Come home to divinity. Come home to calm. Come home to the world of art and craft. Come home to beauty. Come home to nature's nest. Come home to adventure. Come home to self-discovery. Come home to serenity. Come home to history. to eco in Orissa. Orissa, India's best kept secret. Come home to nature. Come home to divinity. Come home to calm. Come home to the world of art and craft. Come home to beauty. Come home to nature's nest. Come home to adventure. Come home to self-discovery. Come home.
So thank you for coming in early. So we have this next session by Dr. Luna Samanta. She will be talking, she is a professor, Department of Geology, Ravenshaw University. She will be talking on the topic comparative proteomics reveals augmentation in allergens as a function of waterborne toxic metals in the notopetrus from Mahanadi River. So let's hear her. Can we have a wonderful round of applause for the lady first? Thank you.
Thank you very much. Yeah. Very good morning, everyone. Thank you very much for giving me this platform to present our findings. And it is always a privilege at the same time, anxious one to present before our former Vice Chancellor, Professor B.C. Tripathi, who happens to be my direct teacher as well. And after listening to the dignitaries like him, and when I was invited to present a talk on agri-vision, where I'm a basic clinical scientist. So I was just wondering what to do. So one of my students was working on Notopterus notopterus, that's a fish, your knife fish, which is known as chittal or folly macha. So this one we started when we didn't have a good lab. So whatever could we started with that preliminary work, and surprisingly, it gave us a pretty good result. So I will be looking for this. Why I chose this one? Because the riverine fish catch contributes to production of around 1 lakh ton of fish in India, while Odisha contributes somewhere 20,000 tons. And the fish is known as a superfood, particularly for the coastal India, where rice and fish is the staple food. And besides, it has the white meat, which supplies the required amount of animal protein and as well as omega-3 fatty acids. So people are more concerned about propagating fish as a superfood for human consumption. And what could be better than the Cuttack people to have the geographies? Because this is a delicacy for, or it's a requirement or mandatory preparation for all the Cuttack people. So, however, with rapid urbanization and industrialization, there are a lot of pollutants entering into different river systems. And out of this, these pollutants tend to accumulate both in the sediment as well as water. And although this rivers is a floating system and it may pass on, but the organisms inhabiting there accumulate a lot of diverse type of compounds in their tissue, which go on biomagnification and pose a threat to not only to these organisms, but is transmitted through food chain and even reach the human systems. So metals being non-biodegradable and persistent make the most dangerous pollutant worldwide. And why we chose metals is that if you look into the Mahanadi river system, from Hirakud till Cuttack, mostly it is the agricultural runoff or the normal anthropogenic activities where there is no more industries in between. The most industries are in this Sambalpur on the Hirakud, upstream of the Hirakud river system or downstream of the Cuttack. So we chose here one of the site which is relatively free from industrial effluent disposal, and that is your Mundali upstream of Mundali, and then your Chaudhwa Jagatpur industrial area, 2 sites were chosen. And what we found is that in our preliminary experimentation is that the metal ion concentration in all these 3 sites are according to WHO, ICMR as well as BSI criteria. It is much lower. So all our industries are actually following the guidelines proposed by World Health Organization or Indian Council of Medical Research or the Bureau of Standards of India. However, what happens there, because of the biomagnification or the residual toxicity, all the fish tend to accumulate. And surprisingly, we find that in all the fish tissues, muscle, which is the consumable part, liver, the potential metabolic organ, as well as the gill, which is the site of water exchange, all of them contain higher amount of metals than actually advocated by ICMR, WHO or BSI. So what does it say? It says that these transition metals and the toxic heavy metals do accumulate in the fish. So what happens there? These fish, because they are the transition metal, they have a tendency to take part in redox reactions where they generate free radicals through pentane types of reaction, or they may be sequestered into the metalloenzymes by replacing the actual metals there, and thereby cause a serious threat to the fish health. So what we found from a discriminant function analysis, which is a multivariate statistical analysis, that the peroxidation of membrane lipids in the fish tissues, carbonylation of the proteins, as well as downregulation of superoxide dismutase and glutathione transferase are the major markers of metal-induced toxicity, and which came off in chemosphere. So with this background, okay, nowadays, whatever you study, there will be generation of free radical, there will be antioxidant depletion, and it's there in every terms, like every disease or every systematic disorders. But what is the cause? It's a consequence of the cause. That's for that we wanted to devise another experiment where we wanted to go for the shotgun proteomic study, because genomics is there, because free radicals, these heavy metals are known to be genotoxic, and they usually do DNA transition, mutation, and so on and so forth. And of course, when there is a genomic mutation, there will be transcriptional changes, but the actual functional part is your proteins, which form the structural functional basis of the organism. So we wanted to carry on proteomic analysis to find out which are the pathways they are particularly affected by this, and whether there is substantial change in the fish physiology, or thereby making it actually palatable or not. For that, we took from the same site, the same fish, which one was already published in Chemosphere, and we did liquid chromatography tandem mass spectroscopy approach to find out the differentially expressed proteins in these fish sites. And what we found that there are somewhere like 348 different proteins identified in this fish species from the 3 different sites, where 81 species of proteins were identical, means like commonly found in all. This is the global proteomic analysis. When we look for the differentially expressed protein, that is log 2 fold change, that is at least 2-fold change in their expression profile, which is statistically significant at least probability of 5%. Then we took that out of that, so 31 proteins show absolutely differential expression, where a single protein is very specific to site 2, whereas 10 proteins are very specific to site 3 in comparison to site 1. 21 were upregulated, 4 were downregulated. But what are these? Where are the then we did a hierarchical cluster analysis and a principal component analysis, which shows that the experimental sites, the proteins expressed in experimental sites are completely statistically significant from that of the control site. This green one are the control ones, red and blue are the experimental proteins. You can see here the 3D principal component analysis, that how significantly they are segregated from each other. And when you do the heat map of the hierarchical cluster, you can see that these are completely segregated, both in terms of their fold change as well as their distribution. So then we did the functional annotation analysis and enrichment for this one. So what we found that most of the proteins that is differentially expressed are cytosolic in nature. And when you look for the biological processes, it is the metabolic process which is really affected, and the oxidation reduction process and carbohydrate metabolism in particular. And then when we did the molecular function analysis, it is again hydrolase activity, oxidoreductase activity, paradoxical phosphate binding, and again oxidoreductase active aldehyde metabolism, and most importantly the redox balance, NAD, NADP mediated reactions. And from this, what we found, we again did a pathway analysis using the Cytoscape Plugo and Clopedia plugin. And here what we found that most of the proteins, those who are which are like green nodes are the upregulated proteins, red are the downregulated protein, and the red and pink is actually the different gradation depending upon the fold chain that has occurred. So here what we found, most often the oxidoreductase activity are the most affected one, including the mitochondrial one. This is what we have found. So the mitochondrial proteins are including their electron transport chain proteins, as well as all the metabolic proteins, carbohydrate, lipid, as well as protein metabolism proteins were drastically affected, as revealed by Cakes and reactome pathways, where we have a false discovery rate of pretty low, 0.05. And what we found here, you can see that the impaired metabolic pathway based on the David software, you find the lipid metabolism is affected, where you can see these red ones are highly significantly altered. And the glycan biosynthesis metabolism pathway, when you see here, what we found, enolase and pravalbumin alpha and pravalbumin beta, both of them are differentially expressed and very highly expressed. The fold change is more than 11-fold. And along with that, you can find here that the FOXO signaling pathway, superoxide dismutase and glutathione S transferase were also drastically reduced as observed in our biochemical assays. So what we found from this one is that, so when these metal pollutants enter this one, why I am interested particularly in beta enolase and pravalbumin, both pravalbumin and beta enolase are known as fish allergens. So the concentration of these proteins in fish muscle is an indicator whether by consuming that fish we are going to develop allergy or not. And particularly, though fish allergy is not that common in India, but it is now in an increasing side. We may not be actually accepting that, but the thing is that there is an increased incidence of respiratory disorders as well as asthma. So people with asthma, when exposed to this type of fish allergens, the severity increases, and their particularly the IgE level, the immunoglobulin E mediated hypersensitivity reaction rises, thereby increases the chance of hospitalization for these patients. Besides this, pravalbumin has a particular problem because it's a very conserved protein and is present in almost all animal tissues, and particularly in the muscle. But the thing is that over the course of evolution, it is so conserved that if a person is allergic to any such type, it has an EF handle, which is conserved. And this so tremendous cross reactivity with other proteins as well. Like if you are consuming like a goat meat or the frog meat or whatever it is, it's most it has a tremendous cross reactivity and mounts a severe hypersensitivity reaction, thereby increases the chance of hospitalization for this. So yes, we are advocating for fishery and fish catch for the riverine system. This is from the riverine system. This may be true for our culture ponds as well, because urban ponds also have the similar type of effluents discharged into it. This urban effluent also are pretty rich in heavy metal pollution, and so this we should keep in mind when we are advocating for fish sale, fish marketing. There should be a quality control in place, or a SOP for this quality control, that these food species, these fish mussels doesn't contain the allowed limited labels of these allergens, because in the long run it is not only the allergic symptom, it also responsible for small cell lung carcinoma. So we may be consuming that, okay, zebra fish is very fresh and this is just cut, and I am consuming a fresh meat which is white meat, so I am not going to be fat. I am getting my required amount of omega-3 fatty acids which will combat my aging process, but actually we are consuming poisons along with it. So this should be taken into account in all pollution monitoring programs, that it is not only the allowed level of heavy metals that is present in the water sample to be taken as an indicator of pollution, but also how much is accumulated in different organisms as they can transmit through the food chain and reach the human system as well. So be conscious, be cautious, and try to follow the SDG goal by being absolutely pollution free or zero tolerance for these pollutions. Thank you all. So this project was supported by UGC as well as the Higher Education Department of Odisha, and this is the PhD work of my student Dipali Mahanti. And I would like to take any questions you may for. Sir, actually, I have done another one. I have not presented it over here. I can say you a small step towards it that has also come off in environmental toxicology and pharmacology, where we saw that simply co-culturing spirulina with it and harvest the spirulina every day, so that you can concentrate these heavy metals in spirulina itself, so that water is relatively free from this contamination. There might be some other plant species or algal or cyanobacterial species which can be co-cultured along with the fish so as to reduce the pollutions.
Okay, if you co-culture in a pond, okay, the spirulina will grow,
yeah,
and they will die, accumulate heavy metal, they will die.
Yeah,
and they will go down in the bottom of the.
That's
bottom of the pond.
Yeah,
and then again biodegraded, and the
metals will be. So that's why I said that no. That's why I said that you have to co-culture it and harvest this spirulina species for degradation. So we did it for 3 months, and it's pretty accumulation is pretty good in that, and the fishes are quite healthy.
So you have to take it and throw it somewhere else.
You have to actually again, like you have to modify that one and then do it things. So something has to, because with development we have to, there has to be pollution.
You have to throw it in land,
Yes, land will suffer. Yeah. Very difficult thing, very persistent, very difficult. But at least all the fishes will not accumulate the similar type of heavy metals, neither they will be that much susceptible to develop the allergens. So we can, just like we have a guideline, how much should be the heavy metal concentration in the fish species. Similarly, we should also have a guideline, how much should be the allergen concentration in these fishes, which will be ready for good enough for human consumption. Thank you, everyone. Thank you, sir. It's always energizing to discuss with you something academic and to learn a lot from you. Thank you very much.
Thank you so much, ma'am. So that was very insightful. And with that question, it became a little more interesting. So now let's have Dr. Vijay Kumar Shahu, Dean ISSO University. He'll be talking on the topic organic farming for sustainable agriculture. Let's have him with a huge round of applause. Is he not? I guess he's not here yet. So do we have Dr. Raghunath Satpathy, Assistant Professor, Gangadhar Meher University, Sambalpur, India? His talk would be virtual, I guess. So Dr. Raghunath Satpathy would be delivering his talk over the virtual screen. He'll be talking on the topic bioinformatics tools and techniques for mining of simple sequence repeat makers in plant genomes. So you're not audible. I hope you can hear us. So we can't hear you.
Is it audible?
Hello. Hello.
Is it audible, madam?
So we can't hear you yet. So if you can check with your system, I hope.
I'm not able to share my screen. That's the problem. Chat it.
Please help him to share his screen. He is not able to share his screen with us.
Is it audible?
Not audible yet, sir. The volume is very low. So try not using your earplugs. Probably that is why we can't hear you clearly.
Is it audible, madam?
Yes, sir, it's audible. Now you can start.
Give it, full screen. Bioinformatics tools and techniques for mining of simple sequence and so this talk will be a basic overview that how these bioinformatics tools and techniques that are used for mining and repeating these simple sequence repeat markers. Let us see what are these molecular markers, why these SSRs are important.
Did we lose connection with him? Acha. So we try and reconnect with him. His link is there. I can see him though. So you're not audible. I hope you're connected with a Wi-Fi. Sir, unmute your button, please. We can't hear you. You are on a mute mode. Unmute yourself. So on your right corner, there would be an unmute button. Please unmute yourself.
Yeah, now it is.
Yes, sir. Now it is audible. Please share your screen and then you can start off.
Now it is visible.
Yes, sir. It's visible.
So let us see about this. So among different types of markers are found in both the coding and non-coding throughout the genome, and it is associated with specific traits that affect the gene regulation activity. So that's why SSRs are very, very important. So these SSRs are specific representative form of the repetitive DNA, which contains repeats of basically in the range of 2 to 5 base pairs. Origin of this repeat pattern basically due to the replication. It is very due to the replication errors, we can say, while the mispairing of the bases occurs. And this is why this contains some important features like local specificity, high reproducibility, which are being codominant in nature, and also hypervariability. So that's why SSR is very, very important. Then you have to amplify this, then you have to use electrophoresis, then score these markers. A lot of these are basically complex, time consuming in nature. And also the perfect identification means identification of this marker is also a difficult task. That's why we are to identify these SSRs. So basically, this bioinformatics is a sophisticated computational approach which is used in testing and analyzing any kind of biology or molecular information. And several computer programs and tools as well as the have been developed to facilitate this particular. Basic
the basic advancement of the genome sequencing technology is one of them. We have many number of sequences that have been published, and that is basically available in the database, and this opens the path to use these particular tools so that we can mine the SSRs for the different plants, basically what we are going to discuss today. So let us see about what is this next generation sequencing technology. So in comparison to earlier sequencing technology, this next generation sequencing technology that generates millions of small sequence read and that can be analyzed by the computer programs. As the genomics platforms are very developed, it takes very less time to sequence the complete genome. As earlier speaker was talking about something about this. And presently also many of the work has been initiated to utilize or to analyze this NGS data available in the database, and successfully the SSR markers has been mined, has been predicted along with it. And read sequence is coming and is stored. For example, the platform basically Illumina sequencing platform is very widely used because it corresponds to 99% sequence sequencing accuracy. So that's why this Illumina platform, what are the sequences has been derived? This contains 100 to 150 base pair reads are basically used, and it is more frequently. This is basically what the technique in this particular how the NGS data is generated. So the DNA it is sequencing by synthesis kind of, while the base pairing occurs due to this fluorescence dye, the image captured, and it occurs in not a single way but massive parallel way. That's the next generation is also known as a massive parallel sequencing, so that million number of reads that can be generated and that can be stored. So this is, let us see the basic roadmap for this SSR prediction. We can do two things. We can go from the whole genome data. Also, we can go for the EST sequence data to find out this or to mine this SSRs. So the next generation sequence data from the databases that can be sequence read can be obtained. Then quality control is to be done. Then sequence assembly is done. If we are getting the sequence from the EST database, then also we have to go collect this EST. Then we go for the sequence assembly. Then the SSRs are predicted. Then the functional annotation is done. So all these steps are done by help of specific computational tools. So let us discuss what type of tools are basically used to do this. The database, as I told, all this next generation sequencing data that are generated experimentally, that is deposited in a SRA is the database. It is sequence read achieve from the NCBI. It is available in this particular web. Online it is freely available. This particular NGS data is stored in a particular format, specific format that is known as FASTQ format. So let us see about how this data is there. So it is a search engine where this data is FASTQ data, and this FASTQ data contains different types of symbolic representation. Just like line 1 is a @ sign, which is again followed by the sequence. Along then there is a plus character which is optional. Sometimes you will get, sometimes you will not get. And line 4, it is called some quality values. What is this quality values? Quality values is given in basically represented in terms of some ASCII character, for example, and it is calculated by a formula that is Q is equal to -10 log 10 to the power P. where the quality factor represents a particular. For example, the quality score 10 means the base call accuracy is 90%. Similarly, if it is 30, similarly, it is 99.9%. Likewise, so depending upon, we can see the quality features. So, once we get the, there are many of the tools, as I told, the cleaning. Why cleaning is necessary? Cleaning is necessary to remove the adapter sequences that might be attached to this particular sequence while experimental sequences is performed. So there are few of the tools that is very popular tool I have given here, so that cleaning of the sequences can be done, like NGS QC toolkit, Fast QC, Clean QC, Galaxy is an online platform, EGQC. So these are few of the very popular tools that can be used to clean the sequence data, because we need only sequence to that. Then, after getting this assembly, it is a very important part of this thing. So after cleaning, we can go for the assembly. Otherwise, if we get the sequences from express sequence tag sequences, then also we have to go for the assembly. There are few of the tools that is basically used, very popular tools, they are used for this sequence assembly purpose, like EG assembler, then CLC, Genomic Workbench, Trinity, TopHat, Canoe, Cavo. So these CLC is a commercial kind of things, and it's very useful because while sequence assembly is there, we need a reference sequences always. If the reference sequence is not available, it is a related species, I can say. If the sequences is already done, if it is not available, then you can go for without the reference sequences, that is called de novo sequence assembly. That can be also performed by using this particular tool. Then the processing of this assembly data. Once this assembly of the sequences is done, then we have to go for getting these contigs. How many contigs are there? So all these things we can get it, okay, along with this SSR data. What is the particular popular tools are available? This assembly can be further analyzed to get this SSR informations, like WebSat, SSRIT, SSROM, then MISA. These are all very popular tools, and most of them are freely available. So once assembly is done, we can this assembly can be fed to this particular software to get this particular to SSR prediction things. So once the SSRs we predict, so next process is how to annotate functionally these SSRs. So this is called gene ontology based annotation, ontology based process. So many of the popular tools is in InterFloScan, BlastX, Blast2Geo, which is also commercially academic license is also available for this. Amigo, Revigo, QuickGo, and Navigo, and the rest of the things. Many I have given only popular 8 tools. There are many others are also. They are you can annotate about this particular SSRs. So what is the application? Once you annotate all these things, what we will get? Because SSR identification, as we know, experimental it is a difficult task, but in terms of bioinformatics, by using these bioinformatics tools, we can predict it, and that can be used for many approaches. For example, genetic analysis, mapping, quantitative trait locus mapping, marker assisted selection, plant biodiversity study. So, so many papers are available by the researchers. They are doing some particular task by mining this SSR. Also, we can understand the specific trait like stress resistance, drought tolerance, salt tolerance, and all other things. Also, the EST SSR based, those SSRs can be predicted from the EST sequences. That can be also easily identified, that can be used to identify the presence of the novel genes in these particular aspects. So these, let us see about the challenges. What are the challenges are there in case of this SSR prediction? So as I told, the steps, as I discussed, there are 5-6 steps we have to use. There is no complete unique bioinformatics platform is there. So you have to use this sequence and it will predict this SSRs as well as the function. So series of tools you have to use. That is one of the problems, because sometimes some of the tools require a long run time while analyzing this genomic level data. And some of the data, and also the accuracy, statistical accuracy, what we are saying, that also differs from software to software. Also, certain tools also display the platform dependence, specifically working particular operating system. Some will be in Linux, some will be in Windows, and many programs in particular this aspect, they do not contain the GUI interface. So these are not clickable. We have to do by the command line. So very much technical expertise is required to interpret or use these particular tools. So plant-specific databases containing this SSR data have been developed by researchers, but this numbers is very few. So what we can do, we can predict the SSR, because as the complete genomics sequences, we can predict this SSR. So a complete SSR-based database of a particular plant of economical interest that can be developed, and we can make a database. Similarly, novel algorithms can be used in this particular area to identify the correct SSR planking sequences and correct SSR predictions. Because as I already told, this is a challenge, because statistical accuracy is very, very important while predicting any kind of these SSRs. Then we can design of knowledge based on the gene interaction models in case of specific plant species. So this SSR function can be correlated. I can give you few of the examples that few of the things has been done for the plant specific SSR development, right? There are many of the databases available. For example, MSDP, the microsatellite database, it contains for the 86 plant species. Similarly, PMDbase, it is about 110 plant species. Then there are certain marker database like specific for Solanum and other things. Then Gramini marker database is also there. Okay, and these are few of the Oriza base, that is SSR markers from Oriza sativa, and lot of things there are, but the number is few as comparison to the number of whole genome sequences is available in the databases. So these are few of the reference and this thing. Thank you all.
Thank you so much, sir. So now we'll have on stage Dr. Vijay Kumar Sahu, Dean IAS, SOA University, talking about the topic organic farming for sustainable agriculture.
So good afternoon. The faculty and students of this esteemed prestigious Ravensa University, and all the researchers, faculties from the ICR, State Agricultural Universities, and Centurion University and other institutes, and also our NRRA Kotak, then my dear students. So I am Dr. Vijay Kumar Sahu. Basically, I am an agronomist. Now I am retired professor from OUAT and presently working at Dean Institute of Agricultural Science, Shiksha Anusandhan, Dean University. So let me thank the organizers, especially Divyansu, for giving me an opportunity to interact with you about this topic. And my congratulations to him for he and his group for organizing this excellent international seminar. So have a clapping to all of them. Thank you. Thank you. So coming directly to the topic, my topic is organic farming for sustainable agriculture. So why should you go for organic farming? And as you know that the modern agriculture has been a great help in alleviating the hunger from the world. However, even now globally, almost 800 million people still go hungry. And in our Indian context, as you know that we are the second highest populous country in the world, and gradually our population rising. And of course, our first green revolution, second green revolution, it has and the contribution of the agricultural scientists and the farmer. So we are getting our food regularly. Till that we have no problem. And about the India's own achievements after the Green Revolution, we are having highly varieties of seeds, hybrid varieties of seeds, then potential irrigation development. We are using different types of chemical and biofertilizer and different types of pesticides, including organics, and also we have a network of farm machinery improvement. But the overuse of pesticides, it resulted in the residues much above the safe level. And next one is, so there are many ill effects of the organic farming. Ill effect of the modern farming, not organic, sorry. Number one is, as you go back to different dates, our pesticide DDT, it became available for the civilian use in 1945. Then after that, Rachel Carson, he tell about the silent spring because the DDT was even hazardous not only to the birds but also to the babies who are taking the mother's milk. Then after that, he became banned. Then Rachel, a native of rural Pennsylvania, he was a renowned nature author, writer, and a poet, and a marine biologist in the US Fish and Wildlife Service. He made a survey. Then this DDT, it is most powerful pesticide of the world, and it exposed to nature's vulnerability. And the detail of DDT I should not tell, but DDT, it entered to the food chain and accumulated in the fatty tissues of the animal, including human being, through the process of biological magnification, and it caused cancer and also the genetic damage. Then coming to our organic farming, we are hearing a lot about the organic farming, and even some of the states of India, it like Sikkim, it has been declared as completely organic state. Of course, our own state, Odisha, some portion of it is still organic by default. Then coming to the term organic farming is coined by Lord Northbourne in his book Look to the Land, written in 1939. And from his conception, the farm organism, he described a holistic, ecologically balanced approach to the farming. Then the organic agricultural methods, as per the International Federation of Organic Agriculture movement, and we are under an international umbrella. Then Albert Howard, a British botanist, he referred a book, Father of the Modern Organic Farming. And also he worked in India from 1905 to 1924, and he regarded our traditional Indian farming practice, where our farmers, they are not acquainted with the chemical fertilizer. You go back to our just pre-independence period or immediately after the independence, where we got the high-yielding varieties, Taichung Native 1, we use lot of chemical fertilizer, lot of pesticide. We got higher yield, but still then we have polluted our environment. Then coming to the next, what are the goals of the organic farming as per the IFOM? It is a production system that sustains the health of soil, ecosystem and the people. It relies on the ecological process, biodiversity, and cycle adapted to local conditions rather than the use of input with adverse effect. Then it combines tradition, innovation, and science to benefit the shared environment and promote fair relationship and good quality of life for all those who have been involved. There are some synonyms of this organic farming like organic agriculture, natural farming, ecological farming, holistic farming, biological farming, regenerative farming, then biodynamic agriculture, so many things. Actually, it's not a technology, but it is a system. Then what are the different features? It gives healthy, nutritious and quality food, encourages and enhances biological cycle involving microorganisms, plants and animal, and it sustains your soil, which is our mother soil. Then it promotes long-term fertility of the soil, because what we are doing, we are applying chemical fertilizer, we are applying different pesticides, and by that we are not polluting our above ground environment, but also our below ground environment. And for the future generation, it is a caution. Then the organic farming, it maintains the genetic diversity, it minimizes all the forms of pollution that may result from agricultural practices. And here we use all the on-farm resources which are there and available. Nowadays we are calling about the different types of organic farming, and we are culturing our own material which can be reused in our own farm. Then it considers social ecological impact on farming resources, and also it fetches the minimum price. There is a time if somebody sells in the market, these are the organic products, there is no buyer. But nowadays they are buyer for the organic farming produces. Then these are the principles of organic farming, and in this, nature is the best role model for the organic farming, since it does not use any input nor demand on a reasonable quantity of water, and the entire system is based on intimate understanding of the nature's ways. The soil is a living entity. We have to think the soil as a living entity. It's not dead, because in soil there are many microorganisms, they harbor inside the soil which help us. Then the total environment of the soil, from the soil structure to soil cover, is also more important. We should keep in mind on that. What are the guiding principles? Because it uses local resources, then least effect on the environment, plant animal integration and crop rotation, and also we maintain some fallow, then feed the soil rather than the crop, maintains or conserve the biodiversity, emphasize intermixed cropping and minimum use of the off-farm inputs. Then these are the four basic principles, it and everybody know, that is the principles of health, ecology, fairness and care. Then the organic resources, there are the different nutrients. For example, we are advising the farmer that you grow a legume crop, either a green gram or a black gram or any type of legume in between two cereals. Why? Because it balances, it adds nitrogen to the soil. So the legume cropping, green manuring or cover cropping, then intercropping including the legumes, then use of farm manure, use of different compost, especially vermicompost. Now we are encouraging the farmer, you go for production of vermicompost, because the vermicompost, the Isina foetida, which is available in our doorstep, we can use it. Then use all of farm waste except plastic, then rubber, then your glass or any other material, it can be converted to vermicompost. Then about the green leaf manuring, then mulch cum manuring and the residue management, and also use of biofertilizer. About the plant protection, the different types of biopesticides like neem cake, neem leaf extract, NSK, then suppressing pest by use of bioagents, then egg parasitoids, example trichogramma, because trichogramma cards are also available in different places, either in government or private sector. then predaceous ladybird beetle, and nuclear NPVs, then Sporoptera, then Heliothis NPV, and also BT. We are now using this, we are getting in the market the BT brinjal and other thing that is also available, and use of different traps to trap the insect which are harmful to us. Then about the market and export potential, because the organic products nowadays, it has got a very high demand, and the market for organic product was very negligible during the 90s. It was only hardly $51 billion in 2018 in the world, but gradually the demand has increased, and also the estimated area under the organic agriculture in India is, even if it is there, but there is no central agency in different places to collect the sample, and also of course in our own state and in the government level, the organic certification agencies are available. Then coming to the export market, the organic agriculture export market is one of the major drivers of the green agriculture in India. The current production of organic crops is around 14,000 tons. And out of these, tea, rice contributes around 24%. Then fruits, vegetable combined make 17%. From India, around 11,900 tons of organic product is exported, that makes around 85% of the total organic production. And the major export market for India is Australia, Belgium, Canada, etc., etc. Then these are the some of the figures just I have collected from the internet. This is export from India during 2021-22, and the major commodities grown under organic certified crops are under cereal, basmati rice, non-basmati center rice, durum wheat, then our finger millet, or we call it as ragi. In Odia, we call it mandia. Then different types of spices, fruits, vegetables, plantation crops, and also the commercial crops. And in Odisha, coming, the local turmeric varieties, even if it is having low curcumin content, but if you go to Koraput, Kondhamal, and there, our turmeric, even if it is having a low curcumin content, but it has got a very good market value. And also, this innovative Jawahar Rozgar Yojana was implemented during 96 to 98. It is the Kandhamal district for the organic farmers to grow turmeric and ginger. And also in Kandhamal, the Kasam, that is Kandhamal Apex Spices Association for Marketing, it is a government sponsor organization. It has already is working. And also recently another project, Kandhamal Women's Organic Turmeric Project, it is being implemented. Then these are the export figures of the Kasam. And the future area for organic farming is the default organic area of the state like Koraput, Kandhamal, it used to gain more priority. Area where the least or moderate use of fertilizer, that you have to take it. Expansion of area under organic certification, awareness to the farmers and also to the planners and officials, promotion and enhancement of the organic market. These are the organic agriculture strategy. So thank you all. And if you have any queries, if possible, I'll answer. Thank you, ma'am. Thank you.
Thank you so much, sir. So we will now have Dr. Gyanaranjan Mahalik, from Associate Professor from Centurion University of Technology and Management. He'll be talking about conservation, cultivation, and sustainable use of medicinal plants. Let's have a wonderful round of applause for the gentleman now.
Hello. 15 minutes. Okay, thank you, ma'am. So very good afternoon, one and all present over here. So myself, Dr. Gyanranjan Mahalik, Associate Professor, Department of Botany, Centurion University. So first of all, I would just like to thank this Evasion, Agri-Vision and Ravensa University for giving me a platform to share something about my topic, that is conservation, cultivation and sustainable use of medicinal plants. So let us first move to my slides. So that medicinal plant. So what is that medicinal plant? So medicinal plant, we can say that it's a medicinal herb which contains loads of phyto constituents or secondary metabolites. So these secondary metabolites can help to cure many diseases. So plant synthesize, as we know that the plant synthesize hundreds of chemical compounds for function, including defense mechanism. Like they can defense against any fungal diseases, that can defense against any bacterial diseases, even if that can be defense for the herbivorous also. That means plant itself contains so many secondary metabolites that can defense itself. So as we know that as per the WHO, World Health Organization, so 80% of the world population, they completely depend on the medicinal plant for their primary health care. So medicinal plants are widely used as an alternative, or we can say that the complementary medicine, because they are very readily available. That means we can get any medicinal plant in and around us. Then it's a very cheaper also. And the most important is the side effect. It is very less side effect. Then the demand of for the medicinal plant is said to be increased year after year. This necessity, the conservation is required. So as we know that in our country, if I talk about the percentage wise, so if we talk about percentage wise, out of 100%, only 20% till now is identified. That means the plants been identified only the 20%. If you just thought for that, if we can identify more, another 80%, that means the new phytoconstituent or new compound we will get, which can cure many diseases. So in our country, so 17,000 to 18,000 plant species or have been identified, but out of that, only 6,000 to your 7,000 plant species been recorded. In that means they are used by Ayurveda, Siddha, Unani, and other homeopathy organizations. Then the demand for the medicinal plant is putting a heavy strain on the natural resources. As we know that the plant itself contains a secondary metabolites in their natural habitat. Okay, so this has resulted in depletion of number of higher species, which are categorized as either threatened or endangered species. So due to the heavy, we can say, high demand of this medicinal plant, now plant become in RAT category, that is rare, endangered, and threatened categories. So this is the few data, or we can say the few graphs that indicate that in Ayurveda, yeah, so in Ayurveda, 81.70% plants been used, and that followed by your folklore claims. Folklore claims means in Odia it is called as टुटुका. जो हम लोग बोलते हैं ना गांव में दादी दादा जो बोलते हैं, that is टुटुका. That means the folklore claims that is 67.97% was used, and the Siddha is the third highest, that is 56, then Unani, then Tibetan. Then if I talk about the medicinal plant parts, as we know that the roots have higher number of secondary metabolites, that means the root contain, that means the high number of roots are used, that is the 32% of the plant roots are used for the preparation of any Ayurvedic drugs. That followed by your whole plant, that is 19% fruit, and seed, that is your 18%. Then that followed 10, 8, 5, and other gum and resins are less than 2%, we can say. So let us come to the main topic, that is the important medicinal plants of Odisha and their uses, and we can say that the therapeutic uses. So you can see that, so the first picture showed that the one plant, that is the state plant of our Odisha, that is Saraka Asoka. So Saraka Asoka that belongs to family Sigilpiniaceae. The local name is your Asoka. Then if I talk about the ethnic uses, so this plant have high number of secondary metabolite and highly medicinous plant. The decoction of its bark is used in treatment of gynecological disorders. So suppose a patient suffer from any gynec problem, then in the tribal men or tribal women, they are just using the bark, the decoction of the bark, and that gynec problem will be cured. And the second picture you can show, that is the Mesoaphera, then belongs to family Clusiaceae. The local name is Nageswara or Nagochampa, we can say. So the ethnic uses the fresh flower of this plant along with the decoction of Glycosmis pentaphylla. So Glycosmis pentaphylla is very much similar with your Rauwolfia serpentina. So that means the appearance is very much similar. But if I talk about the morphological character, is very much different. So if we can use the fresh flower of the plant along with the root decoction of the Glycosmis to cure the piles. Then this slide contains your Pederia foetida, belongs to family Rubiaceae. The local name is Posaroni. Then the ethnic uses the decoction of root. Here you can see the root is very much important. The root is used to treat the colic pain, and the leaf are used to cure rheumatic pains. Then the next one is Piper longum, that is the Pipoli, belongs to family Piperaceae. The ethnic uses, we can say the dried fruit along with sugar candy. If sugar candy is not available, you can take with honey also. Is cure common cold and cough. Then next one is your your Cassia fistula, that belongs to your family Sigilpiniaceae. Then the local name is Suneri. So it's a very beautiful plant. If you roaming around the Bhubaneswar and Cuttack and Bhubaneswar, you will get in the roadside avenue plantation. These lots of plants are available in our surroundings. So this ethnic uses, if I talk about the ethnic uses, the leaves along with decoction of leaf of Tamarindus indica or imli, we can say, are used to cure skin infection, and the fruit pulp as a laxative. The next one is your Citrus grandis, that is belongs to family Rutaceae, and the local name is Batapi. The ethnic use of the leaf juice along with the honey is used to cure epilepsy. Then this is your Caleophyllaceae family, that is your Caleophyllum Inophyllum. The Odia name is Polango. As you know that Polango, the oil is very much used. The Polango oil is very useful and very high price also. Then the seed in the tribal area, the seeds are collected and the oils are extracted, and which are used against to cure urinary tract infections. Then the next one is your Terminalia chabula, belongs to family Combretaceae. The local name is Horrida. Ethnic uses the unripe fruit juice with the root decoction of Cyperus rotundus is used to cure gastritis. Then this one is Comifera mucul, because time is very short. I have to be very fast. Okay, so this is your Comifera viti. So another species is there, that is Comifera mucul, that is available in our state. So viti also available, but very few numbers are there. So it belongs to family Burseraceae. Then the local name is Gugula. Then decoction of its bark is used to reduce the cholesterol. And one very famous Ayurvedic medicines is available, that is Mahayogaraj Gugula, that is to cure the rheumatism. Then next one is Bixa olerana, that is Bixaceae family, that is Cucuma. Now this plant is very rising in condition because of the old cosmetic industry. They run for this plant for the cosmetic, any preparation of any cosmetics, we can say. Then the seeds are grounded and applied on the skin affect due to the sunburn. So these are the few plants. Of other plants, we can say some important plants which are basically cure urinary tract infection and any skin diseases, as well as to maintain or to manage the diabetes also. So I am not going through this slide, so because lots of things are there. So these are the ethnic uses. Now come to the red list medicinal plants of Odisha. So according to CHAMP or Conservation Assessment and Management Prioritization, so that data concluded that in our state 41 medicinal plants are in threatened condition. Out of that, 2 are that is critically endangered, and 11 are endangered, and 26 are vulnerable. So that 2 species which are in critically endangered plant, that are Saraka asoka, that state plant, asoka, and Symplococcus racemosus, that is Lodha. So these are the endangered species, that is that starting from Gloriosia superba, your Rauwolfia serpentina to your Gylocarpus. I'm not going through that. Then the vulnerable, the 26 vulnerable plants are there, that is Hedicium to up to your Xanthozylum. So now come to the cultivation practice of medicinal plants. So as we know that, so why this cultivation practice is important? Because the medicinal plant demand is very high, we have to cultivate that medicinal plant. So what are the various practices? That means the method of propagation. So we can say in simple language, we can say that there are 2 methods, that is the sexual propagation method and asexual propagation method. So sexual propagation means both male and female gametes are fused and that form a zygote and you will get a seed. That is the natural one. And your asexual propagation, we can say the plant propagate by their own root. That means by cutting and layering, budding and grafting are also there. These are the some advanced, we can say, technologies. And the most important is tissue culture. Nowadays, tissue culture is now rising in stage. So if we can tissue culture all the arete category plants, that means we can solve the problem of that arete category plant. That means rare, endangered, threatened kind of thing we can solve by the tissue culture techniques. So need for conservation. So why the need for conservation? I have already spoke about that need for conservation. That means it is very cheaper. It is very we can say readily available in our surrounding and the less side effect. So that means the conservation is very much important. Then the major causes of medicinal plant depletion. So what are the major causes? That means the non-sustainable destructive high density harvest by plant collectors. As we know that the medicinal plant was collected by some collectors are there. They can collect the medicinal plants. Then declination in traditional knowledge also. Then over harvesting of medicinal plant, increase international demand for the medicinal plant, commercialization of medicinal plant and increase the accessibility for the traders to remote forest areas, over deforestation. If I give one example of our state Odisha, you know very well that our state contains a black diamond, that is coal. But we think for the present situation, but we forgot about the future situation. We are for running the black diamond, we completely eradicate the diamond of future. That means our medicinal plant. They are the future diamonds. We are completely remove that concept. So before mining anything, we have to think for rehabilitation of that plants. Then come to the conservation strategies. So how we can conserve this medicinal plant? So basically we can say that there are 4 processes we can conserve, that is in situ conservation, ex situ conservation, propagation, then domestication of crop variety, then reduction of anthropogenic activity and rehabilitations of the medicinal plants. So I'm not going through that. Then the sustainable uses of medicinal plant, we can say for sustainable utilization of medicinal plant, the department, institute, agency must be identified for the preparation of document of various aspects of cultivation and conservation covering availability of the plant materials. Then the various issues to be addressed for the sustainable utilization of medicinal plant are identify the medicinal important species and study their biology. Then identify the therapeutic importance ingredients, develop scope for their commercial exploitation, then set up artificial propagation, then maintain the germplasm. That means the in situ conservation and ex situ conservation, we have to think for that. Then what is our role? What can we do for conservation? How we can conserve this medicinal plant? The first and the foremost thing is awareness programs. That is the only way we have to conserve our medicinal plant. That means awareness of new species and new variety. Then educate the drug collectors we can educate the drug collector who collect from the forest pockets of the various area. We can educate them. Then substitute a plant or with other plant of the plants. Then program to be promote a herbal garden, or we can say the herbal garden and your around, we can set up a herbal garden in or around your areas, or in or around your public places, or in schools, in universities. Then facilitate link between farmer and manufacturer. Then reduce dependence on the wild for the collection of drugs. Then biodiversity, by that manner we can conserve our medicinal plant. So finally, what I can conclude, that means we for conservation of plants, we have to be concerned about the plants. That means you know that there will be over exploitation of plant. For that, the habitat of that plant was disruption, or we can say destruction of that plant was occurred. So we have to think for the conservation by ex situ, in situ, or by a propagation method. We can conserve the plants. So with this, so thank you. And one more thing before leaving this podium. So very recently, the Ayush Mantralaya had declared a national medicinal plant. Can you tell me that name? It's a very famous. That is immunomodulator plant. That is Tinospora cordifolia. Ayush Mantralaya already declared that plant as a national medicinal plant of our country, India. That is Tinospora cordifolia. That is your Guruchi. Okay, with this, so thank you. Thank you.
Thank you so much, sir, for finishing this on time, first of all, and sharing so much of knowledge. And now requesting Dr. Ranjan Kumar Sahu, HOD, Department of Biotechnology, CUTM, to join us on this podium and talk on the topic Azotobacter vineland helps to combat chromium stress in rice by maintaining antioxidant machineries.
ना 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 दरकार हाँ हाँ बहुत Hello, good. Thank you very much to Agri Vision that they give me this opportunity to present here. Myself, Dr. Ranjan Kumar Sahu. So, good afternoon, all my respected teachers, eminent scientists, and also PhD scholars, and also some research scholars. They are present here. Thank you all. So first of all, my here topic is Azotobacter vinelandii helps to combat chromium stress in rice by maintaining antioxidant machinery. So first of all, we have to know what is stress and why to just overcome this stress, because nowadays due to stresses, it may be abiotic, it may be biotic, so there are a huge crop loss. It has been estimated that approximately 70% crop loss is due to abiotic stresses, and also 30 to 40% crop losses are due to biotic stresses. As we are living in Odisha, which is more climatic prone zone of India, and we are always affecting by abiotic or biotic stresses. And due to abiotic biotic stresses, there is a huge loss of crop. So how to overcome these stresses? There are different strategies, and one of the strategy is to use of plant growth promoting rhizobacteria. That is one of the plant growth promoting rhizobacteria is Azotobacter vinelandii. So the stress, as all of we know, that any external factor which can negatively affect the productivity of crop. How to overcome it? We have to accept different strategy. Nowadays there are different other strategies also there. to overcome these stresses. These are different types of stresses, maybe abiotic or maybe biotic. Abiotic are there are due to water, temperature, heat, and also salinity, drought, you can say. So biotic, it may due to the pathogenic attack or it may be due to the insect. So here you can say actually the world population, it is increasing day by day. So it is estimated that now the world population is 7.9 billion as of January, month of January 2022. So it may reach around 9 billion by 2050. So, and day by day, the agricultural land, their area is decreasing. So how to feed this huge population? So we have to develop some new strategy to overcome these stresses and to maintain our productivity, to maintain the yield of each and every crop. So here I am focusing about the chromium stress, that is one of the heavy metal stress. And this, due to this chromium stress, there is a huge crop loss. So how to overcome it? There are different methods that we can develop transgenic rice plants. We can edit the gene of rice plants to get new varieties which can overcome these stresses. We can also develop some varieties, new varieties through crossbreeding, and also we can use plant growth promoting rhizobacteria, because it has been reported that many plant growth promoting rhizobacteria, they are maintaining some of the for example, we can say nitrogen fixation, nitrogen fixing bacteria, potash solubilizing bacteria, phosphorus solubilizing bacteria. There are different plant growth promoting rhizobacteria. So apart from that, those bacteria, they are also responsible to give nutrients also to plant, so that and to protect these plants from the different types of biotic and as well as abiotic stresses. Here we can see our soil environment, they are affected by drought, temperature, salinity, flooding, pathogen and heavy metals. And PGPRs, they have the capacity, they are phosphate solubilizing, nitrogen fixation, they are some growth hormones, they are also producing like indole acetic acid, cytokinin, riboflavin, hydrogen cyanide, siderophore. So these all are produced by plant growth promoting rhizobacteria. So you may know that is Azotobacter, Azospirillum, some of the Pseudomonas, Bacillus species, they are all are responsible for protecting the plant. So here we have taken Azotobacter vinelandi, which is a free living bacteria. It is living in the rhizosphere of any plant, and they have the capacity because they are just not only fix nitrogen, besides nitrogen fixation, they protect the plant from the pathogen attack. Also, they produce some of the hormones like indole acetic acid, gibberellic acid, and thiamine. And it has been reported that they are protecting the plant from the fungal disease also, fungal pathogens. So we have taken this Azotobacter vinelandi, and we tried that whether it has some role in just giving protection against chromium stress or not. So for that purpose, we have just taken rice plant. Because why? Because rice is consumed by the half of the population of the world, and our India is the second producer second producer country of the world, and also Odisha, our 60% people, they totally depend upon rice. So that's why we choose rice variety here, and also we choose Azotobacter vinelandi. First of all, we tried to test the we gave some of the chromium stress starting from 50 micromole to 250 micromole, and we examine here that whether in normal condition, in normal rice condition, that if we will provide some of the chromium stress, whether the plant can sustain or not. So for that purpose, we just put our plants for 24-hour chromium stress of different concentrations of chromium, and we go for the a fold change to relative gene expression of some of the antioxidant genes, that is catalase, ascorbate peroxidase, and glutathione reductase. So these relative gene expression, we can see the relative gene expression is high, up to 200 micromole. But when it is reached up to 250, then there is some decrease of the fold chain. That means plant cannot survive with 250 micromole of chromium stress. So we just pick that one, that 200 micromole concentration of chromium, and give stress to the rice plant for 15 days. Here you can see that, and we have taken Azotobacter vinylandi, and we different concentrations of Azotobacter vinylandi has provided to the plants. That is 3 treatments are there, T1, T2, and T3, and C is for the control. That means there is no application of Azotobacter vinylandi in the control plant. Here you can see there is a tank filled with 200 micromolar chromium, and all the plants T1, T2, T3. That means T1 it is treated with 10% concentration of Azotobacter vinylandi. T2 is with 15%, and T3 is with 20%. And we have applied the stress for 15 days. After that, we have seen that the control plant, that means the plant without any Azotobacter vinylandi, they did not survive. They died, but the plants of other treatment, T1, T2, and T3, they are growing, and also viable seeds are available in those plants. So after that, we tried to see the hormone concentration at the period of stress. So that indolacetic acid, gibberellic acid, and zeatin, we found in all the treatments T1, T2, and T3, there is a higher endogenous hormone content. But in the control plant, there is very less amount of hormones, the hormone concentration are found. Then during that stress period, we have also checked the relative gene expression of the enzyme activities. On the previous experiment, we have checked it for only 24 hours, but here with application of Azotobacter vinylandi and with stress up to for 15 days, we have checked the relative gene expression. We have checked it, the enzyme activities as well as the relative gene expression. In all these cases, we have seen the treatments like T1, T2, and T3, they are performing a better. That means better antioxidant machinery is working, and with comparison to control plants, and also relative gene expression, all these enzymes are also highly present in those plants to protect them from the negative effect of the stress. Then we have done some of the electrolytic leakage, hydrogen peroxide content, some of the sugar levels, that is glucose and fructose, relative water content, lipid peroxidation, that is MDA content also. So we have checked in all the cases these treatments of Azotobacter vinylandi, they are performing better than the control plants. Here we have checked also some agronomic attributes. Those are available after the stress. You can see here with all these data, the treatments, other three treatments, T1, T2, T3, they are performing better than the control plants. So in this experiment, we concluded that Azotobacter vinylandi has the capacity to protect the plant from the negative impact of the heavy metal stress. For other heavy metals, we can also check. We can in the future plan, we can perform some other experiments using those azotobacter vinylandi, and also we can produce this data to the farmers so that they can use this biofertilizer, that azotobacter vinylandi, which has no negative impact on the plant, and it can used as a very good assets for those farmers. They are those who are struggling regularly with some of the natural calamities. So with this, thank you very much. So any questions for discussion? Sir, 200 micromolar, yes sir. Control plant diet, 200 micromolar for 15 days, continuously for 15 days. No, that we have prepared one blocked tank. That tank is filled with 200 micromolar chromium, and those plants, they are dipped with azotobacter vinylandi concentration and without azotobacter vinylandi treatment. So we found that after 15 days, the control plant died, and the other plants, they grow happily, and also viable seeds are available in those plants. Sir, actually, it has been previously reported that azotobacter vinylandi, they produce gibberellic acid, and also some of the iron chelating siderophores. So here we have not examined that one, because there is some mechanism behind it which is working to protect those plants against the chromium stress. So that because in the chromium stress, the expression level of gene also, we have seen gene expression level also high. That means the bacteria, azotobacter vinylandi, they produce some of the I think some of the vitamins, also hormones, those can protect the plant. Probably gibberellic acid also some of the role for active cell division. So it may be one of the cause for the protection. Okay, sir.
Thank you so much, sir. So we will now proceed for the lunch. It will continue for half an hour, and sharp at 2, we'll start the next session. Thank you so much, everybody. So as far as we have seen, once we start the session, people keep joining in. So we won't wait. We won't wait for a further ado. We are already late. So here we have the next session by Dr. Manish Pandey, who is a scientist, Bhabha Atomic Research Center, Mumbai. He'll be talking on the topic combined application of gamma irradiated chitosan and theoria. Wow, what a wonderful pronunciation, which I can't even pronounce. Improves growth and productivity of rice under nitrogen deficient conditions. So that's what the difference between a scientist and a common person is. Let's have a wonderful round of applause for the gentleman, please. Thank you. Sir, do you need a color mic?
No, it's okay. Hmm. Thanks for a nice introduction. Good afternoon to all, and special thanks to AgriVision for giving me an opportunity to present my work over here. The title of my presentation is Combined Application of Gamma Irradiated Chitosan and Thiourea Improves Growth and Productivity of Rice Under Nitrogen Deficient Condition. Before coming to our actual presentation, I would like to tell about some basic concepts about nitrogen and nitrogen use efficiency. Nitrogen, as we all know, is the major is a major contributor of the atmosphere, contributing about 78%, and it is an integral part of the major of the biomolecules like nucleic acid, amino acids, hemoglobin, and chlorophyll. So nitrogen is in plenty in environment, but we faces a deficiency. Why? Because the entry of nitrogen in food chain is very limited. It is through microbes and plants only. And if we incorporate some extra nitrogen in plant, it boosts the plant growth. But at the same time, the excess of nitrogen, if it goes in atmosphere, it creates undesirable kinds of thing. It is a contributor of a pollution. So the entry of nitrogen in plant is basically through either nitrate or ammonium. It get its entry in plant, and then it get fixed. So these are the three you can say these are the three steps: uptake, transport, and assimilation. They are the major target for the improving nitrogen use efficiency in plant. So now the question comes: do we really need excess nitrogen fertigation or not? Because thus the balanced nitrogen is improving the growth, but at the same time, if the nitrogen is in excess and it goes in environment, it is hazardous. So it is not desirable. So here in this data, you can show the first data. You can see that the average ammonia emission is peak in India between May to August. That is the major cropping season in India. It means whatever we incorporate in soil, major of chunk goes in the atmosphere. The second graph is a comparison between the population growth and the nitrogen fertigation. So you can see there is a steep increase in nitrogen fertigation as compared to population explosion. It simply means we are adding more and gaining less, and it is not desirable. Our primary motto is to add less and getting more. So what is the solution for that? The only solution for that is to increase the nitrogen use efficiency of the plant. So for worldwide, there are three different approaches by which we can increase the nitrogen use efficiency in plant. First is well known is a transgenic biotechnological approach. Another one is screening of germ plants for better nitrogen use efficiency. And the third one is the requirement based fertigation. When it is needed, then we fertilize it. We here use an alternate approach of plant growth regulators for improving the nitrogen use efficiency. PBRs, plant bioregulators, represents small molecules whose low dose application can boost growth and can impart a stress tolerance and can improve the yield potential of the plant, considering the nitrogen status and then economically feasibility, because there are n number of PBRs are there, but they are not economically sound. So for considering their economically feasibility and nitrogen status, we choose two of the PBRs, thiourea and chitosan, and they are repurposed for the use for increasing nitrogen use efficiency in rice. Both individuals are shown to have their growth promoting and stress tolerance power in various plants. So to start with chitosan, chitosan is a linear polymer of an acetyl glucosamine, and the source is a byproduct of fish industries. So it is available there in a plenty form, but the only drawback is that chitin is not soluble. So how to make it soluble? First, we deacetylate it, then it converted into chitosan, and this chitosan is also a very springly soluble. So BRC has developed the radiation approach to get it solubilized, and with the help of radiation, it changes the physical properties of chitosan. So chitosan, it changes its color, it changes its viscosity. The most important thing is the change in the size of particle. As you can see here, the red one is non-irradiated chitosan. The on an average particle size is 5 to 8 nanometer. When we irradiate it, then the peak shifted towards a mere 57 nanometer. So with the help of radiation, we decreases the size of chitosan, and now it is in nano formulation form. Okay. Now our second PBR is thiourea. Thiourea is structurally very similar to urea, except for the point the O group. Urea is that NH2CO NH2, and here O is replaced with that of S. And with this replacement, it becomes a redox active because it can change its form between thiol and thione's form. And thiourea is also known as a potent anti-nitrification agent. That means it can control the slow release of nitrogen from urea. So it has both of these components has potential to be used for improving the nitrogen use efficiency. For proof of concept, all the treatments like irradiated chitosan, thiourea, and chitosan and its combination is screened out individually. And as you can see here, this combination show a kind of additive effect in the form of seedling vigor and the plant performance is under stress. So this is the layout of my experiment, in which all the plants are grown in control condition up to 45 days. Then after, it is split into two batches. One batch is subjected to a full dose of nitrogen with a foliar spray of chitosan plus thiourea, and another one is only 25% is nitrogen is or this is 50% of nitrogen, and that is at the time of port filling stage is given in this and this condition is I subsequently use the term NS for nitrogen sufficient and ND for nitrogen deficient. The experimental variety is IR64. So as you can see, the effect of the treatment, this is nitrogen sufficient in chitosan. So you can see when I give in the treatment, it induces the inflorescence. You can see here the number of panicles are more in treatment as compared to wild water spray under control condition, and at the same time under nitrogen deficient condition, the number of senescent leaves get decreased in chitosan plus thiourea treatment. Now this is the time point study for the accumulation of the photosynthetic pigment. As we all know, if we deprive nitrogen, there is a very strong correlation with that of chlorophyll content. It get decreased. So here you can see that dashed arrow is with your treatment arrow. The black one is under nitrogen sufficient condition, and the red one is under nitrogen deficient condition. And you can see both under control as well under nitrogen deficient condition, the chlorophyll, the amount of photopigments is at upper hand in case of treatment as compared to their respective controls. Means the respective control for NS is the only control and deficient for its respective control. Now the photosynthetic efficiency. as there is an improvement in photopigments, there should be a improvement in photosynthetic efficiency. You can see here that is a photosynthetic rate under control condition and under nitrogen deficient condition. There is about 30% decrease in the photosynthetic efficiency or photosynthetic rate under nitrogen deficiency, and it is restricted to only 12% when chitosan treatment is given under nitrogen deficiency. All other parameters like ETR, electron transport rate, you can see here there is a improvement in ETR. So these all factors decided the photosynthetic efficiency of a plant. Now coming to, as I say, uptake, import, translocation and redistribution. These are the three primary sites for increasing the nitrogen use efficiency. So what is the status of these transporter or genes under our condition? For that, we have analyzed n number of genes like these NRT and NAR1. This is basically involved in the import or what it is, nitrogen transport from soil to root. So you can see here, this is ND, means nitrogen deficiency, and this is nitrogen deficiency with chitosan treatment. You can see the expression of NRT 2.1, NAR 2. Both get increased with our treatment, as well as this NPF that is basically involved in the translocation of nitrate from root to shoot. It get improved very significantly under our chito treatment. And this is the third one is AMT. There is a strong indication of induction of ammonia transporter under our treatment. So yeah, this is the classical pathway of how nitrogen get assimilated in leaf. After nitrogen come from root to shoot, it get assimilated in amino acid with the mediation of this NR, NIR. So there is a sequential reduction of sequential reduction of nitrate to ammonia, and this ammonia finally get incorporated in amino acid backbone, and further all biomolecules is synthesized from these kinds of. So all these NR, NIR, glutamine synthetase and GOGAT, these enzymes are mostly important for the assimilation part. So you can see here, you just see this is ND, the C is under control condition, it is nitrogen deficient, and this is under nitrogen deficient plus our treatment. So all the enzymes, NR, NIR, GS and GOGAT, all get improved under treatment, under treatment with our nitrogen deficient condition. Similarly, this glutamine synthase and GOGAT enzymatic activity get improved in sink portion. Sink portion is the portion where photoassimilate get utilized, stored, whatever. In this case, the sink portion is seeds. So this shows there is a coordinated improvement in nitrogen assimilation in both leaf as well as seeds. Finally, all things, whatever yield we get, it is a coordinated action of source and sink. Source here is a leaf and sink here is a developing seed. Source means an organ that can form sucrose or photoassimilates, and sink is an organ or something that can drag that photoassimilate for its utilization or storage. So this is a classical pathway of source-sink relationship. The CO2 get fixed under sunlight and form triose phosphate. It get channelized in cytoplasm and converted to sucrose with the mediation of two key enzymes, FBPase and SPS. This sucrose then after translocation get to sink portion where it get unloaded and sequentially channelized for the synthesis of starch. So these key enzymes like SPS, FBPase and SUSY, sucrose synthase, we quantified and found that all the activities of all three enzymes get improved under our chito treatment. So it means there is a harmonization between source-sink relationship with that of our chito treatment. And this is surprisingly, we have also analyzed this GR. GR is a glutathione reductase that is a very important enzyme that regulates the redox biology of a cell. So we have also found the same trend that the activity is improved, the GR activity is improved our chito treatment. Now coming to our final thing, that is the yield. As you can see, This is under nitrogen deficiency without any treatment, and this is under our Kyto-3 treatment. So you can see here the number of tillers as well as panicle length both get improved. And what it finally reflect? It finally reflect in the form of improved yield. You can see here there is a significant increase in 1000 seed weight. There is a increase in seed yield per plant. So overall there is a yield improvement. So this is a kind of mechanistic, what mechanism till now, what the data reflects, it is something. Kyto-T is, the formulation works on these three things. It improves the nitrogen uptake and assimilation. It increases the photosynthetic efficiency, and it increases the source things homeostasis. Both three things are interrelated. And the coordinated actions of all three improve the yield. These are some future guidelines, future lines, what we are supposed to do next. Now, the first thing, as I say, there is a clear indication of role of ammonia transport under our treatment. So we try to delineate the ammonium and nitrate ratio in response to our Kyto-3 treatment. Then the second thing is nutritional profiling, because all the nitrogen get assimilated in amino acids. So finally, this part is also pending because we want to find out what is the nutritional improvement is there under our Kyto-3 treatment. The third thing, as we know, the GR is also there and it is regulated both NR and SPS activity. So we try to establish the molecular link between redox, nitrogen and carbon metabolism. And the fourth one is multi-location field trials using widely cultivated varieties. So that's all. Thank you. I think I finished on time. Thank you. On time. Thank you.
Thank you so much, sir. Now we would be hearing from Dr. Sanghamitra Samantrai, Principal Scientist, ICAR National Rice Research Institute. She'll be talking on biotechnology in rice improvement, recent developments and future perspectives. Let's have a wonderful round of applause for the lady. Okay. Achha. Okay, it will work. Okay. It will also work. This will also work. Okay. So you can unmute this. सामने पनीर दे लो मतलब okay. Good afternoon to one and all. First of all, I am very thankful to AgriVision to give me an opportunity to give a keynote speech and biotechnology in rice improvement, recent developments and future perspectives. Everybody knows that this rice scenarios, like it was expected that in 2021 that will achieve the 127.93, and also the agriculture ministers, whatever in the speech, he has told that we have achieved the success, and also that now we are also in the surplus in rice production. Still, why we again go for much more production by 2035 to meet the growing population demand
which is predicted in 2035. Then how will bridge the gap to attain the yield of rice by 2035? Because rapidly increasing population, plating yield trend of high-yielding varieties, what it has been developed, then requirement of 150 metric ton rice by 2035. And other stress condition, we may not meet the global demand for the increasing population due to the declining resources like land, water, unavailability of labor, biotic and abiotic stress. Hence, there is a requirement of the cost-effective technology which will be discussed in my presentation. First, everybody knows that what is hybrid rice. We know that high-yielding varieties has come to the plateau. That's why if we play with the hybrid rice to achieve our goal, we have to have our more heterosis, means that hybrid vigor which can meet the demand of the population. Then rice hybrids, first it was developed by Wan Langping in indica rice in 1964. Then I am not going detail to it. It has come to India around 1980. Then why hybrid rice has not become so much popular in India? Though hybrid rice is giving much more yield, giving the heterosis more heterosis, why it is not being popular? Because it is the poor grain quality, low head rice recovery, which causes low consumer acceptability and marketability. And further, farmers need to buy the seeds every year, which is a costly affair for the marginal farmers. And because everyone knows that those who are working on hybrid rice, they must be knowing that inconsistent heterosis over the environments, it is observed, and restricted area for large-scale hybrid seed production in their country. Therefore, which technique will apply so that will make the hybrid rice to make it inbred lines and which will meet the demand of the growers? We know that conventional breeding, we can achieve the success, but it is very labor intensive, time consuming, which is not cost effective. That's what that developed technology, I will let you know that how consistent it is as compared to the inbred lines developed by through conventional breeding, and how much time it requires as compared to the conventional breeding. And here I am giving some of the examples in development technology, how it will be utilized, its application in various research. That is development of homozygous lines. When it is in conventional breeding, it is taking 8 to 10 years. As compared to the conventional breeding, development technology, it will be in 3 years, we can develop a variety which can be released also. And therefore, this approach is a very good option for the mutational studies. And this production of biotic and abiotic stress resistant tolerant plant also we can achieve. I will show that how the gene, how multiple genes in 4 seasons it has been fixed as compared to the conventional breeding, which takes several years. Then the transgenics, whatever the in the M0 generation, then how instant homozygosity will achieve by applying the double haploid technology. Then also we can map biparental mapping. That haploid technology, it can be applied for the QTL mapping to identify the candidate genes for development of the markers or for development of for the studies, and development of superior recombinants with upper yield of the hybrids along with better quality. And also, I have not mentioned here, in also this developed technology, if it is applied to the CRISPR, this genome editing technology, instantly also, if there is some heterozygous condition, we can achieve the success in getting the homozygous instantly. Then this is the significance of double haploids. What I have already told, that if we go through conventional breeding, we will be getting heterozygous. And here you can see that how meiosis it is working, and then how these haploids, whatever haploids we are getting, it is getting doubled, and in conventional breeding, when we are getting that is in heterozygous condition. Then this is the significance of double haploid, which first of all Blacksley et al. 1922, Datura stramnonium, he identified one mutant, and subsequently Guha and Maheshwari, they produced embryos from anthers which were published in Nature. These are the reports in the Nature. And what are the different methods involved for haploid production? Then in vivo methods means we will be going for distal hybridization, crossed followed by chromosome elimination, bulbosome technique, parthenogenesis, inducer based approach. But I am focusing on in vitro methods of androgenesis. Then androgenesis is nothing but that is anther culture. Then in rice, how we have got the success in our research in rice development. This is the anther culture. This is how we are taking over, that is rice plant. Then we will take the anthers. Then from the anthers, the microspore, which is haploid in nature, that will be producing kernels, and then it will be go for regeneration. Then we are getting the plants. From the plants, how we will discriminating the double haploids and haploids like this. And it is in one season, if you get the double haploids, next season, in 1 year, you can achieve the success like double homozygous plant production. Then in rice, anther culture, though anther culture is the simplest, most efficient technique, but in indica rice, it has shows its success in japonica rice. But in indica rice, it is recalcitrant in nature. That's what we have taken the challenge to develop one androgen, that universal protocol in androgenesis, which can be taken up by the rice researcher to get the instant homozygosity. That this is the two-step method, what I am discussing now. This is because we are developing kernels, then we are going for organogenesis, then we are developing, then whatever the so to regeneration, we are getting the roots, then the double haploid and the haploids we are getting. What are the factors affecting the response in vitro? These are the chemical factors and some physical factors also involves in the success of androgenesis. Then first, everybody must be thinking that androgenesis, it is very simplest method. You go take the anther, you culture it, you get the plants. but it is a very cumbersome process. That's why this indica rice was not capitalized for this, for the using in the research purposes or developing the DH plants. That's why means when we are taking the microspore in the anthers, there are microspores. The micro stage, it should be first of all identified which stage we should take, which can give the better result. Then from these are our students' work. These are the colors, whatever it has been induced in indica rice, especially. Then the multiple colors you can see. Then green shoot induction from Kalai, developed from anthers, then the plants, then another obstacle, another frustrating problem, it was albino plant. That's why it is not being capitalized, which we have been eradicated this problem, and we have applied one patent, and also the last hearing it was done. We very near future we will be getting that certificate. And these are the stages of androgenesis. And these are the DH plants, whatever it has been developed, it is being grown. These are being grown in the net house and showing promising growth, and which are in the also tested in the field. It is showing whatever the inbred lines, whatever the DH lines, it was developed, it is showing at per yield with the hybrid. That's what I think our that the problems of the farmer we can definitely nullify by giving the seeds of these hybrid derived DH lines. These are some of the publications. And this is the another application of DHs, that stigma exertion was a major problem in the hybrid rice, because if there is no stigma exertion, then we cannot get the seeds, sufficient seeds, F1 seeds to meet our demand. That's why what we have done, that we have taken one cytoplasmic male sterile lines with the restorer, which was showing good stigma exertion. Then we have developed the DHs, and which showed that high stigma exertion, like 40 to 80%. Then it will be also utilized in this. Now we are converting it into CMS line because it is in the maintainer line. That's why we are converting it to the CMS line, cytoplasmic male sterile lines, which is used in the test curves also. It is showing high seed yield. And simultaneously, we have work done through this androgenesis. We tried to fix the genes, which was quite promising. Means four genes, you can see that 4 donors we have taken and we have intercrusted, and from the intercrust, whatever the DH these were developed, some of the DHs, it carries all the genes. Means what we can tell, that DH is efficient enough to fix the genes within 4 seasons, where conventional backcrossing approach, if we follow, then we can say that it may take several years together for fixing all the genes in one plant. But a case is, everybody's mind, it will be coming that the background recovery. Background recovery means if we want to go for background recovery, then also we can go for a backcross, then 2 backcross, and then 75 to 80% background recovery. Then we can produce the DS, and all the characters along with the background recovery, we can get it. And another interesting, what I was telling, also QTL mapping. Then interestingly, when the Savitri Pokhali, through conventional approach, F1 some real population, it was developed. But unfortunately, it was, these seeds were not germinated. Then what to do? Then because somebody is doing some research work, some PhD work, then they cannot wait for several years together to get the PhD. That's why what we have done, that we have taken these two plants and made them crossed it, and F1 developed. Then out of that, several plants were produced, and the DH, whatever the DH, 118 DHs were further taken up for this QTL mapping. You can see here that these are the QTLs. It was mapped by Sandeep Singh. He is sitting here. He has completed his PhD with this work. These are the QTL mapped for several characters of the dot. And these are the chromosome position where we, and QTL name, whatever the, how many traits you can see that we have achieved it through this double haploid technology. You can see that what is the beauty of the double haploid technology now. Then coming to the marker state selection, if double haploid technology integrated with marker state selection, then our research, our this time, it will be very much certain. That's what. But marker state selection, how we'll get the markers? That DNA markers is a DNA sequences located in close vicinity of the gene of interest. Sometimes it may occur in the gene itself. That's what we have to do. Then first of all, we have to go for QTL mapping, and from the QTL mapping, then we will go for when we will be getting that QTL, then that QTL will be going for fine mapping. Then we can get the candidate genes, and candidate genes we can validate it, and out of that, then from the gene we can develop the marker, that is functional marker. And another way, the marker trait association, that was several SSR markers have been developed, which can, suppose, for example, it is the submergence. Submergence means we have to see the genomoid association. This will take many markers, and we can correlate the traits, which traits is giving that submergence tolerance. That is, in two approach, we can develop some of the markers. Then this is the what I have told, that marker validation in an alternative genotypes population, and whatever the marker developed, then that is marker state selection. We can utilize in, suppose any germplasm, you have to screen it, evaluate it, then these markers can be applied to get your desired traits in the desired germplasm. Then also it is the marker state breeding, whatever I have told, that suppose with the submergence trait we want to identify in some of the germplasm, some of the plants, then we can go for 2 plants, and then we can develop that F2, and from this we can, through marker state selection, we can identify that whatever the susceptible, what is the tolerant plants, and also further QTL analysis we can go. Then pyramiding of multiple resistant genes in a single cultivar through marker state backcross breeding, that if markers are available, then what I have told, then we can integrate the DH approach along with the marker state selection. Then in one way we will be achieving very quickly, and in another way, through conventional breeding, if we go, then it will take several years together. That's what, if we take the markers and identify these traits, then it will be very much this thing for useful to the rice researchers, and means to shorten the breeding cycle. Then another interesting is apomictic line. Hybrid rice, I have told the problems. Then if we fix the apomixis, already this type of work has been done in China, and that's what if we go for the genome editing in this, for this making the clonal propagation of hybridized, I think it will solve all the problems. That first of all, 3 meiotic genes which are involved in this apomictic, to make it apomictic, that is 3 genes REC8, pair 1, already it has been identified, OST1. And when it was 3 genes, it was edited, these were edited, what the researchers they found that it is giving tetraploid plant after cross-pollination. Then we have to develop the double haploid and subsequently double haploid plants. Now what we have to do? Then we have to, then another haploid induction lines, it was identified, and another way, baby boom, it was the ectotopic expression. It was in ovule, but it was expressed in the sperm cell. That's a 2 approach they followed, and they got the desired, that clonal propagation in hybridized. Then this, whatever the that baby boom gene already I have told, and this Miami genes. Miami genes means it produces the haploid induction system, and from the haploid induction system, we can this with all these 3 genes. It was again targeted, and where they got the clonal propagation. But one problem is there: the fertility is very less, like 6 to 7%. That's that is the schematic model of 2 clonal reproduction strategies through genome engineering, that Miami and baby boom, these 2 also we can take, and these meiosis genes, if we manipulate it, then we can achieve the apomictic line. Then CRISPR-Cas also, our laboratory, they are working, and they are targeting that already haploid inducer line. One of my student is working, and now even if the wild lice and all the germplasm he has studied, and that our intention is they are getting the fertility very less, how to get the success rate more. That's why we are now targeting the haploid inducer line. Then haploid inducer lines, already this type of work already has been taken up, and also we have got one project, and these are the haploid lines. That this is the schematic diagram. I am not going detail to it. And this in our laboratory also we are targeting the yield-related gene, because we have to achieve more success in terms of yield. That's why one IPA1, that is ideal plant architecture, we have edited in Swarna and Navin. We have achieved more than 23% yield, which in future, if the policy, if there is the policy, if it will be the government, they will be neutralizing this policy, then we can now, then I think it will be the definitely it will fulfill the demand of the farmers to achieve the doubling the farmers' income. Then to summarize this in brief, hybridized technology is such a miracle in rice breeding that increase the production and to achieve the yield plateau. It is possible to use unfair as a breeding approach, as new varieties can be developed in a short period of time as compared to conventional breeding approach. Marker estate selection, if integrated with double haploid technology, it can enhance the time requirement for as against the this conventional breeding, and apomictic line also can fulfill the demand of the farmers who want them. When it will be fixed as the hybrid vigor more, then it can be utilized as a variety which will fulfill the demand of the farmer for the doubling of the farmer's income. Then editing of yield-related genes can be fulfilled the demand of the growing population. Thank you.
Thank you so much, ma'am. And now requesting Dr. Samrendra Mahapatra, HOD, Department of Agri Business Management, OUAT. Sir will be speaking on the topic strategies to double farmers' income by 2022 through sustainable agricultural system. Please clap for the gentleman and let's start the presentation. Thank you.
রেকর্ড হয়েছে। Good afternoon, all participants and delegates, those who are present here. So, is a brief presentation of summing up what are the efforts, initiatives being taken by the corporates as well as government for improving the productivity, profitability, sustainability of in the agricultural system. We see briefly, the strategy is for developing the primary sector to raise the farm output as well as the food security, especially the focus on nutritional security rather. Green revolution of 60s made India self-sufficient in food as well as a net exporting nation. The diversification process of improving the productivity, efficiency in the resource use is the key to hike the cropping intensity. The government initiatives towards developmental as well as technological advancement through various schemes, programs, and acts that we will see subsequently. Here is a brief idea about the average yearly income of Indian farmers as per NSSO data in 2013. Punjab being the highest, in then decreasing order it is given, is 2.2 lakhs, Haryana 1.7, J&K 1.5, Kerala 1.4 lakhs, Karnataka 1 lakh, Gujarat 96,000, Maharashtra is 88,000, Rajasthan again 88, Tamil Nadu 83,000, Assam 80,000, Madhya Pradesh 74,000, Andhra Pradesh 73,000, Chhattisgarh is 62,000, Odisha 59,000, Uttar Pradesh is 58,000, Jharkhand 56,000, West Bengal 48,000, Bihar 43,000, and all India average 77,124. If we see the growth rate, the income growth rate, it was 21.5% on average from between 2003 to 2012 and 13. And if you see the growth rate between 2012-13 to 2018-19, it is around 4.5%. So it shows there is a decrease in the farmers' income on an average. Agricultural Orientation Index India is among the top 10 nations in government spending towards agriculture. The Agriculture Orientation Index is developed as a goal for the Zero Hunger of 2030 Agenda of Sustainable Development Goals. As per SDG 2 emphasizes increase in the investment in the rural infrastructure, agriculture research, extension services, technology development for agricultural productivity and poverty elimination in the low as well as middle income nations. AOI is the ratio of the government spending towards agriculture and agricultural sector contribution to the gross domestic product. India is at 38th rank in the world in AOI ranking, and India is the largest producer as well as consumer for several crops. India is having low productivity with average land holding size of 1.08 hectares. The budgetary support is low in crop insurance, minimum support price, market intervention schemes, and price support schemes. The intensification of the government spending in agriculture sector is key to attaining the sustainable development goals for higher growth. The capital investment is crucial to price support programs in agricultural sector. That is the reason FDI is allowed foreign direct investment as well as private investment in the field of agriculture, because government alone spending is not adequate. Focus on irrigation and market connectivity, integrating the small holding farmers into agricultural supply chain. If you see the Government of India policy initiatives and efforts, the Federation of Indian Farmer Producer Organizations and Aggregators called FIFA, supported by NAFED, provides market linkage, market information, business plan, then cluster-based business organizations, input, and grocery sales. The farmer producer organization is key to development, which empowers the 85% of the small and marginal farmers in India. The production of high value agricultural produce by value chain analysis as well as mapping to know closing the gap between the existing and what is the benchmark in terms of production, product quality, the processes as well as the services. So to identify the gap and to thereby to close the gap by mapping of value chain, and that is the concept of value chain development, especially focused on high value agricultural products. Then through contract farming, as you know, it is shifting the risk, the market risk for the producing farmers, while tying up with the corporates for selling their produce with respect to specific agricultural commodities, the quality with respect to the production schedule, as well as the price. All these aspects, they have to tie up with the corporates. And government is also modified the Contract Farming Act in the interest of the majority of the stakeholders, that is the producing farmers. Then focusing on integrated farming system, that is the crop and livestock to go side by side, supporting each other in eco-friendly manner, and also improving the productivity, profitability, as well as sustainability. Then the concept of Kisan Rail of Government of India, which entitles 50% subsidy for carrying the perishable agricultural products from the farmland to the nearest marketplace. So that is through the specific railway service called Kisan Rail. And the concept, the support through the agri-commodity exchanges, especially the measure to Commodity Exchange, that is NCDEX, National Commodity and Derivative Exchange, as well as the multi-commodity exchanges, which is started in Mumbai in 2003, and giving a platform with a transparent transaction of agricultural commodities between the registered farmers, farming as a producers, as well as the registered vendors, and getting maximum mileage out of that in a win-win situation. Then the major government schemes which supports is like example the few, the Prime Minister Fasal Bima Yojana, which is the modified version, covers all stages of crop cycle and post-harvest risk with the low premium. And then next is the because this again covers the risk, production risk, and also against the natural calamities and other adverse conditions. Then Prime Minister's Kisan Samman Nidhi scheme with 100% support, and Prime Minister Kisan Mandhan Yojana for old age pension to the small farmers, small holding farmers after the age of 60 years at the rate of 3,000 per month. Then Prime Minister Krishi Sinchai Yojana for improving the farmers' productivity, efficient use of the water resources. Then Atmanirbhar Bharat for agriculture and rural development, especially huge financial support given by Government of India for improving the agricultural infrastructure and market reforms, and increase in the minimum support price for the Kharif as well as Rabi crops at the rate of 150% of the production cost. Then soil health card to the farmers based on the soil conditions and how to improve the against the soil degradation, soil salinity and other adverse conditions, and guide properly what is the best use of the fertilizer NPK combinations and plant nutrients, micronutrients to improve the soil health. Then pot drop more crop, that is the slogan, through drip and sprinkler irrigation for water productivity, efficient use of water resources through this system been focused. Then Paramparagat Krishi Vikas Yojana, again focused on the organic farming for sustainability. And then your concept of e-NAM and e-trading, that is an online national agricultural marketing portal, which is linking to not only the under the APMC market yards exist in various states, also it links to the agri commodity exchanges, giving a wider platform across the nation to the farmers to get best mileage out of that and maximum price realization for their produce. Then promoting agroforestry like bamboo mission, etc. That also helps in improving the income. And Prime Minister Annada as Sarankyan Abhijan to remunerative prices to the farmers' produce is also a scheme to support the farmers. Then mission of integrated development of horticulture for horti business to hike the productivity, focus through various schemes, subsidy schemes being available for different horticultural crop, and not only production but processing and market linkage, entire agricultural chain. Then relief to the farmers affected by natural calamities, support by the government through the financial institutions and banks. Then Kisan credit card to the farmers for animal husbandry and fisheries sector, which is included. Then market reforms through Model APMC Act and APLMC Act, Agricultural Produce and Livestock Marketing Committee Act, and Grameen Agri Markets as the aggregation platform, which helps the larger sector of the farming community. and agri export policy to double the income by 2022 as the target set by through the APEDA and MPEDA, the apex organizations formed by the government. Then Farmers Produce Trade and Commerce Act 2020, that is a reform, market reform ordinance which is passed by the parliament. And the second one is Farmers Agreement on Price Assurance and Farm Services Ordinance 2020. Again, the second farm reform ordinance passed by parliament. Third one is the Essential Commodities Act modified in 2020. That is also helps a lot for improving, increasing the business, the commercialization mode, the traditional agriculture, trading in bulk quantity and eliminating the stock limit restrictions. Then promotion of 10,000 farmer producer organizations in general and farmer producer companies in specific target set by 2024 with the support of apex organizations like NAFED, who has been entrusted the responsibility for funding, as well as the NABARD and NCDC, National Cooperative Development Corporation, and the fourth is the SFAC, Small Farmers Agri-Business Consortium, who is responsible for providing the seed capital to the small and marginal farmers, forming the farmer producer company with the slogan of one crop, one district, that is identifying the major production clusters district wise and thereby combining the small and marginal farmers, but the farmers interest groups, and then joining to form the farmer producer companies with the support of these apex organizations, that is NABARD, NAFED, NCDC, and SFAC, Small Farmers Agri-Business Consortium. And for healthy, not only formation of farmer producer companies, but also how healthy functioning of these organizations and better professional management. And as you know, this is a hybrid structure of corporate as well as cooperatives. It is taking the benefit of cooperatives as well as corporate. The FPOs have been formed and it is in the process. Empowering, that empowers the small and marginal farmers who constitute around 85% of the farming community in India. Then the Corpus Fund for micro irrigation of 500 crores have been allotted. Then Agricultural Infrastructure Fund of 1 lakh crore being provided for improvement of agricultural infrastructure and amenities for not only production but processing as well as market linkage in the entire supply chain in the agricultural system. So, in conclusion, measures for sustainable agriculture development with the focus on water conservation and water storage structures, incentives for drought tolerant crop species, and managing the crops to reduce the water loss, which is a perennial problem, that is efficiency in water uses, and renewable energy application in agriculture, that is the biogas, solar. As you know, the solar pumps and other solar water heating systems and all have been given priority with the subsidy, and biogas plant have been installed with the slurry being used as a organic manure. The byproduct of biogas plant is used as a manure in the plants, organic manure. So it is a multi-purpose use. In the villages also it works for electrification. So and windmills, all the aspects of the alternate renewable solar energy sources have been developed, and now the government is trying to increase the production of these alternate sources of energy, and also reducing the manufacturing cost to make it affordable to the majority of the sector. Then crop rotation and as well as crop diversity. So that will end the monopoly, and this also the continuous use of the land for a particular crop, monocropping, which reduces the soil fertility. So there has to be improvement through soil crop rotation as well as crop diversification. Then integrated pest management approach, which best use of the not only the fertilizer but biofertilizers and organic manure, so that to improve the soil fertility and reduce the dependence on the chemical fertilizers, and eliminating the chemical pesticides. Then integrated nutrient management practices for the best use of the micronutrients and the NPK ratio. Integrating the livestock as well as the crops for which is supporting each other mutually to improve the productivity in a environmental friendly manner, which is a need of the time. And the managing the landscapes, agroecology for improving the environmental health, then economic profitability, social equity also has to be ensured simultaneously. Organic manures and biofertilizers been given more focus, and optimum use of chemical fertilizer and mix of both is also taken care. Resource use efficiency, all the resources, and also conserving the nature. Rural livelihood and social well-being is a major aspect to be considered, focused by the government. Decreasing use of capital inputs, so that will not only reduce the cost of inputs but also increase the affordability and also more sustainability through more and more use of organic manure, biofertilizers, biopesticides, etc. Sustainable intensification process, and climate resilient zero budget and natural farming instead of more dependence on chemical fertilizer. Then at the end, I will suggest the sustainable development goals that is set up by United Nations General Assembly to achieve a better future for all in 2000 by 2030 is a collection of 17 interlinked global goals, starting from goal 1, no poverty, goal 2 is zero hunger, goal 3, good health and well-being, and goal 4 is quality education, goal 5 is gender equality, goal 6 is clean water and sanitation, 7 is affordability and clean energy, and goal 8 decent work and economic growth, goal 9 is industry innovation and infrastructure. If you see, majority of the goals are linked to agri-allied sector development. Goal 10 is reduced inequalities, goal 11 sustainable cities and communities, goal 12 responsible consumption and production, goal 13 is climate action, goal 14 life below the water, goal 15 is life on land, goal 16 is peace, justice and strong institutional structure and mechanism, goal 17 is partnerships for the goals. Because that keeping in view these 17 goals set by the United Nations, all the countries are in line with that in different sectors of the society. Thank you.
So one of the presenter for this session, she'll be not able to join us today. She'll be joining us tomorrow. Hence, and the virtual sessions which we are having for this session will continue on time. So post tea break, we had one session with Dr. M. Nedusia on the topic arrowroot starch source rhizotomatous crop. He is head ICAR CTCRI Regional Center. Sir is already here with us, so we will continue with his session first. So let's have him here. And the number of people, whoever is sitting, please have a wonderful round of applause for the gentleman. So would you need the column mic?
No, so. Good afternoon, everybody. Just I'm presenting on arrowroot. Arrowroot, it is a very important minor tuber crop in Odisha. And in Odisha, arrowroot is exclusively used for starch extraction. In Odisha, there are three types of arrowroots are available. One is a West Indian arrowroot, another one is East Indian arrowroot, third one in Queensland arrowroot. So these three arrowroots are very popularly cultivated exclusively for starch purpose. And these arrowroots can be directly boiled and consumed, and can be starch can be extracted. The starch is used for sweets and bakery product production and pharma and nutraceutical industries. Queensland arrowroot, this is very popular in Rayagada and Koraput districts of Odisha. It is cultivated 125 hectares exclusively for starch purpose. However, the rhizomes are edible and they can cook and eat as a vegetable. The tubers are very delicious. Starch is extracted mainly from these tubers, and it is easily digestible. And especially this starch is given to children and invalids whenever they are suffering from any ailments. And tuber tops, especially vegetative part, is very useful for livestock feed. And this arrowroot is usually cultivated, planting starts in the April, June. Rhizome is the planting material, around 2 tons per hectare is required. And with the spacing of 60 by 30 centimeter, it is planted. Most of the time it is intercultivation with maize as a intercrop. After 8 to 9 months, the crop is harvested. It yields around 20 to 25 tons per hectare. If you see that this is a planting material, rhizome is used for planting, usually with wider spacing, 60 by 60 centimeter. So to get some intercrop yield in between, because this crop duration is 8 to 9 months, the gestation period is more. To get some income in between, they grow maize. Maize is a very good crop. You can see this is a very extensive area. It is cultivated in Koraput districts, very extensive. Each and every farmer will be having minimum 2 acres. And all these tubers are 99% goes for starch extraction. And the leaves of these plants can be used as a leaf plate. Usually what they do, farmers, tribal farmers, they periodically go to the field, they cut older leaves and they take to market and they used to sell it as a paper plate. And this is a small time business for them. They get some income because starch extraction takes 8 months. In between, this is a small business for them. And this is a rhizome when it matures. This rhizome, matured rhizomes only, they used to extract starch. This is a traditional way of extracting starch at present. If you go to the tribal farmers in the month of December, January, they start harvesting the tubers and they used to rub it on a perforated jolly, one kind of perforated jolly will be there. On that they go and rub and they collect the leachates. This is they collect the pulp, and this pulp, from this they extract the starch. What they do, the pulp, this gentleman is extracting, pulping the tubers, and this is the way of extraction. First, they used to filter it, whatever the pulp is there, they put it on an aluminum pot, and that a muslin cloth, usually whatever cloth available with the tribal farmers, they put on that the pulp, they keep and go on rubbing the pulp, adding the water. So they continue that one so that the leachates will be collected on the aluminum pot and the roughage will be thrown out. So like that they continue. Now you have seen here 3 pots are there. This is the first one where you can see that blackish water. That means already maybe 2 to 3 hours over after filtration. Here this turbidity of water you can see, this is maybe an hour before it was filled. And this lady is doing third part. So after 4 to 5 hours, whatever starch is there in the extract, that will settle down in bottom of the container. Any container, generally they use aluminum container. And they go for sun drying. Once sun dried, this starch will be like this. And they used to pack it on a polythene gunny bag and they used to store it. This starch, they used to sell it around 150 to 200 rupees per kg. And if you go Koraput, and each and every tribal farmer is having minimum 50 to 60 kg of starch. And usually this starch is collected by aggregators. Aggregators are there. They used to collect from individual tribal farmer household. And then they used to market to big agencies. This is the way they are marketing. And so imagine that 125 hectares cultivation of this species, Queensland arrowroot, Canna edulis is there. The average yield, if you are taking 10 tons also, it comes around 1,200 tons of tubers. If you crush it, at least you will get 100 tons of starch from this tuber. So 100 tons of starch is marketed. They are marketing, they are consuming, of course, 5 to 10% they consume. Every tribes, they used to keep for a year long to solve their nutritional problem. They used to keep 10 to 15 kg, remaining they used to sell. And this practice is very popular in Koraput, Rayagada. And the problem in this is, as you have seen, that they extract the starch in very unhygienic conditions. And lot of impurities are there. Mainly soil, dust, and the water they use is mostly contains lot of iron. So, regional center of CTCRI, our involvement in this is to improve the quality of starch. For that, we have fabricated one starch extraction machine. This is a mobile starch extraction machine, very small machine, costing around 1 lakh rupees. It is operated both diesel and electricity. And it's a very simple mobile, you can carry from household to household. And we have introduced this machine, and tribal farmers started using. This is a tribal farmer is extracting using this Iskanna tubers. You can see what is the difference. The traditional way, if the farmer do the extraction pulping, only 1 kg per hour they can do, and the starch recovery is 12%. Whereas when they use our machine, and the capacity is 20 kg per hour, 20% starch recovery will be there. The cost is around 1 rupee per kg only it is coming by using this machine. And by this way, you can reduce the drudgery of the woman, and also you can improve the quality of the starch. And by seeing, observing the utility of this technology, this mission, at present around 250 starch extraction machines are there in Koraput area, and they have modified our starch extraction mission. They have seen that their need is only pulping, so crushing the tuber. Other remaining things they can do themselves. For that they modified. For crushing only the need is rasper. You see this one, this is a very simple version of starch extraction machine, only diesel operated, not power operated. It costs around 20 to 30,000 only. Whereas my mission cost was around 1 lakh rupees. And further, the farmers himself, they modified the raspers. This is called rasper blade, steel blade they use. They modified it with the different perforations so that suitable for their conditions, suitable for their starch quality. And you see a farmer, a tribal farmer, is having wooden frame only. Only he has fitted the rasper, only he has fitted and extracting the starch. That means he understood his problem. Crushing is the problem. So for the crushing, he has taken my technology only rasper, and remaining thing as per his socio-economic conditions, he modified and using. And what I want to tell, this is the technology very suitable for tribal farmers, and they made some innovation on that and modified according to their socio-economic conditions. So this is a tribal farmer, happy farmer, using our starch extraction machine. They extract the starch. This is one thing. And what we are doing now, because around 100 tons of starch is available in Koraput region. So some innovators, some we can say incubative, one is joined in our institute. He want to make Palua, arrowroot laddu, Palua laddu, using that starch, purchasing from them, and he want to do export. And now he is working on that, and definitely he will be successful because resources are available, and techniques or technologies are there, and it has a great potential in the export also, demand also there, and we are working on that. The other one is East Indian arrowroot, curcuma species. There are 2 species of curcuma is there, and it is very popularly cultivated in Kyonjar and the Mayurbhanj districts only. And these 2 species, curcuma species, you know, curcuma longa is mainly turmeric. So those other than curcuma longa, some species are accumulating the starch, and you can very well distinguish this starch accumulating curcuma or not that much yellow in color, light yellow in color. Maybe in center portion yellow color and remaining portion white color. White color indicates accumulation of starch. So 2 species are very popular in Odisha. They exclusively cultivated for starch extraction. One is curcuma angustifolia, other one is curcuma jadaria. Around 50 hectare is cultivated, this both the species in Kyonjar and Mayurbhanj district, and it contains around 14% starch. Mostly this East Indian starch only you will find in many of the applications. Suppose Britannia arrowroot, thin arrowroot biscuit company, they are selling biscuits. If you go and see the content of the Britannia thin arrowroot, it is only curcuma angustifolia. Maybe sometimes they refer curcuma species. Only they have mentioned, and the starch content will be 0.02%. It is a curcuma. They very well written. It is a curcuma only. They have not mentioned the species, but only 2 species are very prominently cultivated for starch extraction. And this cultivation starts with the pre-monsoon shower. Usually the planting goes April, June. Rhizome is the planting material. Around 2 tons of rhizomes are required for cultivation. Generally, they go for 60 into 30 centimeter spacing, and it is also harvested 8 to 9 months. Generally, the harvesting commences in the month of December. It goes up to March. Beyond March, they will not go for starch extraction. Around 20 to 24 tons is the rhizome yield. So this is the crop. You can see this crop looks like turmeric only if you see from long distance. only botanists can identify the species. This is a tuber, Curcuma angustifolia. This is one of the tribal farmers field. It goes up to 5 to 6 feet length. This is a Curcuma angustifolia field in tribal area. And this is a farmer, a tribal farmer used to cut into small pieces and they go for locally grinding. This is other type of grinding so that they extract the pulp. From the pulp, again, repeated washing, they used to extract starch. This is a general practice of, in case of Curcuma angustifolia. This is another arrowroot, palua. This palua is called West Indian arrowroot. This palua is very popular. You can see in Bhubaneswar and Cuttack region, they used to boil and consume this palua. It is a very popular, and around 10 to 15% of this tuber goes for starch extraction. Usually farmers, those who are cultivating in homestead garden, they will be having 10 to 15 plants. So they used to extract few plants starch by sitting during off season, and the remaining things, this palua, they used to consume. This is the arrowroot. Generally, when you speak about arrowroot means they refer only West Indian arrowroot only. It is a Maranta arundinaceae, the botanical name is. It's around 75 hectares it is cultivated, Khurda and Nayagar districts only. And almost every household you can find this one. And this crop usually planting starts with April, June. Rhizome and suckers, both are used as a planting material. Suckers will be there. What they do, generally farmers, they harvest in the month of December, January, tuber they remove. Then again, the suckers, they put it in the soil, they water it. Again, root growth will be there. Then again, June, July, what they do, they lift it and separate the suckers. Again, they go for main field planting. Around 2 tons required if you take it as seed rate. And generally intercropping in coconut garden is very popular. In Khurda district, if you go, any household, generally in home garden, usually in a shady place only it is grown. It is a highly shade tolerant crop. If you take the biomass yield, it is the only plant gives highest biomass under shaded condition. And 7 to 8 months after planting, they harvest 15 to 20%, 15 to 20 tons per hectare yield you will find. Now, among all the 3 arrowroot, West Indian arrowroot, East Indian arrowroot, Queensland arrowroot, West Indian arrowroot said to be the finest starch in the world and easily digestible, and it costs around 500 to 600 rupees kg starch cost. The second best is East Indian arrowroot, Curcuma species. The starch cost is around 300 to 400 rupees. That is very widely marketed. Even if you go and Palua, if you ask Cuttack Medical Hospital surroundings, Palua powder, they will give Curcuma species only mainly. For Cuttack, the supply comes from Keonjhar district, Keonjhar and Mayurbhanj district. And if you go Koraput and surrounding, Koraput, Raipur and Jagadalpur, those areas, you can say those areas, it is marketed Kanna Yodulis, Queensland arrowroot, that costs around 150 to 200 rupees per kg. This is the classification of starch. And this is the way of planting. Curcuma, this Maranta arundinaceae, large scale planting. By seeing the utility of this arrowroot and as a potential for in Koraput region, this is the crop. You can see the Maranta species, West Indian arrowroot. This is the crop, and they do for staggered harvesting. They cut for 1 feet above, so that you can see it is called topping other way, so that the tubers will be, rhizomes will be maturing. This is a way of increasing the, hastening the maturity. And by observing this one, we have developed a tuber crop nursery in Koraput, where all these arrowroot planting materials will be available to the farmers. Along with that, we have taken other tuber crop also. And tuber crop nursery we have developed in Dayanedi-Guda and Koraput. It still exists, and farmer whoever want any tuber crop species planting material, they can get, and especially arrowroot, all the 3 species are available in this nursery. With this, I am concluding. Thank you very much. Try to eat arrowroot at least in a month. It is a good for health. Thank you.
Thank you so much, sir. Now we have Dr. Papa Madhyalakke. He is virtually presenting his presentation. The topic is insight of the economic impact of aflatoxin contamination of maize and groundnut in Senegal. He is from Gaston Berger University, Dakar, Senegal. He's there connecting online with us. So we hope.
Hello, yeah, I'm here.
Sir, we could hear you. Please share your screen.
Okay, I'm trying to share now.
Yes.
Oh, could you please allow me to share the screen? Okay, it's now coming.
Sir, the slides are visible now.
Okay, thank you. Okay, thank you. Hello to everybody. Thank you. I am Papa Madhyalakke Jeju. I come from Senegal. I'm at the University Gaston Berger, as you said. I really want to thank, the organizers of this AgriVision meeting, conference. And so straight to the talk. My talk's title is Insight of the Economic Impact of Aflatoxin Contamination of Maize and Groundnut in Senegal. And as an introduction, what I want to say is that aflatoxin is produced by the Aspergillus species, Aspergillus flavus and parasiticus. And the known forms are the B1, the B2, G1, G2, and M1 that is found in milk. And it is found in a lot of crops like peanut and maize that are usually contaminated in different countries. And as you can see here, aflatoxin contamination is a big concern in the intertropical region here between the parallels 35 north and south. And you can see the whole African continent is within those 2 parallels. And you can see Senegal over there. And I think India is also there. Now, how aflatoxin comes into our foodstuff? The contamination can occur in the field. And it is associated with insect damage, birds, and also stress, but also during nighttime when the temperatures are high and the condition dry. It still can also come into our foodstuff after the crop has matured. And also when the storage conditions are poor or the transportation conditions, here with a lot of associated with a lot of moisture, aflatoxin can accumulate in the crops and in the food. Now, what is the danger of exposure to aflatoxin? Aflatoxin can trigger liver cancer. All organs are really vulnerable to aflatoxin, but especially liver and kidneys are really very susceptible to aflatoxin. The potential of aflatoxin to cause liver cancer is 30 times higher in people with infection of hepatitis B virus. And it is known also to cause birth defect in children, as well as promote low socioeconomic status and cause immunosuppression. Now, with respect to our analysis framework, we consider the whole Senegalese country, and you can see the map, at your right-hand side here. From north to south, we have 2 agroecological zones. The one in north is the Sahelian zone, where the rainfall is really very low, less than 500 millimeter. In yellow, the rainfall is between 500 and 700 millimeter. And the zone in green receives at least 1,000 millimeter a year. And it is known that in Senegal, the prevalence of hepatitis B virus is around 17%. These are official numbers. Now, we also needed to collect production and consumption data, and also we had to look for aflatoxin content on stuff, but here with data that already is available that we call also secondary data. We used also the Agri software, for simulation, because most of the time the data is scarce. And now, when looking for impact, we looked for the trade impact, but here the legal framework is not existing. Therefore, the trade impact was not assessed because the legal framework was not there. And we focused on the health impact based on two approaches. The one was the doses response approach, based on the ingestion of the food stuff based on peanut and also on maize, but also we used the aflatoxin albumin adduct, meaning that we used the actual aflatoxin content in blood of living adults in Senegal to come to our result. That was based on work we did back in 2014 in the different agroecological zones. Now, always with the health effect, we used the disability adjusted life years, which represent what? The loss of the equivalent of 1 year of full health. This is the sum of two things. The years of life lost due to premature death, and also the years lived in suboptimal death, in suboptimal health, together to make the tally. And now, with respect to the prevalence of aflatoxin, now for groundnut, what we can see here, we used two reference values, the 4 ppb value, which is the threshold set by the European Union for aflatoxins, and the 20 ppb, which is the threshold set for the United States. And taking into account the quantity of groundnut that are produced yearly in Senegal, and also the probability of getting this groundnut having a quantity of aflatoxin above this threshold, we can see that for the 4 ppb value, almost 300,000 tons of the groundnut would be unfit for consumption, while when we consider the 20 ppb value, this comes down to 115,000 tons, which means a kind of challenge between food security and food safety. And when we screened the data across now the agroecological zones, you can see with the 4 ppb value, in the Sahelian zone, on top, 17% of the production would be unfit for consumption. It rised 46% in the Sudano-Sahelian zone and came down to 33% in the Sudanian zone. Now, considering the 20 ppb value, we had 2% of the production that would be unfit, 16% in the Sudano-Sahelian zone, and 15% in the Sudanian zone. Now, when we looked at maize doing the same exercise, with the production of almost 180,000 tons, you can see that for the P4 ppb mark, we will be having 130,000 tons that would be unfit, whereas when we use the 20 ppb value, only 22,000 tons would be unfit for consumption. Now, when also the data was screened across the agroecological zones, you can see both for the 4 ppb and the 20 ppb threshold that we had no data in the Sahelian zone, but that's not a surprise to it, because the rainfall is very low, so the crop cannot complete a life cycle within this zone. So in the Sudano-Sahelian zone, the contamination rate was 27%, both for the 4 ppb and the 20 ppb value. And down in the south, all the production was fit for consumption. Now, to assess the level of exposition of people to aflatoxin, we had to consider, the ingestion. And what we did was we considered the amount and also the contamination rate of foodstuff based on maize and of peanut that we consumed for breakfast, lunch, dinner, but also for snacks. And using it, we were able to calculate in a system where people eat either groundnut alone or combined per day. And as you can see here, the quantity of aflatoxin that people will be ingesting would be varying between 11.7 to 200 ppb, which is a lot. Now, with the impact on health, so here we use the data that we generated in 2014 that considered peanut ingestion and maize ingestion together, one in large amount or in different proportion. And here as an example, you can see on top that for people using low peanut and low maize, the level of aflatoxin that was found in blood was at minimum 17 and the maximum was 29.5. While in high peanut, high aflatoxin conditions, the minimum was 50.4 and it reached up to 84.2. Now, this distributed design across the country, you can see that in areas in yellow, including Dakar, which is the capital city, that the incidence of liver cancer would be the lowest, between 3 and 6 liver cancer cases per year per 100,000 inhabitants, and the highest rate being in the red zone where people produce a lot of maize, but also a lot of peanut and do consume a lot of those two stuff. Now, when we went over and calculate the number of liver cancer cases through aflatoxin exposure, our results show that in a confidence interval of 90%, it is to be expected that in Senegal, between 1,057 cases per year and 1,477 cases per year are to be expected in this confidence interval. The mean value being 1,256. And as you can see on the bottom, the GloboCan, which is the website of the International Agency for Cancer, reported 1,084 liver cancer cases in Senegal in 2020, with a mean over 5 years of 1,243, which is within our model. Now, when we went and we continued and calculate the DALI, the loss due to Senegal, we have found that for the whole country, yearly, almost 1,000 DALI are lost. And in terms of economic value, it means that every year, Senegal is losing between $92 million and $161 million, which is 0.6 of GDP, between 0.6 and 1.1 of GDP, which is a lot for small countries like ours. Now, in terms of conclusion, we can here highlight the fact that aflatoxin contamination has a high economic and social impact in Senegalese. And through our calculation, we have found that the cost of intervention should have been from a tenth to a fifth of the losses that we are experiencing only through the health impact. And till now, action is still needed, but it is still to come. This is what I had to share with the audience. I thank you very much and wish a really very successful conference. Thank you.
Sure. Thank you so much for joining in, sir. That was a wonderful presentation from your side. We are happy that you could find time for us.
Thank you. Thank you. Thank you. Welcome. Thank you very much.
Then we have a break, tea break. We'll meet at 4:30.
Come home to nature. Come home to divinity. Come home to calm. Come home to the world of art and craft. Come home to beauty. Come home to nature's nest. Come home to adventure. Come home to self-discovery. Come home to serenity. To history. Come home to eco in Orissa. Orissa, India's best kept secret. Come home to nature. Come home to divinity. Come home to calm. Come home to the world of art nature's nest. Come home to adventure. Come home to self-discovery. Come home to serenity. Come home to eco in Orissa. Orissa, India's best kept secret. Come home to nature. Come home to divinity. Come home to calm. Come home to the world of art and craft. Come home to beauty. Come home to nature's nest. Come home to adventure. To serenity. To history. Come home to eco in Orissa. Orissa, India's best kept secret. Come home to nature. Come home to divinity.
Hello.
To calm. Come home to the world of art and craft.
Check. So since we had time, because two of our speakers could not join us today, they'll be joining us tomorrow. So the last two sessions, which were to be done by Dr. Debashis Mahapatra and Dr. Shashmita simultaneously, we'll continue with that one by one. So we have here Dr. Shashmita from Ravenshaw University. She is talking on the topic biopriming of rice for physiochemical induction of defense against Xanthomonas oryzae. Okay, thank you. So here we have her. Let's have a wonderful round of applause, and the next 10 minutes is of her.
Good afternoon to all. Myself, Dr. Sashmita. I am going to give an oral presentation on the topic of biopriming. Biopriming of rice for physiochemical induction of defense against Xanthomonas oryzae pathovar oryzae. So, as we all know, rice, it belongs to family Poaceae and it is one of the important staple food of Asian countries. Then bacterial blight, it is a serious disease of rice which is caused by bacterial pathogen named Xanthomonas oryzae pathovar oryzae. Xanthomonas, it usually enters into the plant through wounds or natural openings and then it enters into the xylem. After that, it spreads through xylem and infects the whole plant. Bacterial blight, it is responsible for about 50% reduction in the yield of the plant. It causes actually leaf tissue necrosis, which downregulates the photosynthesis process. Then objectives of the study. Excessive use of the agrochemicals to increase the yield of the rice grain. It causes environmental issues and it makes the pathogen resistant to the agrochemicals. Moreover, it has been estimated by 2050, the population will increase by 9 billion and to increase the, to provide a food security, the grain yield should be increased by about 70%. So use of agrochemicals will not be sufficient to meet the food security. Then my objective is to identify safe and novel bacterial agents and fungal agents which can induce resistance in rice against bacterial blight pathogen and to understand its physiological and biochemical mechanism of induction of defense. This is the materials and method. First, seeds of Trichum native 1, which is susceptible to bacterial blight disease, has been soaked in water for 24 hours. Then after that, it is treated with the bacterial and fungal agents for 6 hours. I have chosen bacterium Bacillus megaterium and Bacillus subtilis, and among fungal agent, I have used Trichoderma erinaceum. Then when the plants were 30 days old, they were treated with the same bacterial and fungal agent, and after that pathogen inoculation has been done by leaf clipping method, and then physiological, pathological and biochemical studies have been conducted, and with the best result I have conducted the field study. These are the treatments. Then these are the chlorophyll fluorescence and chlorophyll content and defense enzyme analysis. Defense enzyme was usually estimated from 0 to 96 hours after pathogen inoculation, and chlorophyll content and chlorophyll study has been taken on 0, 5, 10 and 15 days after pathogen inoculation. All the experiments have been repeated thrice. Then coming to the significant findings, with the increase in number of days, the disease severity increases with the increase in number of days, and you can see on 25 days after pathogen inoculation, there was 95% infection in the diseased plants, whereas the treated plants, the plants treated with Bacillus subtilis and Trichoderma erinaceum, they have provided around 56 to 70% protection. This is the picture which is taken on 25 days after pathogen inoculation. The disease plant, there is complete damage of the photosynthetic tissue, and whereas in the treated plants, in trichoderma treated and Bacillus subtilis treated plants, there is very less infection. Then we have estimated the chlorophyll content. The chlorophyll content usually decreases with the increase in number of days after pathogen inoculation, which may be due to senescence. But the amount of decrease in the treated plants, in the plants which have been treated with Bacillus subtilis and Trichoderma erinaceum, was less. Bacillus megaterium also provided some sort of protection, but it was not efficient as Bacillus subtilis and Trichoderma erinaceum. Then coming to the chlorophyll fluorescence study, on 0 day after pathogen inoculation, there was almost no significant differences in all the treated and untreated plants. On 5 day after pathogen inoculation, there was decrease in the OGIP rise of the diseased plants which have been only pathogen inoculated and not treated with any of the bacterial or fungal agents. Then the chlorophyll fluorescence of diseased plant, it decreases up to 15 days after pathogen inoculation, and on 15 DAI, it was almost, the fluorescence rise was almost negligible. Then Bacillus megaterium treated plants and infected with zoo, they also show decrease in the fluorescence rise, whereas the plants which have been treated with Bacillus subtilis, they show fluorescence rise which was almost same as the control plants. Then these are the some derived fluorescence parameters, stress indicating parameters such as VJ, which denotes relative variable fluorescence at the J phase, and WK, which denotes damage to the oxygen evolving complex, and phi D0, which denotes the dissipation energy. It was high for the disease plants, and on 15 DI, it was also high for Bacillus megaterium treated plants, whereas these stress indicating parameters, they were very less for the Bacillus subtilis and Trichoderma treated plants. Then energy fluxes such as ABS by RC and DI0 by RC, it was very high for diseased plants, but it was almost same for control and other treated plants. Then electron transport, which was very efficient in case of Bacillus subtilis and Trichoderma erinaceum treated plants, whereas in diseased plants, the electron transport efficiency was poor. Then coming to the performance indices, phi P0, which denotes maximal quantum yield of primary photochemistry, and quantum yield of electron transport flux, it was very high for, it was almost same for the control and Bacillus subtilis treated plants, whereas for disease plants, it was almost, it decreases along the increasing days after pathogen inoculation. Then performance index of the Bacillus treated plant was very high, and it was almost equal as control plants, but it decreases for Bacillus megaterium treated plants and for the disease plants. Then antioxidant enzyme activity has been measured on up to 0 to 96 hours after pathogen inoculation. There was huge increase in the activities of superoxide dismutase, catalase, polyphenol oxidase, and peroxidase with the on 24 hours after pathogen inoculation, but after that it decreases in the diseased plants, whereas the defense enzyme activity of all the treated plants, mainly Bacillus subtilis treated plant, it was very high even on 96 hours after pathogen inoculation. Then as Bacillus subtilis was found to be most efficient bacterial agent for inducing defense in plants, a field study has been conducted. It has been found that Bacillus subtilis treated and disease infected plants, they have showed increase in plant height, increase in the yield, increase in fresh biomass, dry biomass, number of tillers, and the panicle number length. So the conclusion and future prospects, among different biotic agents tested, Bacillus subtilis was found to be most promising for controlling the disease. It also enhances yield of the plant in the field condition. So Bacillus subtilis, it actually alters the physiology of the plant by modifying the, it protects the photosystem too from being damaged by the pathogen attack. So future work can be done in preparing commercial formulations of Bacillus subtilis and to to understand the molecular mechanism of Bacillus subtilis in inducing the defense in rice plants. I would like to thank my supervisors Arup Kumar Mukherjee, Somendra Kumar Naik, and Professor Pradipta Kumar Mahapatra for supervising the research work, and DST for providing me the DST Inspired Fellowship. Thank you.
Thank you so much, ma'am. That was very clean and a crisp work done. Now we have Dr. Debashish Mahapatra from Ravensa University talking on the topic, a comparison between tolerant and wild type strain of Aspergillus niger while degradation of 4-OP insecticides. So here we have him. Let's have a wonderful round of applause for the gentleman.
So a very good afternoon to one and all, a respected member in the dais, my fellow researchers, and my dear students. First of all, I am thankful to the organizers for giving me a chance to talk something on my topic, a comparison between tolerant Aspergillus niger MRU01 and a wild type strain on degradation of 4-OP insecticides. So before coming to insecticides, we should know that the beginning of the human civilization started with the beginning of the agriculture. And with the agriculture, we mean man-made ecosystem, which need a lot of input of energy from outside. And to reduce the damages, we use pesticides. So the biggest problem with the pesticide is not only it affects the pest, but the residue which is left behind causes problem to the organisms which are good for the health of the soil. So main aim of my work is to degrade these leftover pesticides in the soil, mainly organophosphates, which are widely used in agriculture processes. And we know that, so to remove these pesticides, there are many processes like incineration, then leaching, but bioremediation has been proved the most useful one because of its less toxicity to the environment and very much commercially means feasible. So among the organisms used for bioremediation, bacteria are, there are too many works on bacteria, and fungus are also one of them. But fungus as an organism is much more advantageous over bacteria because of their mycelial nature and to grow even below the soil. Okay, so there are many fungal species which like Aspergillus flavus, fumigators, Niger, Sidoi, and Aspergillus terris, which have such potentials. But I have chosen Aspergillus niger for my work because it is one of the most common soil fungus found in soil. Okay, and it is non-pathogenic. Okay, only when if it is taken in a huge amount, it can cause any disease. So, and I've tried to develop a tolerant of this strain so that these OP insecticides, which are left over in the soil, can be degraded. The objectives, first is to develop a stabilized tolerant by enrichment culture, then biochemical and enzymatic analysis of the tolerant strain, then comparison between the tolerant and the wild type, then evaluation of a suitable carrier to develop a fungal preparation, then standardization of the conditions for the development and storage of the fungal preparation so that they can be distributed to the farmers for field use. So this is the overall work. First, we took a normal Aspergillus niger, normal strain or wild strain. Then we constantly enriched culture it with malathion to develop the tolerant strain. Then the tolerant strain was mixed with corbit as a carrier and packed and kept to see the activity after 60 days. Up to 60 days we have studied. So the methodologies mainly are we developed a tolerant of Aspergillus niger by repeated growth in a malathion 1 millimole. Then GC analysis was done to find out the insecticide residue after different time durations. Then esterase activity, which is one of the main defense enzyme produced by these organisms to combat against any stress. Then dehydrogenase activity and different carbohydrate enzymes were studied, which actually these enzymes mainly help the organism when it is growing in natural conditions so that it can use different carbon source as its nutrient. So while comparison, first we compared the dry weight and the fresh weight. It is clear that the tolerant strain as compared to control wild type, it's showing almost normal growth as a control is growing in the absence of pesticide. It is growing in the presence of pesticide. So the tolerant strain shows much more efficiency to grow in the presence of the pesticide as compared to a wild strain which has never been exposed to the pesticide. It is seen both in dry weight and the fresh weight. Then the 3 enzyme activity, it is very clear that the activity of the tolerant strain is much more as compared to the wild type when it is treated with insecticide, and the results of the tolerant strain is almost normal as compared to the control where there is no pesticide taken, only the organism is growing. So this again proves that not only there is an increase in growth, but also the enzyme system, which is important to give the activity to this strain, is also intact and is enhanced when it is becoming tolerant. Then esterase activity, as I told, esterase is one of the most important enzyme system which the fungus used to combat against any stress. So it is very clear that the tolerant strain, not only in the malathion in which it was made tolerant, but in all the 3 insecticides, it shows very good activity as compared to other control and the wild type. So it shows that the organism not only can degrade one OP insecticide, but it is capable of degrading broad range of OP insecticide which are available in the nature. So it's like all-in-one solution for all the OP insecticides which are used in the market. Then dehydrogenase activity, the tolerant again shows the same activity as a control where there was no insecticide used, but the wild type showed a significant reduced in the activity of the insecticide when it was treated with 500 micromole of the malathion. Then the residual activity, it is very clear that the tolerant strain shows almost double the degradation ability as compared to the normal strain when it is treated with any of the insecticide which were taken. So almost in all the insecticide, it may be malathion, parathion, chlorpyrifos and dimethod, the tolerant strain shows more removal capacity, more degrading capacity as compared to the normal strain. Then these were the fungal preparation which we had prepared. Actually, we have here I have given only one parameter, that is the effect of aeration on the fungal preparation. The red ones are the fungal preparations which have pores, and the green ones are the fungal preparation which don't have pores. It is clear that the activity is decreasing with the increase in the duration of storage, but the packets which have pores are showing more activity as compared to the packets which don't have pores. Okay, so it shows that the aeration is one of the important factor which decides that the organisms can survive better when aeration is provided as compared to when they are given an anaerobic condition or no aeration is there. Then the alkaline acid and total phosphatase activity. Here also the same trend is found. The activity decreases with the increase in the duration of the incubation period. When the maximum activity is seen in 30 days. After that, it goes on decreasing, and this decrease may be because of the loss of the viability of the mycelium, which were present in the carrier, which got reduced when the incubation period increased. Then the 3 enzymes also, these were also tested, because as I already told that these enzymes are very much important for the organism to establish themselves in the native form. So it is very clear that the preparation shows activity up to 60 days. There is not much change in this activity, though there is decrease, but the decrease is very less. So this preparation can be useful for field use, as these activities are important for the organism to utilize any other carbon source which they find in the environment. The dehydrogenase activity, it has remained almost constant up to 60 days. There is a slight decrease in the activity from 15 days to 60 days. And this is the most contrasting result, where the organism still shows the activity in 60 days where there is pores and is able to degrade the insecticide, and the residue is very less. But in case of the packets which did not had pores, the degradation is almost half, and there is a huge amount of residue left after 60 days of incubation with the packets. So these are my findings. So the tolerant strain was capable of degrading the pesticides more as compared to the wild type, which was never been exposed, and it shows almost 50% more degradation ability as compared to the wild type. And there was a gradual decrease in the dehydrogenase activity with the increase in the incubation period, and the organism shows more invertase activity as compared to the cellulase and amylase activity, which can be due to the continuous growth of the organism in the sucrose medium, because the organism were constantly grown in a Zappach-Dox medium, which uses sucrose as a carbon source, and the activity of both phosphatase and esterase was high in the tolerant strain as compared to the wild type. This is a patent which we have applied for the fungal preparation. So it is in the process to be accepted. Thank you.
Thank you so much, sir, for a wonderful presentation. Now we have this virtual session, which is a plenary session by Professor K.C. Bansal, Secretary, National Academy of Agricultural Science, New Delhi. He'll be talking about genome editing, application in agriculture, and meeting SDGs. Can we please connect with the gentleman? Very good evening, sir.
Good evening.
So, yes, sir, we can hear you.
So what's the program? Should I start or what?
Yes, sir, you can start.
can you give me a little brief on the audience? How many are there already there in the hall, or because I'm joining, I think online. Otherwise, the meeting is totally physical, right?
Yes, sir. It's in hybrid mode.
No, because the time slotted for my presentation is 4:30, so it's not 4:30.
You're right, sir. It was 4:30 earlier, but then since the students are already here, so we just shifted it 10 minutes early.
So we can start, right?
Yes, sir. We can start.
So I'll share my screen.
Please.
Host has disabled me. You have to allow me sharing the screen, please.
Yes, sir. We are doing that. Please try this now, sir. So please try sharing your screen.
I'm trying it.
Okay.
But nothing is happening. I think it's coming slowly. Can you see my screen now?
Yes, sir. It has just started sharing screen. Yes, we can see your screen now.
Is it a full slide mode?
Not in a full slide mode, but it's. Please press F5.
S5.
F5.
Is it okay now?
Yes, perfect, sir.
Can you see my full presentation?
Yes, so we can see your full presentation and we are completely audible, right?
Okay, well, thank you very much, the organizers, for inviting me to this particular mega event. As I see, there are several sessions organized, and my dear participants, dear students, ladies and gentlemen. Well, I'm so happy to be talking today on the subject which is allotted to me, genome editing, particularly with regard to applications in agriculture and meeting sustainable developmental goals. And I was told that yesterday an excellent presentation was made by Dr. Kutub Molla on genome editing, the basics. And I'm sure he has covered several aspects, about genome editing. But nevertheless, I will today reiterating some of the facts that he would have made with regard to the technology, particularly giving emphasis on applications for various traits, including resistance to biotic and abiotic stresses, and how can we use genome editing for making agriculture more climate resilient. Before I go into the details of the applications and the technology on genome editing, it's better for the students to note down the major milestones, which have ultimately resulted into the development of these, technologies through the advancements made in modern science. To start with, we all know, that DNA structure was given by Watson and Crick, in 1953, for which they got the Nobel Prize. You all know it. And then with regard to crop improvement, particularly my emphasis in this presentation, as you all know, will be with regard to agriculture. So with regard to agriculture for crop improvement, the major breakthrough came in 1960s, as we call it as Green Revolution, and which was then culminated with Dr. Norman Borlaug getting a Nobel Prize in 1970. And then this was followed by the discovery of a recombinant DNA technology, again in 1970, wherein we could recombine the DNA or the genes from different organisms into one particular gene construct, we call it. And this could be then utilized, for transferring those different genes or genes driven by different promoters, into various organisms, including plants. And the first such discovery for transferring the gene, from, let's say, unrelated sources, for example, bacteria to plants, was achieved was this agrobacteria mediated transformation method to plants, which was developed, or which was shown, to take place in 1977, and then the first GM plant as a result of this agrobacteria mediated plant transformation, which was discovered in 1977, using this particular methodology, the first GM plant was developed in 1982. I'll give you some of those details about this. And then agrobacterium actually was able to take up the transferring of the genes only to some of the dicot plants like tomato, potato, of solanaceous species, particularly brinjal, etc., but not to the monocots, which were very important agriculturally, like maize, or wheat or rice. So therefore, another method was discovered in 1986 to transfer the genes, from any source for that matter, from exogenous sources, to the plants like cereals. And the gene gun, we call it as ballistic delivery, in fact, which is utilizing a physical force, and here right now, earlier it used to be, really speaking, gunpowder, when it was discovered in 1986, but later in about, early 90s, it was, it was actually a helium-driven, a helium-driven device, which was created to transfer the genes through a physical force. And then this was then able to transfer the genes to any organism, or for that matter, irrespective, because this is a physical device, while this is a biological system already known in the literature, in the system, by the way, already available in the nature, that agrobacterium is able to transfer part of its gene, to the plant chromosomes. And so this gene gun was discovered in 1986. And as a result of all these methods, of recombinant DNA technology, agrobacterium-mediated plant transformation, the first GM crop shown in tobacco, for example, 1982. Then the first crop of importance to agriculture was developed, in 1988. We call it as BT GM maize. BT stands for, Bacillus thuringiensis, from where a gene was taken and incorporated into maize to protect it from stem borer. And that of agricultural importance crop like GM maize, resistance to insect pest, was developed in 1988. And then the first GM crop, which was commercialized in India, is BT cotton, which occurred in 2002. And then this was all followed by, these are different, actually speaking, advancements of science, which I'm talking to you, of course, particularly with regard to, agriculture. And then also after the, GM crops were developed in general globally, then the focus was more on sequencing the genomes of various organisms, including plants. The first crop rice genome was sequenced, with India as a partnership. Therefore, I'm showing this to you, particularly in 2002. And then CRISPR technology, on which, the scientific principles on which, this particular genome editing is based, was first applied to plants for genome, was in 2013. Though by itself, the CRISPR-Cas mediated genome editing took place, the birth, was in 2012. But it was first applied, in 2013. So I'm going to give you basically the details of this particular technology, but I thought to give you a perspective and put it in the, comprehensive way, is that this all, you can see here, right from 1953 to 2013, 60 years of advancement in science, and therefore development of a particular technology, which could be utilized, for the welfare of mankind. And here I'm talking about with emphasis on agriculture, and particularly climate resilient agriculture. But to also not to undermine the importance, of conventional breeding. I'm talking to you again here of green revolution, and for which the Norman Borlaug bagged the Nobel Prize in 1970. This was all achieved through conventional plant breeding methods, which are still very much important, and breeders continue to use those, methodologies for developing our crop plants. But for the students, I'm sure they already know it. The idea was that time Dr. Norman Borlaug was looking for some of the genotypes which were very small, as shown here, very dwarf, because this was the one which was cultivated by the farmers. And once they have got ear heads developed, it will lodge flat on the ground and its quality will be totally deteriorated. And that was a big problem. And as such, they were very low yielding, just less than 1 ton per hectare, for example, their productivity. So he was looking for some kind of, through conventional breeding, a source of within the plant of the same wheat plant, that which is a smaller in size, like dwarf, we call it technically, so that he could make a cross of these two through conventional plant breeding, and which he was finally able to, and succeeded in developing some of those genotypes. We call them as, semi-dwarf. This is dwarf, so they are semi-dwarf in between the two. And here also you see some genetic variation. And as a result of that, it is now not getting lost, and the productivity increased also beyond 1 ton per hectare. Now, today we are talking about 3 to 4 tons per hectare, as a result of conventional plant breeding. But then, there are many issues. Now we are talking about it. For example, this is the green revolution. I'm giving an example here of conventional plant breeding. these tall varieties were converted to these dwarf varieties. And then similarly, the rice tall varieties were converted to these dwarf varieties of rice, by conventional plant breeding. And India was proud to witness the Green Revolution. And we came out from a situation which was earlier from ship to mouth, today to Right to Food Act we have in India, actually, which was enacted in 2013. So this is the method, utilized here is conventional plant breeding, and which also we call it systematic plant breeding. And I'm sure Dr. Kudomola would have covered several other aspects, related to the conventional methods, or mutagenesis as we call it, or creating a new genetic variation, which is the basis of genetic improvement of crop plants. But then we have to move beyond these conventional methods. Why it is so important? That, first time ever, United Nations organized a food system summit in 2021, which was actually, the last day was 23rd September 2021, wherein the Secretary General, gave a message in the summary in his report that global initiatives are important, to reinforce the ambition of science-based solutions. And these science-based solutions will only act as the key to deliver on the 2030 agenda. I'll just tell you in a minute what is the 2030 agenda. But the point I'm trying to emphasize here is that while conventional plant breeding at one time was also not considered as science per se, mostly when I was a student, it was taught to us it's more of an art than science. But later, with the discovery of those genes which were responsible for making the plant tall to dwarf, they were later discovered to be as dwarfing genes. And this was all the kind of advancements which took place, in science technology. So here, this is the same actually message is reinforced, that science-based solutions are important, actually speaking, to move forward even beyond Green Revolution or beyond even GM crops, to have this increased production in a sustainable way. And this is what this whole agenda is about. We call it as 2030 agenda. And that is actually speaking, that means the United Nations have developed these sustainable development goals for themselves to be achieved by 2030. Earlier, Green Revolution was not sustainable because that was more responsive to water. You give more water in irrigation, more fertilizers, more agrochemicals. The idea was there to produce more for our own self-sufficiency in India. But once we are self-sufficient, now we can, direct our efforts more towards, the ones which are science-based. So therefore, and it has to be in a sustainable way. So these sustainable developmental goals are 17 of them. The first one is no poverty, of course, and they can all be tackled through, the science and technology advancement. So therefore, role of science technology is very crucial, even achieving these goals. Goal 1 is no poverty. The goal 2 is entirely related to agriculture, zero hunger. That whatever the population we have today or is going to rise in future, it has to be all dealt with by increased production in a scientific manner. So what is this development goal is end hunger. Development goal number 2 is end hunger, achieve food security and improve nutrition and promote sustainable agriculture. Remember, sustainable agriculture with least harming the environment, not like we had otherwise done by indiscriminate use of fertilizers or indiscriminate use of water or indiscriminate use of pesticides. No, this is all damaging to the environment. No doubt we have harvested excellent dividends and we got a lot of increased production as a result of Green Revolution. We cannot forget that. This is very important. We should always remember. But since the technology and science have advanced so much, now we can talk of sustainable agriculture. And why it is important is to further increase the production. By 2050, it is expected that another 2.4 billion people would have been added to the population of particularly the developing countries in the world, including India. If in India is today 1.37, 1.3 or 4 billions, I think it will be 1.7 at the people talking about it, I think by 2050. So globally also by 2050, the agriculture sector must expand by 60% to meet the increasing demand due to continuous increasing human population. We need to expand the agriculture sector or increase production, by 60%. And this is all possible only by increasing crop productivity. In a given piece of land, we cannot expand the land area, which is already under cultivation. So what we can do is we can try to improve the crop productivity, or we call it as land productivity, or the productivity or the production of crops, agricultural crops, more and more per unit time and per unit land area. So therefore, genetic improvement is important, and that also all has to happen under climate change conditions, which is very important. So there is a big challenge. So therefore, latest advancements in scientific technology, today we are talking about genome editing, which has all this potential to achieve zero hunger, develop more resilient and climate smart crops to counter the mounting threats of climate change and its adverse impact on global food security. So finally, while we have a climate change over the years taking place, fortunately, scientists, we must, congratulate them, we must compliment them that they have been making these discoveries in the basic sciences so as to have technologies like genome editing, which have become a boon, really speaking, to use them for addressing some of these issues and challenges, facing agriculture. So coming back to, the different advances in science and technology, to start with, I mentioned about through the use of conventional plant breeding, Norman Borlaug got a Nobel Prize in 1970. That means it was so important. Then again in 2013, these three scientists, they became the World Food Prize winners. They got World Food Prize, all three of them together. And who were these scientists? They were the ones who finally, sort of developed methodologies for developing GM crops or genetically modified crops or transgenic crops through the process of genetic engineering. I talked to you about recombinant DNA technology, which was, of course, developed in 1970 through the discovery of certain restriction enzymes and modifying enzymes, as you all know. But then the method of transferring some of those genes from unrelated sources to plants were discovered by these three scientists, Dr. Mark van Montagu, Dr. Maribel Chilton, Dr. Mark van Montagu from Belgium, Maribel Chilton from University of Washington, in USA, and Robert Fraley again from USA, who was heading, the Monsanto lab that time. And all these three of them, they got this Nobel, the World Food Prize, which is considered to be equivalent to a Nobel Prize actually in agriculture. They got it in, 2013. And they got it 30 years after. The first paper they published to transfer the gene was in 1983, as I mentioned before. And so 30 years later, they got this Nobel Prize. Now, continuing with the advances in science technology, today we have, actually speaking, genome editing, which also got a Nobel Prize 2 years ago in 2020. And this Nobel Prize actually was, I would say, the fastest after a certain discovery was made by these two scientists in 2012. I said genome editing took birth in 2012. It was first applied in plants in 2013. And these two, female scientists, women scientists, they got Nobel Prize just 7 to 8 years later in 2020. They got this Nobel Prize. So actually speaking, what's the benefit of all this, whether we talk of conventional breeding or GM crops, particularly these two technologies, before I come to genome editing, you can see here, this started in 1960. And after 1960, as a result of conventional breeding or as a result of GM crop technology, you can see the production in different parts of the world started increasing. This is the cereal productivity, actually, tons per hectare. You can see whether you're talking of, this is Western Europe, or you talk of North America, like Canada and US, or you talk of South America or Asia or Africa. In all continents, you can see here, there's an increasing trend in the productivity, particularly as shown here in case of cereals. There's a benefit to have these advancements in science and technology, to address the challenges of agriculture, and we develop and genetically improve the crop plants and develop new varieties so that we have witnessed increased production, let's say from 1960 onward until 2016. But then what is now the trend is, since we have now genome editing in place, in vogue, and it's being used heavily, in different walks of life, not only in agriculture, but also pharma and health sectors, you see this has its own potential of further increasing the productivity. These are only I have added here, from, like for example, after this every 8 years window, 2016 was, if this was the level of productivity, which was, let us say, in case of these Western Europe or North America, was close to 7 to 8 tons per hectare. Here it is 3 to 4 or 5 tons per hectare. Here it is only about 2 tons per hectare. So this can now increase further dramatically by application of genome editing, let's say by 2024, which is of course, 2 years down the line, because there are already, advances made in developing several crops. I'll give you some of those examples, and particularly meeting the challenges of climate change. So what I'm trying to say is that these technologies each have their own role to play, in increasing production, and all of them are important. You cannot say that only genome editing can do the job, or only genetic engineering, or only conventional breeding. The message I'm trying to give you, all the students, through this particular slide is that each and every technology is important, depending upon which particular continent of the world we are talking about it, which crop, which trait, under what conditions you want to increase the production, what are the challenges. So there could be only this, or only this, or only this, but the best is a kind of an integrated approach where you can utilize, all the three. And this is anyway the mother of all technologies, I would say this, actually speaking, conventional plant breeding, because you cannot develop any further variety, even alone by genetic engineering technology, or alone by genome editing. You have to resort to conventional plant breeding tools by making cross hybridization of one crop with another, or one variety of the same crop with another, finally to develop a product, in the shortest possible time. While this takes about 8 to 12 to 15 years, depending upon the crop, to develop a new variety from the time that you made a cross. This could take still up to 5 to 10 years, or at least 7 to 10 years. But this is one which can save you a lot of time and energy, and it is possible within 2 to 5 years. Okay, because this is again, sort of randomly, the genes are getting reshuffled when you make crosses of one variety with another. Here, while it is targeted gene is identified. Here, there's thousands of gene, come together in a particular F1 hybrid, and then you restore the one parent which is required. Takes a lot of time. But here, the one which is already a high-yielding variety, a targeted gene is transferred through any of the methods I mentioned to you, but the integration of that gene on a genome is random. There's no control over that. While we can find out later through molecular mechanisms that where that gene landed up on the chromosome, but then however there is kind of a position effect which people have studied, and it takes therefore some time to identify the best event which has got all those advantages of the traits which you wanted to target and develop. But this is in fact a step further when you talk of accuracy, when you talk of precision, when you talk of targeted integration, or bringing any targeted change into a genome, or targeted mutation if you want to create it in any given gene. And by this time, we know which are the genes out in a plant crop, which need to be either knocked out, or to be added, or to be mutagenized with few bases. Actually, this all could be done, through this, technology. So this is more targeted. So therefore, this kind of integration of this is very, very important. So now, in fact, I'm going to talk to you about, as I mentioned before, that the birth of genome editing took place in 2012. This is one of those two scientists. This is Jennifer Doudna in the USA, who was one of the discoverers of this technology, and a co-partner with Dr. Emmanuel Charpentier from France for the Nobel Prize. And this is her message, which she delivered on October 5, 2021 in a meeting, that CRISPR-based genome editing, I'm talking about it, will allow farmers to adapt to a changing climate and sequester more carbon while preserving prized regional varieties. There is an incredibly important role for this technology in the protection of small farmers. What the message is, if you get here, is what? Farmers to adapt to a changing climate. We did not have that during 60s, 70s. We were talking about it, conventional plant breeding that time. And we never worried too much about sequestering more carbon, in the soil, back into the soil, while preserving the prized regional varieties. That time, during Green Revolution, we promoted mostly wheat and rice, but here we're talking of all prized regional varieties, wherein genome editing could be applied, and finally, to protect the interests of these small farmers. This is the message directly coming from the Nobel Prize winner. I'm sure Dr. Qutub Mullah, who is a wonderful scientist. In fact, I'm sure all of you would know, he's a very good teacher. He's the one who has the best expertise, one of those scientists. We have very few scientists in the country, who has all that expertise. He would have explained to you, I'm sure, but nevertheless, it's so important technology. If I go with you once more, that there is a Cas9 enzyme, which creates this, kind of a nick here, which creates a double-stranded break. We call it double-stranded break. And this double-stranded break can be repaired with different mechanisms, which are possible in a plant cell. For example, this is one non-homologous end joining, which gives you a kind of deletion or addition of new bases at random, actually speaking. So there'll be function of the gene which will be knocked out. and in a way, this is, we call it in the parlance of editing, delete. Okay. Similarly, there is already a gene, here, this is the gene we want, or this is the gene, sorry, we want to correct it, and we want to replace some of those sequences here with some of the bases we wanted, and which could be achieved in a real sense. This is only genome editing. This is deletion, while this is only genome editing, actually speaking. And this is done through homology directed repair. You take a template, there is a homology, you can see here, these are the flanking sequences, there is a homology, they get recombined as a result of homologous recombination. And when they recombine, this sequence with this and this sequence with this, then this will automatically be replacing these, in a way, not desired bases here in the genome, which you want to correct in a gene. So finally, this shown here in the green will land up here, and the gene is corrected. And this is what is really editing the letters A, G, C, T, which must be present here will be different, but then we have edited it and becomes more functional. And this is called edit. And similarly, we can insert a new gene altogether, actually speaking, not only this, using the same homology directed repair system, HDR system, as we call it, which is applied or supplied exogenously into the system. So then here, this whole new gene altogether, as we do in case of transgenic crops, can be inserted on a well-defined, location onto the genome, which is not possible when we talk of transgenic development, either through agrobacterium mediated transformation or through gene gun. But however, the advantage here is, while you still develop a GM crop here, but it is targeting that particular precision addition of a gene on a well-defined location on the genome. That's an advantage. So that kind of a position effect, that kind of extra time required for selecting the best event will be saved. So therefore, it saves a lot of time. So coming down to the applications of genome editing in agriculture, and coming back to the talk given by, Dr. Jennifer Doudna on October 5, 2021, and she addressed, these two applications actually, as shown here in green. But the message was again the same, that, the population is increasing and the greenhouse gas emissions are also increasing, and the food system, including agriculture, is responsible for 35% of the greenhouse gas emissions, which is causing, warming, global warming, temperatures are increasing. So these are the two burning problems. One is population rise, another is the greenhouse gas emissions or global warming. So what is the message with regard to the application of genome editing from her? Message is, the genome editing can help feed our growing population. The message for this problem. And the message for this problem is that the plants and microbes, also microbes, of course, can be the part of the solution and not part of the problem. Okay, so we say that while food system, including agriculture, is contributing to these 35% emission of greenhouse gases. So that means agriculture is adding to the problem. Okay, but then we have a technology now that the plants themselves can be edited in a manner that they will not add any more greenhouse gases to the atmosphere, and they can become themselves a part of the solution. Because we cannot live without plants, we cannot live without agriculture. We have to have enough food anyway. So the food system is required. Okay, so therefore it is important that we have a technology which can itself now allow these plants or agriculture or the food system to be part of the solution rather than adding to the problem, and increase the carbon sequestration back in the soil. And how it could be, for example, feeding the population, how it could be done by increasing yields, I mentioned before, or increasing land use efficiency, that is per unit area of land, per unit time. How can we increase productivity beyond the way we were able to achieve, during the green revolution time or through GM crops? And helping farmers, therefore, to adapt to the effects of a changing climate, making crop plants more resistant to diseases, okay, and improving nutrition, which is also very important. And how can you reduce the greenhouse gas emissions? And that is also possible by converting, gas emissions, for example, methane, that could be reduced from rice paddies if you do genome editing in rice, or minimizing farmers' inputs, that you can produce the same amount of produce by using less amount of fertilizers or pesticides. Okay, so we are saving here the fertilizers utilization, pesticide utilization, and increasing in the process long-term carbon sequestration, in the farm soils. That's also important, and by increasing productivity. So these are some of the examples I'm giving you here of developing disease-resistant tomato plants. For example, by knocking out, one particular gene, which is, we call it as wild type allele of mildew-resistant locust, MLO. This is the gene. And if the gene is functional, the plants are susceptible to a particular disease, which is caused by this particular, fungus, which is causing powdery mildew, So this is susceptible if the gene is functional. But using SDN1 technology, wherein you can knock down the portion of the genes, you can make this plant, you can knock down this gene, you can make this gene non-functional. And as a result, you can see here, the genome edited line of tomato here becomes totally almost 100% resistant. So that's an advantage. Similarly, this is another burning issue in those areas where rice is grown, that there is there's a problem of bacterial leaf blight. When there's a problem of bacterial leaf blight, you can see here the leaves become yellow, and these plants are susceptible to this particular organism called Xanthomonas oryzae, which is the bacteria which is causing this particular disease. I don't want to go into the mechanisms of it. There's a wonderful, beautiful mechanism existing in the nature, which the scientists have studied. And as a result, they could mutate, only certain bases in the promoter sequence of some of the genes. We call them as sweet genes. So this is the genome-edited sweet gene promoters, to which certain protein effectors will bind, to produce more of glucose, so that it is transported from inside the cell, from cytosol to the cell membrane or cell wall, where the bacteria is eating and sitting and eating the plant. So by genome editing this gene promoter, you can make these plants resistant, as shown here in case of rice. Please excuse me. Moving further, this is the increased rice growth and yield. Sorry. By mutating ABA receptors. Okay, ABA receptors are known to provide some kind of resistance to abiotic stresses, particularly drought. And you can see here wild type plant versus genome edited plants. So out of different ABA receptors which is present, for example, in rice, 12 to 13 of them, if you mutate and make them non-functional, for example, if you take receptor 1 to 6, if you mutate them all, the plants grow better than the wild type under stress conditions. And then if you mutate only 3 selectively, 1, 4, and 6, the plants even behave better than the ones where 6 of the members of these ABA receptors will mutate. So these plants become resistant to abiotic stresses. Not only they become resistant, they were even growing better. So there is a combination of even better growth and stress adaptation as a result of mutation of some of these ABA receptors. And if you compare the yield per plot of the wild type, which is about 1,500 grams per plant, here it is touching almost 2,000. It is about actually 30 to 35% increase you get, particularly in this particular genome-edited plant. And similarly, this is the work done in India by Dr. Viswanathan at IRI. There again, they have mutated the function of one particular gene, that is DST, droughts and salt tolerance gene. And they have done it, and it's actually speaking the lines have been developed, genome-edited lines, and now there's a another passage going forward for taking them to the stage of commercialization. Interestingly, not only one gene you can mutate at a given time, you can do multiplexing. Actually speaking, there could be a possibility of targeted biallelic, triple mutations. So we call this multiplex QTL editing of grain-related genes, which has been done in case of rice shown here, which improves yield in some of the elite rice cultivars. Here, one of the cultivars, they call it CNXJ. So this is a gene which is controlling grain size, okay, GS3, and this is the gene controlling grain weight, GW2, and this is the grain controlling grain number. So if you mutate biallelic, two different alleles of the same gene, allele 1 and allele 2 of this gene, they mutate here. You see one is deleted, one base is deleted. Here 3 bases are deleted. Here again 10 and 1 bases are deleted. And this grain weight, allele 1 and 2. Similarly, grain number, 2 alleles are deleted here, 1 base is deleted here. And as a result of that, you see the genotype here, that this particular where all 3 have been mutated, grain size, grain weight, and grain number, you can see this, the panicle size is increased compared to the one which is non-edited line. See here. Okay, so this is an advantage, of using this technology in a very precise manner, and very precisely you are deleting some of the bases from the well-defined genes controlling a particular trait. And not only one gene at a time, you can do more than one gene. And some of the products like soybean and tomato here I'm showing, which have already been commercialized. This has been commercialized recently in Japan, a tomato which has been improved, quality with regard to the levels, high levels of gamma aminobutyric acid, which is an amino acid believed to aid relaxation and help lower blood pressure. This has already been commercialized and available off the shelf in Japan. Similarly, in India, one of our friends, Dr. Siddharth Tiwari, is working in a DBT Institute, Nabi Mohali, where he has developed these, yellow pigment containing wheat or genome edited banana, through this particular technology. Compared to wild type, you can see the banana is actually here, this particular beta carotene is 6-fold high, compared to the wild type banana. And this is, very good, because this beta carotene, if you consume, while it is present, of course, in papaya and tomato, many other crops, but those are the crops which are not actually as, easily available or being consumed by, I'm talking about you including poor people, or resource poor people. But this banana is something, which is liked by children and, and elderly people alike. So therefore, this is certainly a target crop, and it could be done in, of course, in more crops as well. So this is what is some of the example I thought of rice and banana from India. But important is also for the students, I'm sure they already know it, that, when you talk of any of the crop cultivars, they are available in a given crop, let's say, whatever the number, would be 500 or 1,000 varieties over the time, let's say from Green Revolution days. If you talk of genetic diversity of those released varieties which are available with the farmers on the farmers' field, the high yielding ones, their base is very narrow, genetically speaking, very narrow genetic base. And as a result, they become vulnerable to any kind of, climate change, whether you talk of resistance to diseases, or susceptibility to diseases, or new emerging pests and pathogens, or including some of the abiotic stress factors. They become, particularly against pathogen pests, they become very susceptible. There's a possibility, the resistance gets broken down as we normally talk about it. So therefore, the genetic diversity has to be increased. And for the increasing genetic diversity, we resort to the use of wild species in any crop for that matter. As shown here in case of tomato, or in case of rice, or in any crop, we talk about it, wild relatives are there. And we have compiled all this information on wild relatives of cultivated plants in India, which wild species is good for which particular trait. So there's a possibility of applying this genome editing now for accelerating the domestication of these wild crops by genome editing. Accelerating domestication. Domestication has taken place for the last about 9,000 to 10,000 years. Okay, but it has taken thousands of years before they were, cultivated by the farmers. Okay, later on, of course, scientists, got this systematic plant breeding, science-based plant breeding into action. But before that, it was all in natural selection. And as a result of that, it will take a lot of time for any wild species to get converted to, as we call it, as cultivated type. But through the process of genome editing, it has been demonstrated that within 2 to 4 years, you can convert a wild tomato into a cultivated type of tomato by mutating, as given here, some of the 5 genes, and particular portions of those genes, because genome editing you must know the gene structure very well. Anywhere in the gene, if you mutate, it will have its own effect on that particular trait, but you must know it beforehand. It's important. So here they selected, for example, 5 genes, and different portions of the genes. For example, here it is the coding sequence, or the cis-acting regulatory sequence, for example, the promoter sequence, or the upstream ORFs, operating frames, or it could be different structural point of view. I'm talking about targets in the genome, in the gene itself, which could be then identified and mutated to have that particular desired trait, as shown here in case of tomato. I'm sure, again, Dr. Kutub Molla would have talked to you all about it. He's the one who who is the first author in this paper. Fortunately, I happen to be a partner here in this review article. And here, beyond genome editing, what is possible is that you can utilize base editors or prime editors, and you can achieve, still many of those desired traits in a very targeted fashion, as for example, herbicide tolerance in some of the crops where it has been demonstrated, like rapeseed mustard or maize, corn, soybean, watermelon, rice, and wheat, okay, which is important to increase the productivity or to protect them from the adverse impacts of the weeds. Many of those crop plants, when they irrigate them, they always got a problem of weeds. Farmers have to resort to labor's not available. So herbicide tolerance is catching up as a trait of interest even in India with many of the farmers. So altered nutrients from nutritional point of view is very, very important. Then rapid domestication, this is what I was referring to with an example in case of tomato. But here you see wild rice can be converted to cultivated rice, within 2 to 4 years using genome editing. Therefore, we call it as rapid domestication or accelerated domestication. Or making plants resistant to diseases, I give an example, or yield improvement, or 3 of them together, it could be done. You can mutate 3 different, regions in the genome, or 3 different methodologies could be utilized. And more importantly, today with regard to climate change, input use efficiency is very important, be it water or nutrients. Here I'm talking about nutrients when you try to increase their uptake or the transport from the soil, or roots to the upper part of the plants, and responsiveness of those plants to produce more with less amount of these nutrients in the soil. So therefore, you increase the input use efficiency. Farmers don't have to dump fertilizer as it's being done today, so that they can save more and it will actually, fertilizer is one again which is adding to the greenhouse gas emissions. Not only the fertilizer which is applied onto the soil on those annual agricultural crops, but also the factory where the synthetic fertilizers are manufactured. They're also contributing to greenhouse gas emissions. Therefore, it is important that we tackle all those issues and make the use of these genomic technology for climate smart agriculture. Like here, I was invited to write a small piece on it, and when I was of the view that you can incorporate tolerance to drought, heat, and soil salinity all together using genome editing, or enhanced spreading tolerance as well could be combined, and improve nutrient acquisition and use efficiency. For example, nitrogen, phosphorus, potassium, or even micronutrients is very important. Increase the radiation and use and photosynthetic efficiency. Not many people are talking about it actually in India. But this is something very, very important. How can we capture and utilize the energy which we're getting from the sun through the process of photosynthesis in a very efficient way? Actually, this is the most sluggish process with regard to efficiency we have in plants in general, and with regard to agriculture as well. And there's a lot of opportunity, therefore, how can you increase the efficiency of utilization of this light energy to convert it to chemical energy through the process of photosynthesis, and which is also very important. And this will help even in sequester more of carbon dioxide from the atmosphere, capture more of it, and sequester more of carbon back into the soil through the root system and through other mechanisms which are possible. Or of course, making the plant resistant to pests and pathogens, or through symbiotic nitrogen fixation, even in non-legumes, it is a possibility. But however, friends, also it is important, I'm sure students should know about it, that there is a regulatory hurdle, I may call it, okay, or a regulatory process through which all such genome edited crops or genetic engineering crops have to go through, okay. And in India, only BT cotton, as you know, has been commercialized. We have not been able to commercialize any of the crops so far, only because we have not been getting approval from Government of India of some of those important GM crops, or the guidelines still pending, to be approved for the commercial release of, or development and commercial release of genome edited crops in India. So message I'm trying to give here is, from my side as a scientist, I believe strongly that genome edited crops or even GM crops could be an integral component of self-reliant India, a slogan which is very important as given by our Honorable Prime Minister. And what I feel, and what scientists feel actually, we organized a roundtable discussion at the Academy, National Academy of Agricultural Sciences, wherein these messages were clear that these 2 categories of products coming out of genome editing process are without the foreign DNA, and therefore they need not be regulated, and therefore it will save a lot of time and energy and resources, and we'll be able to bring the products at a very early pace from the lab to the land. and therefore we need a positive and enabling policy, environment, which is more science-based, and therefore we can have more of relevant GM and gene-edited crops available on the farmer's field, which is very, very important. And I mentioned before, the sustainability is important, no doubt, but now profitability is also important of smallholder farmers, for meeting the sustainable development goals by 2030. So as I gave you the graph and showed the technologies, conventional breeding, GM crops, and genome editing, all are important. Remember, not only so far what India has been making progress, though we are making progress, there's no doubt about it. But I think in the wake of climate change, with the rising population, and with saving our own resources, and even producing more than what we are self-sufficient and exporting, to the other countries, and having a foreign exchange. So there's a lot of possibility that we go beyond conventional breeding and start using the benefits of GM technology, as well as more so now of genome editing technology for the welfare of the farmers, and of course the consumers and the country as a whole. Therefore, my message actually, to not only students, actually to all those who are even policymakers, is this: that we must invest more in basic sciences for bringing more innovations and develop more technologies for food security and economic growth of the country. And as students and as scientists, the message is we must try to harness the full genetic potential of genome editing. There's a very exciting opportunity at the moment available. In fact, in CRRI Cuttack, I mentioned about Dr. Kutub Mola, and we are trying to organize certain workshops, and therefore, because this awareness and communication of advances in science to the society is very, very important. And not only this, capacity building for researchers, students, and scientists is very important. So we have organized already 2 such workshops in the country in the last about a year or so. We are going to have one more in the next 2 to 3 months, which will be online, of course, mostly still by the time the country opens up thoroughly with regard to COVID situation. And so this is also important, not only that we develop these technology ourselves in the lab, but those of you who are beginners, and even for the society, for the farmers, for the consumers, it's important that we as scientists, even our students of science, must try to convey the advances which are already made in science globally, and what potential we have, and how can we utilize as youngsters, as students, that kind of technology for the benefit of our own country. And this is very, very important. And this is what exactly, while Kutub is working in his lab, I'm not, but then I'm playing a role here with other scientists, bringing them together, for the benefit of the scientists, researchers who are beginners, or for the students' benefit to take take this technology forward for all of us. So with this, thank you very much, and I'll be very happy to share, or answer some of the questions if you have.
I guess we have none, sir. Thank you so much for joining in. And that was all for the sessions here for today. But before we close down, let's celebrate that we have successfully completed the second day of our Agri Vision 2022. So we have this beautiful performance, which is an Odyssey number from Nrityanjali dance troupe. This would be on the Durga के 9 रूप के ऊपर ये performance है. Let's have all of them here, the performers, and post that we'll have the closing address. Yes. Let's have a round of applause for the performers. Thank you.
सिंहस्य परि दत्तवामचरण बिभ्रतामाना शुभा पुस्तका खड्गशूला दधति दक्ष करा देवता शारदा वाग्देवता शारदा वाग्देवता शारदा वासिनी दुष्ट विनाशिनी घर घर बातिनी हास्यरते गोपिनी वासिनी दुष्ट ভাবিনি হাজারতে ঘণ্টা বিনোদিনী ডম্বরুবাদিনী ঘণ্টা বিনোদিনী ডম্বরুবাদিনী রুদ্র বিলাসিনী রুদ্ররতে কুঙ্কুমমটিনী কঞ্জলোচনে অন্তরগামিনী গানরতে জয় জয় দুর্গতি তারিণী ভাবিনী জয় জয় দুর্গতি তারিণী ভাবিনী মালিনী পালিনী দাদিনী হে মালিনী পালিনী দাদিনী হে জনতা জন জনতা সত্যি জন জনতা জনতা জন জনতা সত্যি জন জনতা জনক 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 রাধে 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 হিঙ্গুಳ পাদিনী মঙ্গল নাদিনী চম্পক মণ্ডিনী ঘণ্টিনী হে হিঙ্গুಳ পাদিনী মঙ্গল নাদিনী চম্পক মণ্ডিনী ঘণ্টিনী হে ভূরি কুটুম্বিনী ভূরি বিবাদিনী ভূরি কুটুম্বিনী ভূরি বিবাদিনী চণ্ডা বিলাসিনী শঙ্করী হে চণ্ডা বিলাসিনী শঙ্করী হে আদিনী বাসিনী বিশ্ব নিবাসিনী বিশ্ব বিসর্জিনী সেবিনী হে জয় জয় দুর্গতি তারিণী ভাবিনী জয় জয় দুর্গতি তারিণী ভাবিনী মালিনী পালিনী দাদিনী হে মালিনী পালিনী দাদিনী হে ধারিণী দৈত্য নিবারিণী বিন্ধ বিলাসিনী বাজগতে শাস্ত্র বিচারিণী বাক্য বিচারিণী মন্ত্র বিচারিণী যোগরতে দৈত্য নিবারিণী বিলাসিনী বাক্যরতে শাস্ত্র বিচারিণী বাক্য বিচারিণী মন্ত্র বিচারিণী যোগরতে শঙ্কর সেবিনী সৌনক সেবিনী নারদ পদ সেবিনী ভাবগতে জয় জয় দুর্গতি তারিণী ভাবিনী জয় জয় দুর্গতি